There we go. Currently 25 meters off bottom. And coming in here, I get our first view of the seafloor.
Can already see a mixture of sediment and rock down there.
Right.
Well, Astrid, I can already see three fish maybe down there. We're still settling out. We're not ready to do any of our zooms and so on, but just alerting you in advance. Lots of ripples here. Tina 4 drifting through from left to right. There's another pelagic holotherian. I see a shrimp swimming.
Yeah, so we're looking sort of the southwest right now. That's the way D2 is facing. So you're seeing these ripples sort of in our field of view. So they're trending southwest northeast. So we can see the currents are moving orthogonal to those ripple directions, of course.
So Astrid, you'll have your hands full. We have the fish in camera 1, and we had another one in camera 2 flying above the D2.
Oh no, I just got away from the view, but I have you in my ear, so I can hear that I'm missing things. I'm running back now.
Okay. This is a fish that we have not seen before, so run faster.
Any other, want to take a
Don't
guess
hurt
out there? As Astrid sprints back to the EC.
Yeah, this is Chris.
Hi, Chris.
I'm calling from home, so I'm a little bit behind you guys. But I think this is a morid codling, if I'm not mistaken, but it's got really long filaments there on its pelvic fin. So it's a bit of a guess, but it's a really cool fish. I've never seen, I don't think I've ever seen this fish before.
We were seeing some codlings on the Jarvis Island slope dive, but they weren't this color, and I believe at that time Astrid was saying they come in a lot of different colors. The ones we were seeing were a lot more pink in color.
Yeah, it either has its dorsal fin down. A lot of the codlings have a really long first spine or dorsal filament on there. You can't really see that either. So I'm not really positive about what this fish is. My best guess, though, it's in the family Moridae, the codlings, but I don't know.
Well, we've got a really nice view of it. And I see it's passing over a whole lot of very small encrusting sponges on the rock. I'm just mentioning, just saying. I was also looking for parasites on the fish.
What?
Go ahead, Chris.
Yeah, you can see, I guess those are lateral line pores that are extending onto the head, if I'm not mistaken, those little white dots. But
Yep.
maybe Astrid's back already, and she can comment on this stuff.
So we're looking
Just
at
amazing video.
Yeah,
it's just beautiful.
Yeah, we're certainly going to be able to identify it with the video that we have. We're at a depth of 1,102 meters on Whaley seamount. Thank you, pilot. The fish may be about 20 to 25 centimeters in length. Oh, there's a sea pen right in the center just below the lasers. Oh, I didn't even notice that. There's another plexaurid coral on the rock. So Mike was asking the question about the difference between the animals and the community that we see on the hard surface we were expecting as we got higher versus the soft sediments, but here's a really nice contrast. So by the lasers, what we're going to zoom in on here is a kind of octocoral. They're colloquially called sea pens, and they are adapted to live in sediment. The base of the colony has an inflatable, what's called a peduncle, and it can inflate that and anchor itself in sediments. And in fact, some of them build kind of like a tube, so when they're disturbed, they can pull the whole colony down into the sediment. I don't know that this particular type can do that. There's still some debate on shore about what that fish was. So this will be a different a different sea pen for the expedition. I haven't seen this one before. You can see it's got a very stiff central rod going all the way up that main axis. And that's basically one giant polyp. And I think that may have been an arrowworm, a chaetognath that just swam by. So yeah, you see it's kind of brownish beige in color, and the tissue itself is transparent. So it's one giant polyp that has that rod, and it has the inflatable base. And then you see all these other polyps are called secondary polyps are arising from the primary one. And these would be the feeding polyps, the reproductive ones. So this is a really interesting structure. You can see they're coming out paired, but also two polyps pointed towards you. So that'll help us get an identification on this later on. Sea pens have a lot of variability in the way the polyps arise from that primary one. And this is really not one that I recognize. So I have to do a little bit of study on that. So this is an octocoral, and I guess I'll repeat myself. 8 tentacles. The tentacles are pinnate, kind of feather-like. That's a hallmark of all the octocoralia. That purple-pink that you're seeing in the inside, that's basically the pharynx. That's the throat. And then the stomach would be everything below that. Thank you, pilot.
Yeah. The other thing we can see here, Scott, are this sort of darker material on top of the sediments. We saw a lot of that in our dive just yesterday, and lots of people suggesting it might be fallout from a recent bloom up in the shallow water column. So we might be seeing this again here. There might have been a very extensive bloom.
Yeah, I agree. And my sense, I can't tell, but I think there's less. Actually, pilot, on the right side of the screen, you see there's something shiny white. Thank you. I recognize that coral there, so if we don't get a close look, that's okay. That's definitely a plexaurid sea fan, probably a Paramycia, and it had no phuroid, but I wasn't sure if this was a worm or a fish or just some shell. Looks like it's a, great. It's a hermit crab with a really,
wow.
Yeah, this is a tooth shell. I don't think we've ever seen any tooth shells, so this is a kind of mollusk, Dentalium. or tusk shell is another name for it. And typically these shells are buried in the sediment so that the pointy end, the thin end, is sticking up just out of the sediment, and then the bottom end is stuck deep in the sediment, and the mollusk related to a snail has all these little tentacle-like things that it uses to feed in the sediment. It's a deposit feeder. But here, a crab has taken over that shell
I don't
and
know.
is using it for itself.
He might have made a bad choice. It's hard
I
to
don't
drag
know.
that around. Huh?
This is definitely a style choice. Look at the size of that thing. That is well done, Mr. Crab. So that's a pagurid hermit crab, and you can see it's got 3 pairs of legs sticking out there. It's a decapod, like all the other crabs and shrimps that you're familiar with. The last 2 pairs of legs, that would equal the 10, are very highly reduced and are used for holding the shell onto its back. That's good for science. If we go over the rock again, Pilot, there were some very small sponges encrusting it, and we've had a request from shore to have a look at those if we get an opportunity. Les Watling at the University of Hawaii is chiming in the chat room that that was a very strange sea pen. Tina Molitsova believes these may be what are called polyp leaves. Was somebody on shore going to say something?
Oh, I was going to second that. That was bizarre sea pen. I think has all of us scratching our heads.
So we're going into the rock to have a look at the sponges. So there I can see some. And there's also, excuse me, another snail. You can see it's a very long siphon extending out ahead of it. That's what's sucking in the water. And so you see these gray-blue sort of circular encrusting things. These are sponges that are on the rock. They're really tiny. So sometimes, Bill, as you know, when we get a rock sample, I'm sort of combing over the rock. These are the sorts of things that I'm looking for to see if we can find these really small sponges. There you go, Shirley.
Great. Thanks, Scott. Thanks, Pilot. And yeah, Chris, you have any idea what these might be?
Not a clue. Looks like you got a bunch of these. You got a gastropod. I'm a bit delayed because I'm at home. This I'm not on the internet too. But I assume that they're demisponges. Because usually these very small, kind of round ones that hang out on the underside of rocks and things are typically demisponges, but I don't know.
Okay, I better go back into Sea Spread and correct that.
Well, they could be laugh sponges too, Shirley, because, yeah, I just don't know about those, because you get plenty of glass sponges that are small as well. And as I recall, there was one that came up with a rock sample last year, and when I first looked at it, I thought it was going to be a demisponge, and when I got it under the, it was not. It was a hexactinellid, so could be completely wrong.
We have 2 crinoids next to one another here, one of the stalked variety and one of what's called the comatulid, or the feather star variety that doesn't have a stalk. The stalk ones are basically affixed in place, whereas the other ones are free living and they can swim around. So you see the stalked one is very small, he's on the right, and the feather star is larger and on the left. If we have the opportunity, sure. Thank you. There's a smaller one on the rock. Oh, there's a fish. This is a different fish. Maybe we should get that fish first. Thank you. Looks almost like a porcupine fish from a distance. It's a sea toad or something.
Huh.
Go for it, Astrid.
Wow. Yeah, I don't know. It looks like a Lophididae, so like a similar to those other fish that we've imaged before. But maybe that would have been a goosefish. But this is shallower than I'm used to.
It's not one that I recognize, I will tell you that. Very small mouth gape.
Give me a second, guys. I think I could come up with a tentative ID on this, at least to family or genus. This could be Halucia is one option for it. It's I'm pretty sure it's an Octa or a batfish.
A
So it
batfish.
could be in the genus Volusia. It could also be in this other weird genus that Bruce Munday had identified first in the Northwest Hawaiian Islands in 2015. But I'll take a quick look in my guide and see if I can figure it out.
Thanks, Chris. So possible batfish. One thing I'm noting, so we saw a lot of the goosefish on the Jarvis Island slope, and
Right.
they had a really wide mouth, and they had a lure on the head. So they were a type of anglerfish, and I'm not seeing that with this one. It has a much smaller gape to the mouth. It's not facing upward, and it doesn't have that lure. It still has these modified fins that are kind of like legs. But from a non-fish biologist, those are the differences that I'm noting.
Yeah, this one seems to be moving around a little bit more than those goosefish. I don't think we ever saw one of those swim.
A very
when we
good
came up
point.
to it. Yeah,
Yep.
this one.
They're stealth predators.
Yeah, this one's a little more agile.
I also noticed when it sat down, there were a couple of tubes that were sticking out of the sediments and curled over. Those would be polychaete worm tubes. They build these tubes, and then they can stick their heads out. And I also saw, as we were moving on the right side, a cup coral. So we've got nice diversity down here. So we're just getting a quick
So
look.
I've got
Stand by, Chris. We're just getting a quick look to show you the difference between a free-living feather star crinoid on the left and a stalked crinoid, which is also called a sea lily, on the right. They're both related to each other, both in the class Crinoidea, but you see they have very different morphologies, have different ways of life. Thank you. So when video's good, pilot, we're good. Go ahead, Chris.
I'm looking at some pictures of batfishes that we have imaged in previous years, both Okeanos and the Hurl submersibles. And yeah, I'm 99% certain that that's going to be an Octa or a batfish. Now, which genus it is in question. It could be something in the genus Solosisquama. I just learned about that fish in 2015 when we imaged it for the first time with an Okeanos dive up in Papahānaumokuakea Marine National Monument. But anyway, very cool. You don't see them very often, so that's a great start. Those two first fishes are just tremendous. Beautiful finds.
Yeah, great, and we got some wonderful imagery of them. We're settling in now to get a closer look at this snail. So already we've seen two snails, which we don't see too often, certainly in the deeper dives. And this one is facing head down. You can see its soft, muscular foot trailing below the shell. And this one has its siphon more or less retracted, so it's only out a very short distance. Let's see if we can see the foot move at all. It doesn't look to be moving, does it?
No.
Looks like a couple of amphipods just in front of it. Actually, those little brown sticks that you see, I believe those are mud sticks that are built by that little amphipod. It's a little small crustacean you can see just to the left of the shrimp. It's kind of moving back and forth. They build those mud sticks, and I'm presuming they build them so they have something to hang on to when they walk up into or when they hang out higher in the water column. A real continuing theme you see among so many different animals that take advantage of moving water flow to capture their food, and so are looking to get off the bottom. Whatever little advantage you can get.
Yeah, well, if you're that small, every little bump on that rock is a shielding you from the currents, I guess.
Exactly. Tina Molitsova from the Shirshov Institute in Russia, who's spending a very late night watching the beginning of our dive, is suggesting that sea pen that we were looking at before that we found unusual. I wasn't able to identify it, and a couple of colleagues weren't able to identify it. She says it may be the genus Chunella, and she says she's never seen that alive, so she's making a tentative ID. So that was kind of an exciting find. It was something very new to us. You see, I do like the soft sediments too.
Just different things.
They're just different things. It's nice to see difference. There's a couple of stick-like things. Let's see. Okay, we can go on the shrimp. Looks like a yeah. Sure. There's a coral on the far left, but there's also one almost directly above the lasers, slightly left of lasers. They may be dead, but I just want to verify as we're going that direction. It's above the sand patch. There's a rock almost near the top of the screen. Yep, no problem. Yeah, if the closer one is easier to get off the screen to the left. Yep, it's the white one that I'm interested in. We've seen that yellow one. That's the Parameciid sea fan, possible genus Parameciia. But I'm looking at this white one. This is probably going to be in the family Primnoidae, and I will make a prediction right now that it's in the genus Narella. Narella is one of the largest genera in that family, and by that I mean it's got a lot of species, a lot of different species described, very diverse in the Pacific. And that's almost assuredly what this is. And Chris Kelly, who's seen a lot of these, when your video catches up, please offer your opinion. So one of the things we look at when we see these polyps, you can see how rough they are. If you looked at them under a microscope, what you would see is a series of plates or scales that overlap one another to differing degrees, and they have different shapes, and that would help us in the taxonomy and the naming. Also, the number of polyps that are arising in those rings, or whether there are rings or not. And then, to some degree, but not quite as much, the branching structure. You can see coiled around the branch there the arms of a brittle star. There's also a little tiny shrimp. I can see his orange eyes peeking out right in the center there. But these brittle stars, very common on the corals. You can see two corals next to each other. They both have a brittle star in the colony, and we believe there's an association between the brittle star and the coral such that the brittle star may be clearing off things that land on the coral. Doesn't seem to be eating, these kinds don't seem to be eating the coral themselves, but there are others that we may see that do feed on the coral. So lots of these yellow Parameciia sea fans. I've now seen at least 10 of these. And you note that they're all oriented in the same direction. They seem to be cross-slope.
Right, and parallel to the ripples.
Exactly, that was where I was going to go next. So I think both the ripples and the orientation of the sea fan are telling us something about the average current flow here.
Right. And as we saw on our dive the other day, and we were discussing yesterday that with tidal currents, these currents can reverse back and forth. But yeah, it seems from the ripples here, we definitely have a kind of a cross view, the northwest southeast direction.
Looks like we have a tall stalked sea pen here on the left side, really tall. I said sea pen, excuse me, I meant crinoid.
Crinoid, yep.
So we were just looking at a very small stalked crinoid a short time ago. There's a really tall one. It looks like the one we may have collected off of the Jarvis Island slope to me from a distance.
And Del, based on the size of these ripples, the spacing between troughs and their top of the wave here, we can then go ahead and backtrack and calculate the amount of current that's flowing on the bottom. Especially if we know the grain size, we can talk about sort of the shear stress that's acting on it.
Yeah, we could. It's certainly possible that we could back that out. Little out of my wheelhouse, but we can certainly see these are very symmetric ripples. So
sure.
Imagine the current perhaps is moving back and forth. We've seen more asymmetric ripples in some areas.
Well, looks like an eel-like fish. Astrid, if you'd like to comment. Wonder if this one has just eaten recently.
I'm going to hold out until we get close. All right, I'm going to go with a Xenophys eel, a cutthroat eel, but this is a really deep-bodied one that looks really fat.
Yeah, I was wondering if it had recently eaten a big meal or if that could be some indication that it's pregnant. I'm not sure. I don't know enough about the biology of these fish.
So if we could try to get a shot of, if he cooperates, of course, of the dorsal fin insertion and the anal fin insertion, that's usually what we can use to try to tell these guys apart.
And Pilot, don't go too far because I'm seeing several interesting crabs here we'd like to get a close-up of. Thank you.
Yeah, I agree.
Scott, that might have been an Anelopsamia, that white colony that was over on the left, too.
Okay, we'll check that in a second. So just below the lasers now, you go this, bingo. You just passed over it on the face of that rock. So I've seen 3 or 4 of these squat lobster crabs right in this area, but this one looks like it's in a good position and is a fairly large size. And one of the things you'll note is how long the chelipeds are. So that's a specialized name for those legs that have the pincers on the end of them. So we have Tim Shank from the Woods Hole Oceanographic Institution, who's been spending a lot of time looking at these sorts of crabs. There we have 2 of them next to one another, and another snail. This might be a different kind of snail. Looks to me like the shell is much more knobby. You can see there's a lot of hair-like structures on those arms. Those are called setae. And at least in some of these crabs, the setae may catch some of the particulates that are floating in the water column, or even bacteria, and it may be that they could feed right off of the stuff that's getting caught in their setae. I don't know that that's the case in these crabs. Tim, you have any comments or identification? Or Les Watling, who also knows a lot about our crustacean friends. Del, I get the sense it's going to be one of those dives that's busy for biology.
Yeah, we've certainly seen a lot of stuff. I mean,
so Tim Shank says that looks like a chirostylid crab, and we've seen many, many chirostylid squat lobsters associated with corals. So these look like they're a form that is not associated with corals. Thank you, Pilot. That's great. And if we have the time, as you're going back up, there was a yellow colony, and to its left, there is a bright white one on the edge of a rock. We want a look at the white one. There it is. Yeah, this may be just a, can't tell if it's a dead skeleton, but Chris had suggested that perhaps it was Anelopsamia. That would be a stony coral. Right now it's looking more like it's a white morph bubblegum coral. Yeah, I think that's what we have here. So this to me looks like a Paragorgiid of some kind. That's a family name, Paragorgiidae, bubblegum corals, and we've seen several that are kind of pink in color. And those swellings, the polyps right now are retracted, would be inside those swellings. So you can see the yellow, the yellow colony to the right is another octocoral, and you can see all the polyps are extended. You can clearly see the tentacles. They could all withdraw, and so that colony would kind of look smooth. Not quite to the degree that the white one does, but a similar sort of thing. And at the base of the colony, there's actually 2 of them here. You can see the holdfasts where it attaches to the rock. At the base of the upper one, you see a worm-like animal. That is an aplacophorine mollusk. It is a specialized kind of mollusk that feeds on corals. Thank you, Pilot.
So Scott, I'm gonna be the devil's advocate here. I'm actually thinking this could be Paracorallium kishinoi with its polyps retracted, because it's going to have those kind of round ends as well, and that skeleton looks pretty firm. The kishinoi looks like that with the polyps retracted as well. So that's very hard to tell. To me, it could be either a Paragorgia or Paracorallium kishinoi.
Okay, great. Thank you, Chris. So that was Chris Kelly noting that there is another group of corals, the Paracorallium kishinui. It's actually one genus, one species, that has an appearance that could fool you. And if we're still in position, can we go right back onto that rock? There was something on the rock. Yep, thank you. Right above the lasers. Either a Chrysogorgia or a bramble bamboo. So there, it's just on the left side of the screen. That's a Chrysogorgia, a really tiny one. Not the white thing. There we go. A really tiny Chrysogorgia coral. Thank you. We're not going to be able to identify that, but now we know it's there.
so small it doesn't have a squat lobster yet.
There you go. Well done. That is true. So that's actually a really interesting observation. At what point is the coral of an effective size that it is the habitat that the crab wants? So nicely done. This is kind of interesting.
Yeah, so I've been looking at the rocks here, trying to get a sense of what it is. You know, the surface that we're seeing is this manganese crust, it looks like, this kind of dark color. And that can make it a little tricky. I've been trying to see if I see some textures that look like flows or collapses, or if this is more carbonate material. And it's still a little unclear what this is.
I have the impression there at the bottom that there were these sort of blown out, not caverns, but it'd kind of be like a large tube that I'd seen broken just from one edge.
Yeah, you see that in places. In other places, maybe it looks a little more reefy. So we'll have to go along here and we'll see if we can. If anyone else has an opinion or have any geologists
Pilot, can we get a look at the
out there?
brownish
Please.
coral on the right, please? This is going to be another. Go ahead, Pilot. Copy. Understood. This is another primnoid coral, but it's a different species from the one that we saw earlier. You can see the coloration is a little bit different. It's what we call a biflabellate fan, meaning it's a fan, but there's actually two fans kind of together, one in front of the other. Here we see the polyps are in rings of 1, 2, 3, 4, at least 4, maybe 5, and there's kind of oriented downwards somewhat.
Yeah.
So all of those are important characters. Ultimately, as I say, you would need a collection and look under a microscope at some of the other characteristics. But this is certainly helpful.
So Scott, did we collect something like this on one of your earlier dives, or am I thinking of the last cruise? Because I know we collected, I think, this species because nobody knew what it was.
Okay.
And it's some type of primnoid, as you said.
Sure. We did not collect any primnoids on this leg yet, but I do have queued up all the collections from the previous leg. So let me have a look at my primnoid folder here. And sure enough, it looks like on March the 9th, we collected this. So good call, Chris. Good memory.
Yeah.
Thank you, Pilot.
Yeah, it'll be fascinating. Yeah, because I think you're going to see some of those up near Palmyra and Kingman too. So that was a great collection when that happened back in March. Figure out what this thing is. We didn't know what it was in 2005 when the Pisces submersibles were down in Palmyra. All we could do was go to primnoid. So this seems to be a fairly common or reasonably common
Quick zoom on
species
sponge.
down here in the Line Islands.
So, Chris, this may be a Regardrella. Am I learning something? And after we pull out Astrid, it looked like there was a rat tail behind it.
I think you're learning something, Scott.
Wow, it's amazing.
This is
So that was a glass sponge we were just looking at there. Go ahead, Chris.
I was going to say that sponge had an unusual amount of ribbing, but now I can see we've got a pretty interesting fish, so maybe Astrid could take over the discussion.
Stand by,
Yeah,
Pilot.
so we definitely have a rat tail. This is the Macurrid, family Macurridae. These guys are notoriously tricky to ID. So any good shots would be good, especially of the head. But we can see it had kind of a large eye and a more pointed snout than I think we've seen. Well, no, I changed my mind. I think it's a more rounded snout. So could be Nizumia, but hold on, let me look at my rat tails one more time.
Okay, thank you, Pilot. Astrid has a barbel. I don't know if a chin barbel. I don't know if that matters. This one is clear of parasites.
Yeah.
We can move on, Pilot. Thank you.
Yeah,
When video's
Astrid,
ready. Go ahead.
I'll throw out a guess, Astrid. I think this could be Coryphaenoides, and I'm not sure which one, but you see the way that filament's coming off of the pelvic fin and the sloped head and kind of that coppery kind of scaling. Anyway, that would be my guess, is Coryphaenoides.
See a lot of these plexaurid fans, the yellow fans, and then as well the primnoids we thought perhaps would be the genus Narella. Those are the white ones, and you can see very clearly in both of these there are pink masses in the center of these fans, and those are ophioids. There we have one of the maybe coralliferous sea stars. We'll have a look at this. So this is our first sea star of the day, and I'm noting how fat it is, and it does appear to be up on a coral. So coralliferous would imply that it's a coral feeder.
Okay.
So this is a predator. Yeah, so the horror. So now what you can see is a cleared skeleton. So you can clearly see that these corals have an internal protein-based skeleton. This is another primnoid, and you can see many of these branches are bare, so that sea star has been feeding on this colony for some time. And as we come around, this is the kind of sea star, I believe it's a hipposterid in the family Hipposteria, that can evert their stomach out of their mouth and so begin digestion outside of their body. And sure enough, I can already see what looks like lips. Those are not lips, folks. That is the pyloric stomach. You can see the tentacles are not out on this coral. All these polyps are kind of retracted, hoping that they get missed. You can also see the tube feet, so it's a little bit hard to see the stomach, but you can see the stomach. So you see all the tube feet kind of hanging onto the skeleton, and then that sort of loby stuff in there is the stomach of the sea star, and you can see how good a job it does at digesting the polyps off of that skeleton.
Cleaned it all the way up there. Huh? Wow.
You know, Scott, if we could do that, we wouldn't have any need for forks and spoons or anything ourselves.
I'm sorry, I didn't get the last. We wouldn't have any need for what?
Silverware, forks and spoons.
There you go.
If we were able to do that, that's kind of a disgusting vision, isn't it?
Yeah, that'd be interesting at the table. There's also a couple of brittle stars, 3 brittle stars hanging onto the coral here. 2 of them you see have very spiny arms. The 2 that are on the right, there's actually 3. There's a 3rd one at the top, a 4th one. But there was also one of the astroscumatids, which have the smooth arms further off to the left. Copy that, we are. So very interesting. Les Watling was just noticing in the chat room, well, if there's no bamboos to eat, and he's saying that because we often see those sea stars on bamboo corals, and here it was on a primnoid coral, and they just have all those plates, so apparently the plates aren't doing too good a job of protecting that. So this is an astrid, what we saw earlier. We were wondering if it was a codling of some kind.
Wow, this is a really impressive fish. Cool. I don't, maybe a codling because, well, the tail looks a little strange for a codling. Yeah, but you can see a pronounced lateral line. Those are those white pores, and it has more pores up over its head. You can see those really extended filaments. Really interesting fish. Nice shot. Thank you.
Thank you, pilot.
That's Astrid's way of saying that she doesn't have a clue like the rest of us.
Yeah, that's right, Chris. Thank you. Thank you for that.
It's called out. Uh-oh.
Like I said, like the rest of us.
Like the rest of us,
Nobody knows what this is.
I think we saw this the other day. You're talking about in camera 2. I think it was some kind of scorpion fish, if I recall, that holds its fins out and head up. I mean, I'm not 100% certain. That's what I'm looking at. But here, let me switch cameras. Oh, he's moved. Okay. We just also passed over an echinotherid urchin, a pancake urchin. And there's another one of these primnoids that we were imaging before, biflabellate. So the 2 fans kind of are pressed together. And we had made a collection of one of those primnoids on one of the past legs this year. Yeah, I'm seeing some purple. Looks like some of the purple mat or ribbon-like octocoral growing on the side of the rock. And sure enough, it may be overgrowing. Yeah, maybe overgrowing a skeleton. So this is probably the genus Clavularia, what we like to call the royal purple octocoral. Or I do. I just made that name up. Not just now. I mean, I made it up in the past, so if that means anything. Just this beautiful deep purple color. I'm not sure where that comes from. Something in its diet gets those pigments. But it's growing kind of like many of the zoanthids that we see that overgrow existing structures. That's what this is doing.
Okay.
Nope, this is good. Sometimes we see it as a series of ribbons over the rock. Sometimes we see it as a mat. And then sometimes, as here, it's overgrowing a skeleton of a coral that was living there at one point and is now dead. So Les Watling had written in the chat room, so much for the idea of sclerites being a protective device. So I've noted that the primnoids are really interesting because they have all these really elaborate scales, and it's kind of like a suit of armor. And one of the ideas is it's protecting them from predators. Well, apparently it was not protecting it from that sea star. Maybe it protects it from smaller predators like the aplacophron worm-like mollusk. A lot of these plexaurid fans. Now, there was interesting. So if we could have a look at the yellow fan at the top of the rock, just to the left of center. Excuse me, right of center. Right of center. All right, there we go. Number one of the day. Who's keeping score? I see there's also a bottlebrush chrysogorgia below it. But the reason I'm asking to look at this, what do you think, Del?
A squat lobster.
Nope.
No?
It's the absence of a brittle star.
Oh.
So I'm wondering.
That one, not the chrysogorgia. Okay. Yes.
Oh, yeah, very good. Yes,
Sorry.
no, you're right. There's going to be a brittle star in there. I mean, a squat lobster and a chrysogorgia. We know that.
Right. That's our other association, though. A brittle star.
But all of these other fans have had an ophiuroid, and I don't see one here, so I wanted to verify that it does or doesn't. And if it doesn't, why? Is this something different? And it doesn't.
No.
So it's got Ophiocanthid brittle star. These are a different kind of brittle star, although the one here on the foreground on the right, this might be similar, but why isn't there a really large one there? So I do think this may be different. Yeah, this looks to me like a different kind of yellow sea fan than the Paramaricia that we were seeing earlier. I don't have an identification for you, but one of the things that's so instructive is that this is telling you that the species of coral matters to the brittle star. The brittle star isn't just going to go and sit on any kind of coral. It does have a preference, and so presumably there's some kind of chemical attraction, some kind of detection that they use to determine, yep, this is the species that I associate with. Now, there is a brittle star there. It looks different from, I'm talking about the one in the right foreground. It'd be interesting to know how that differs from some of the others. Thank you, Pilot. And let's get a look at the chrysogorgia that's just in front, the bottle brush. There we go. All right, Del, tell us about what's in those branches.
I think I can see a squat lobster in there.
Me too. So earlier we were looking at those squat lobsters that had the really hairy arms. And one of the scientists suggested that looked like the Yeti crab. There's a squat lobster that's seen at hydrothermal vents that is called the Yeti crab. And these chirostylids are very closely related to it. And I noted that there are some chirostylids, actually most of the chirostylids I'm aware of, are adapted to live in corals, but not all of them. So here's a coral specialist. And you can kind of see the golden color of the skeleton. These are Chrysogorgiidae, translates to the golden corals. Thank you, Pilot. You can also see, so that sea fan that's just going out on the left, it's a little bit different color as well. It's a little bit deeper orange yellow versus that yellow one we were just imaging that didn't have that large astroschematid brittle star. And there's another Narella, genus Narella, family Primnoidae. So one thing, if you were to go snorkeling or go to the seashore and look at the rocks there, you typically see all the rocks are just covered with biology. There's no space. And one of the things we talk about is space limitation. Let's have a look at the Iridogorgia top left. We have space limitation, so there's competition among animals just to get a hold of a rock. That's clearly not the case here in most of the deep sea. You know, that beautiful pedestal we saw the other day was
the exception.
Was the exception.
Yeah.
But so there are other things that are controlling the abundance of animals. It's not the lack of space to attach to. This is there's also very interesting medusae just below it on the rock. I believe those are Corallomorpharians, which would be a first for this cruise. But the colony that you're looking at is genus Iridogorgia. This is in the family Chrysogorgiidae, so just like that Chrysogorgia we saw with the squat lobster. It's got this beautiful spiraling morphology with these single branches coming off. And Les Watling, who's in the chat room, University of Hawaii, has written some species descriptions on this group and knows this group well, and perhaps he can suggest what species that might be. But let's have a look at these things that look like sea anemones.
That would have been what I would have said. But.
Right, but I think they may be Corallomorpharians, and they're difficult to tell apart. The Corallomorpharia is actually an order. That's a taxonomic level, an order within Hexacoralia, and the Actiniaria, those are the true sea anemones, are a different order. And so that tells you how different they are. And people describe these as naked stony corals. They have a polyp structure much like a stony coral, but they have no skeleton. And the way I'm using to identify them are two things. One, the white dots at the tips of the tentacles. Some anemones have that, but Corallomorpharians typically do. But also they're very diaphanous looking. They're so transparent, and they look very wispy.
Right.
And that's what I'm used to seeing on Corallomorpharians. And you can kind of see a white slit in the one that's on the right. That's the mouth, and maybe we can get a zoom in and see if we can see the internal part of the body. And I'm also noting all these waving white sticks, the side branches. These are carnivorous corals. Carnivorous sponges. So I'm sure Shirley is very interested in that, and she wants to say something.
Yeah, I'm taking a close look at these right now. These are beautiful. Wow. So this is a demo sponge. These are in the order Peopleiflorida in the family Cladorhizidae, and they're adapted for life in the deep sea, where there may not be that much a small particulate matter for them to filter food. They're adapted so that they can actually capture prey on their surfaces. The prey get trapped in the spicules and small microscopic skeletal elements on the surface of the sponge, and then the cells of the sponge engulf the prey and gradually digest it over time.
So, Shirley, what kind of things would it be catching?
Usually small crustaceans.
You can see just to the left of... Sorry, sir.
They'll digest all of the soft parts, and then kind of looks like it kind of spits out the exoskeleton.
Sorry, Shirley. I was just noting that to the left, there's these brown waving sticks, and those are Amphipod Mud Sticks, and on those sticks are little amphipod small crustaceans. And the reason I was pointing out is that could be a meal for these sponges. So it's these small sort of crustaceans and other things like that that can get hooked by those hook-like spicules that Shirley was talking about. And then the sponge can actually reorganize its cells. The cells literally move, cover up that animal that is caught, and then digest it.
Wow.
Yeah, fascinating. That's a nice little tableau of diversity. So just to go back to the Corallomorpharians for a second, so each one is a polyp. So when we keep talking about polyps in the coral colony, we're essentially talking about these sorts of things with the tentacles and the mouth. And it's just that on the coral colonies and the zoanthids, all those polyps remain connected to one another. Thank you, pilot. Beautiful rock surface. So Les Watling commenting on that Iridogorgia. And Les, I'm thinking that maybe Iridogorgia magnispiralis. Please confirm. He's saying that this is pretty shallow for that species, for these Iridogorgia. We usually see them much deeper, so that's kind of an interesting observation. You can see some nematocarcinid shrimp. These are the thread-legged shrimp, deep red color, walking on the rocks. Occasional feather star crinoid, and again, lots of these sort of orangey Paramaricia-like sea fans that have ophiuroid brittle stars in their branches, and then the white-pink primnoids. So good diversity of sea fans down there. We haven't seen a bamboo coral yet, have we?
No, we haven't.
Yeah, okay.
No.
So far, I mean, we haven't been down very long, but so far we've got a good abundance of the plexaurid sea fans, which isn't unusual when we're getting to these shallower depths, and a good abundance of the primnoids, family Primnoidae, at least 2 different species. And we've seen Chrysogorgia and Iridogorgia. And we saw the possible Paragorgiidae or Coralliidae, that white one. Is this, I'm not sure if that's a loose rock, Del, on the sediments there.
Let's take a look here, I guess.
Just in the middle of this sediment chute. Are you interested for one here, or do you want to be on the cone?
I'd like to get one that's a little more angular when we get up a little bit on the cone, probably.
Okay.
So yeah, I probably don't want to collect here.
Maybe pilot just on the right side, Beageous colony, just almost due right of the lasers. Yeah, that one. Yep, thank you. Is this an okay? Yeah, I want to find out if it's a scleractinian or not. Yeah, great, perfect. So this is a scleractinian. This is a Stoney Coral. And this is what Chris thought he may have seen earlier when we were looking at that possible Paracorallium. And there's a hermit crab on it. Interesting nubby texture to the skeleton. So I'm not sure. This is a kind of Lophelia-like colony. I'll have to check. I'm not so good with my Stoney Corals. But Chris, if you're available close to the phone, maybe you can give me an idea on this one.
Yeah, so definitely I totally agree. It's a Stoney Coral. I don't know. I see what you're saying. It's not quite right for Analopsamia, I don't think, but I'm not sure why.
It's not smooth enough and bright enough for Analopsamia. That's good,
yeah,
pilot.
All of the polyps are coming off of one side of it. I
Yep.
think the other side is going to be, and so that is Analopsamia-like characteristic.
Thanks, Chris.
Yeah, you know, and yeah, I have no idea, but definitely a hard coral, and that's the first one I think you've seen, right?
And here we have, oh geez, yeah, let's get a close look. I was doing all my studying of the different sea stars yesterday.
Give you a hint, Scott. It starts with A.
Oh, that's not where I was going. It's the what is it? The Astropectins or something, or the Asterinids?
No, but no. I'm taking a guess here too from a distance, but given the number of arms and the general appearance of it, I haven't quite seen the zoom in yet. Okay, yep, yeah, that's what I was thinking it was. It's Actinax. It's Actinax. I can't say this. Actinaxis.
As long as you don't expect me to repeat it. Actinaster, I believe you said. You can see a lot of.
And whether it's Actinaxis separaceus or not, I'm not really sure, but I think it's in the family Solastrid or something like that. I'm going to go check the gallery. I've forgotten what family it's in.
That's good, Chris. At least that's what I was going to suggest. It was one of the Solastrids, so kind of like the sun stars. You can see a whole bunch of spines that seem inflated. They have tissue around it. And I don't know if that means that there may be some toxin associated with those little sacs, or if those are simply ampullae that are there for gas exchange. Thank you. That's a wonderful image.
Yeah, and this is in the family Mixasteridae, so the correct pronunciation is Astyanaxis. That's how you pronounce it.
All right, so it's a Mixaster. Thank you. Tina Molitsova was suggesting that earlier Stoney Coral we saw maybe Selenosmilla. Thank you, Tina. Trying to see if these white things on the right are sponges or if it's a base of a coral now gone. I think it's a sponge. Looks like there's a very long stalk and multiple heads to the sponge or something. Yeah. So is this some kind of Caulophacus, Chris? It looks like two heads have merged almost.
Let's see. Okay, now I'm getting a look at this. Wow, this is a weird one.
Yeah, isn't it weird?
Yeah, this is really weird. I think you're correct, Scott. I think it is some type of Caulophacus, but very bizarre looking. There is one group, one subgenera in the Caulophacus that has multiple puffballs on the stalk. This looks like it's got at least two, maybe even three of them, and two of them may have merged together, I think, as you were pointing out. Very, very bizarre sponge. Never quite seen anything like it.
Thanks. So I should point out that this is a glass sponge, and that long brown rod or stalk that you're seeing are actually a mass of very elongated spicules. These are made of silica, hence the glass part, although all sponges have silica spicules, but they're specially modified in this group of glass sponges. And you can see it kind of attaches to the rock. It's kind of glued to the rock there. And then the feeding part are these globular structures on the end of the stalk. And it's an aquiferous form of feeding. They have cells inside that generate current flow to pull water in, pull particles out of that water, and then eject it back out again.
So this more wispy kind of brown stuff around the center head, that's part of the sponge, or something growing on it?
Exactly, that's something growing on it. So we'd refer to it in general as epifauna or associates. Those are hydroids, hydrozoans. It's a kind of cnidarian, so related to the jellyfish. Actually related to the siphonophores that we see in the water column. So these are the benthic form of hydroids.
I see.
And they're fouling organisms, so you can pretty much go down to any dock or pier piling in the world, in the ocean, and have a look, and you'll probably see hydroid colonies overgrowing it. They're kind of a pest. Some more of these primnoid golden, or what color would you call that?
Brown.
All right.
You can call it golden if you want to sound fancy.
I'm going brown. So it's more of those biflabellate brown primnoid colonies. Looks like it's leveling out a bit here.
Yeah.
Looks like a fish that's head up. Yeah, you see that? Thank you.
Oh, here we go.
Yep. I think I recognize this one. I'll wait for Astrid. Yeah, what's this thing?
Go for it, Scott.
Oh, nice. Sorry, we got distracted by a nice little hydromedusa there. Actually, maybe could be a Scypha medusa. Anyway, it's a jellyfish. Just trying to figure out what group it belongs to. That's beautiful. So you're looking up into the bell, and the mouth would be on the inside of the bell. So you'd basically be looking right into the mouth right now if you're a little crustacean. Might get a little worried. You can see the tentacles dangling down. You can see they're kind of swollen on the end. I heard Amanda say the other day when we were doing our midwater transect that there are many of these hydromedusa have bioluminescent structures at the tips of these arms that they use to attract prey. So kind of dangling them. And
clever, yeah.
Yeah, very clever. Nice color the way it's reflecting the light from the ROV. It's beautiful. So Astrid, my guess on that was going to be a halosaur. Is that close?
that was going to be my guess. I was going to the Halosaurus page right now to look them up, see if we can get it further down than that, but we'll see.
So let's go back to this Hydromedusa again, because we got this beautiful coloration, and I think what we're seeing is a refraction from the light. However, there are some of these Hydromedusa that are capable of bioluminescence, and there is a green fluorescent protein. So I wonder if this is refraction or if this is actually being light generated by the Hydromedusa. Beautiful. This is small, very small animal. Yeah, the way those colors disappear make me think that that's just a refraction from the light. Thank you. Yeah.
So that halosaur might have been in the genus Halosaurus, but I'm going to be tentative on the genus, but stick with Halosauridae.
Astrid, what's the common name for the halosaurs, or is there one?
I actually don't know if there's a common name. Chris, do you know?
Like a little midwater fish drifting by.
Sorry, I couldn't get my mute off my cell phone. Halosaur, that's the common name. I guess salt lizard is what the actual Latin roots mean. So, but I don't think anyone calls them salt lizards. They just call them halosaurs.
So nothing like duck-billed eel or anything like that. That's a totally different sort of fish.
Right.
Okay.
So the soar is lizard. Anyone who knows anything about dinosaurs, right? The terrible lizard. But the halo refers to salt. And so Bruce Mundy has given us some stories about the origin of some of these names. And I think it was last year he was talking about this one. And it kind of stuck with me. I never really thought of halosaurs being salt lizards before, so that was pretty interesting.
Yep, I think it's actually a black coral, so that'll be new for the day. Thank you. Yep, good call. So we may have our first black coral of the day. And I wonder if Tina Molitsova is still awake in Russia. She is a specialist on the black corals. And this is also, yep, it's got a chirostylid squat lobster in its branches, 2 of them, yep. So one of the ways, I'm going to wait, tell you a little bit while I wait to see if Tina can give us an identification. But one of the ways that we know that this is a black coral as we're zooming in is that the polyps are very, very small. You see that the tentacles themselves are smooth. They don't have the side branching pinnules. They don't look feather-like. And they also have only 6 tentacles. Although in this case, in this particular family of black coral, the polyp is a weird structure. And instead of being round like a sea anemone, it's been stretched out along the branch. So you actually have to count 3 pairs of tentacles to know where a polyp begins and ends. But the other clue is that in the center you'd have one raised portion, which is where the mouth would be.
Okay.
But you can see the tissue is very, very thin. You can see the darker skeleton through it. It's the skeleton that gives it the name black coral. And there are a few species that have these chirostylid squat lobsters that are specialized to live with it. So this may be, I still haven't seen anything from Tina, but this may be the genus Trisopathes. All of the black corals, I've noted this before, but all the black corals belong to the order Antipatheria, and that translates to against disease. And the early Greeks would collect these from the Mediterranean and grind the black skeleton, and they thought that the powder of the skeleton had some medicinal properties. There's been no scientific support of that, so I don't know how effective it was. Thank you, pilot. We're good when you are. There are some species of black coral, not those ones, but there are some that are collected for use in the jewelry industry. So in some parts of the world, there are restrictions on collection for black corals. Les Watling says that halosaur translates, or some people say that it's called sea lizard. Yeah, so this looks like it's another primnode octocoral that has met a sea star in its past. You can see most of the branches have been stripped of polyps, but there are a few still alive. Yeah. And because these are asexually growing animals, they can bud off new polyps. So it is possible for this colony to continue to live and maybe spread its tissue back over the colony, but I think it would be a race between settlement of things like the hydroids that we just saw in the sponge and barnacles and various other things that would also like to use this stock. So I don't know of any studies that have been done to see how well these primnoids can recover from this kind of injury, but it is theoretically possible anyway. Thank you, pilot. Yeah, that one doesn't appear to have any tissue left on it, or at least any polyps. There's some white stretches that may be the tissue, but I don't see any of the polyps. Yep. That skeleton is mainly protein. There's a little bit of calcium carbonate in this particular family embedded along with it. It's actually quite a strong skeleton. But there's a variation among the different octocorals. So these yellow fans in the background would be more purely protein and much more flexible. You'll see those move more in the current. Thank you, pilot. Yep, no polyps. So that's been a meal for somebody. And now is basically a perch.
A little perch for a crinoid.
Yep. So Tina Molitsova has weighed in that she believes that may be a Hexapathes, genus Hexapathes, for that last black coral. We're passing over another one of the white stony corals that was suggested, the genus Selenosmilia, or Selenosmilla, excuse me. Another pancake urchin. Once you get back onto the soft sediments, we should start seeing something different. Looks like a crinoid perch there. What's this in the water column? Oh, another one of our pelagic sea cucumbers. The now not rare. That's Pelagiotheria.
So he's going to make a landing there.
I think all we need to do is open the bio box and we'd have a collection of some of those.
We might be able to get one, yeah.
This small fish, let's get a look on this, please. This might be a small rat tail.
I think we've got a rat tail. Yep. Now, I think that's the same species that we saw before. Chris said that he thought it was a Coryphaenoides. To me, the head seems too rounded for Coryphaenoides. Normally, the Coryphaenoides have kind of a flatter lower jaw area. I don't know, Chris, you can weigh in. I think this, to me, doesn't look like a Coryphaenoides at all. It's got a really round head.
Yeah, I agree, but I don't think this was the same one that we saw before. The eye, the large size of the eye and how close it is on the snout and how small the snout is suggests it's a real different one. This could be maybe a Ventrifossa or something. I'm going to try to look in the guide. That head-down posture near the sediment is reminding me of something that I've forgotten the name of. I'm going to try to look it up right now.
Well, Ventrifossa is called Ventrifossa, right, because of that iridescent area on the ventral side, on the belly, and I don't see any iridescence on this guy.
Yeah, okay, I got a possible idea on this this could be Sphagmocurus, because it's got a little bit of dark on the underside of the head and then kind of that steel gray, but not quite right. I don't know. Well, I think we agree it's a macrurid, but I honestly can't really go further than that. But I think it's possibly a Sphagmocurus. But that's a wild guess.
Malcolm Clark is also offering Nezumia. And it's a good thing there's fish people here, because I was just going to say, oh, it's small. It must be Kumba.
Yeah, so we've now it's that's 4 possibilities. Yeah, we really nailed this one, didn't we?
Absolutely. That's why we store the video and the images, so we can go back later with a little bit more information, study it closely. And as everybody who's watching these is seeing, I mean, we just get exquisite imagery that can be analyzed in some detail later on. It does look like there's a couple of parasite hangers-on to this fish that are quite small, so they're not easily or obviously identifiable, but I noticed on the right fin, for example, right pectoral fin, there was something white hanging off of it.
So just to note, Chris mentioned the head down posture, and we actually see that in a lot of macrurids. The bigger abyssal macrurids are almost always seen with their head down and their tails up. And that's like a very typical,
No, we
if
see
they're
these
searching
very
for
often.
a feet plume or searching
Oh,
for
sure.
sediment.
You can see a siphonophore swimming through the water column. And we're going to see, we see so many of these pelagic sea cucumbers, which we thought were rare, but now we understand that they're just, it's a rare morphology. So unlike other sea cucumbers, but we've been seeing them a lot on these dives, every dive. And so we're going to see if we can just swim it into the bile box.
You see that crab?
Yeah, I saw him making a run there.
Okay. That was an interesting idea. He's not flapping. It's just kind of drifting.
Oh. Wow. Almost got him.
Yeah.
Oh.
He's still there. He's on the...
Oh.
Seeing a fish down there straight ahead too, and it's possible that that's a bathygatid, believe it or not. So when we finish with the sea cucumber, and we're still in the area, maybe we could take a look at that.
Okay, Chris, is that the lizard fish that would be on the bottom?
No, it was directly down below the ROV, I think now.
Right, I'm asking, is it benthic form?
Yeah, it's sort of right above the seafloor there. It's not on it.
Too buoyant. Sure. Sure. So for you on shore, we have other cameras that we can see, and for a moment that Pelagithuria was in the bio box, but then it bounced back out. Okay. Sure. Well, nice effort. Thanks.
Heck of a
Thanks,
try,
Bobby.
Bobby.
That was the world's most complex video game. So we saw, if we back up just slightly, there's a fish. And Chris, you'll have to direct me to the kind of fish that it was. Was it swimming above the bottom, or was it settled on the bottom? That's the guidance I need.
It was, it was swimming a bit above the bottom,
Okay.
kind of like some of the McCurds do, very close. But I'm not.
Oh,
seeing
there.
it
Is it
right
on the?
now. He might have taken off or something.
Okay.
You see him?
No, I think that's a rock. I thought that was a fish, but looks like a rock.
Yeah,
Okay.
he was definitely above the bottom and swimming, not kind of stationary on the bottom.
Okay. We'll keep our eyes peeled as we move here.
We might come across another one like that.
So, Chris, what made you think it was a Bathygatus from a distance?
Posture and the way the tail, the way he was swimming. A lot of the Bathygattids tend to be kind of straight, and then right at sort of the last third or last quarter of the body, where the tail, you can see it undulating a bit. It's kind of an unusual swimming posture, and that's what he was doing. So it's I just want to. It'd be nice to confirm. I'm not sure I would log Bathygattid in based on that alone.
Wondering if this is a sea pen that's in the sediments before the rock, in front of the rock. It's very, very straight. So I'm not sure what I'm looking at. Maybe it's an old sponge stock and the sponge is broken off from the top. That's what I'm thinking it is now. There is an Aristeid shrimp swimming down below. You can also see an interesting feeding trace in the sediment behind there. There's a long trail. I wonder what's at the end of that. Yeah, this looks to me like, well, I wonder if it was a sponge or a crinoid. Huh, must be a sponge. Whatever it is no longer at the top.
It's kind of cemented.
Yeah. Copy. Copy that, pilot. Thank you.
Looks like it would be a
Well, that's very interesting.
I think it's a crinoid stock that's missing its whole calyx and everything.
Yeah, it does look like it's got vertebral ossicles. So yeah, I think it is a crinoid, or was a crinoid. So it has lots of hydrozoans growing on it. It's a really thick one.
Yeah, it looks like it's that
It's a big crinoid.
I go for crinoid too.
All right, so it's actually, we know that it's a crinoid and it's dead. There's nothing there, so that's where the body of the arms would have been. All right. So you have to keep in mind as we're moving here that we have 3 different vehicles that are operating together. So we have the surface ship, which is I don't know which. Yeah, I don't know what direction it goes. I would have expected it was a sea urchin or a sea cucumber. No, that's yep, another tusk shell. Interesting. And there's something else, a stalk sticking out just to the right of the trail. Maybe a sponge, maybe a sea pen. It's a sponge, so it's a dead sponge, and so here you can see it was a stalk, and you can see all the frayed spicules. So those are those siliceous spicules that are bound together to form the stalk of these stalk sponges. Thank you. So what I was just pointing out, we have 3 vehicles: the surface ship, we have the Sirius ROV, which is connected by a cable, and then the D2 ROV, which is giving you the main image on camera 1 below that. And so on occasion, we have to wait for the ship to drag Sirius before the D2 ROV can move on. So in those occasions, we spend a little bit more time, we look a little bit more on the bottom, see what we see, and then once the ship is moving and pulling the Sirius, so there's maybe a tripod fish, then it pulls the whole vehicle along. All right, Astrid and Chris.
I agree, Scott.
Yep, looks like a tripod fish.
You want to tell us a little bit about tripod fish? I love these guys.
Go for it, Astrid.
So you can see the way that we call them tripod fish because of the way they're perched on their 2 fins there and on their tail fin there. They've got all these filamentous projections, and they're using that to sense for prey items that might be drifting towards them. So these are ambush predators. They're very sensitive to water motion around them, but they have pretty reduced eye structures. But interesting and ferocious predators here in the deep sea.
Great, thank you, Astrid.
Yeah, so the scientific name of this particular one is Bathipterois. That's the genus. I think it's Bathipterois atricolor, or something very close to that. And the family is Ipnopidae. And they're really, really fascinating. In 2009, the Okeanos Explorer got an image of one of these off of Hawaii. And it was such an absolutely gorgeous image, just like this, that I had it as a screensaver on my laptop computer for years. I just didn't want to change it. It's just really cool fish.
They are very cool, and anyone in the general public listening wants this as their screensaver, you should feel free to go to the Ocean Explorer or Okeanos Explorer website, and all of the images that are made available to the public, you can use them to decorate your own computer or print them out, hang them on your wall.
Whatever you want.
We've got a lot of stunning imagery.
Yeah, we really do.
You see that fish is only about 12 centimeters in length, so not much more than maybe 6 inches. And there was another sea pen, just like the one we'd seen earlier, sticking out of the sediments. Sometimes these sea pens are really hard to see against this light sediment cover. Oh, this is a different kind of fish, I believe. Maybe, is this, what do they call this? The snail something or other?
Yeah, I think this is a live parrot, actually.
Yes.
Little tadpole-like fish.
Yeah, the leparids, Astrid, can you remind me, what's the common name for those?
Yeah, Scott, you got it right. Those are the snail fishes, the live parrot.
Okay. Great. Cool. I think that's the first one we've seen in this expedition.
You don't see those every day.
No.
That was a good one.
Again, a really nice, I love seeing these repeated examples of the rock poking out of the sediment, and that's where corals and sponges are clustered. They need that hard substrate. When the larva settles, they basically glue themselves there. They grow up, and they're growing into the currents, and they have to be on a surface that's stable enough to hold them. So the sediments just don't do it, or the small pebbles in the sediment typically don't do it, whereas things like the sea pens are adapted to live in the sediment. Now, saying that, there are some sponges Shirley had mentioned in our pre-dive call that we might see some of these sponges that, is this a crinoid? Yeah, I think it's just a crinoid. We can get a quick look. Oh, but there is going to be a sea pen that went off screen to the right. Snap on the crinoid? Yep, thank you.
Ah, it's out there in the sediment.
Out there in the sediment. Now, yeah, I don't know if there's anything hard to hang on to, but you can see the cirri are all out, and it's not curled around anything. It's kind of like it's standing on its tippy toes almost. Thank you. And then to the right, we're going to see a different kind of sea pen. This looks like it's in the genus Penatula. And this is the group of sea pens is the Penatulacea. Is that a fish? That's another tiny, tiny tripod fish right there on the left side. Sorry, I'm making you go all over the place. So you can see it really looks like a feather. And now you can actually see the upper part of the peduncle, which is that swollen part that's anchored in the sediments. And so again, you've got one giant polyp, the primary polyp, that is the peduncle plus that main axis. And this has what we call polyp leaves. So each of these sort of arms that are going out left and right are extensions from that main polyp. And then all the other polyps that you see are daughter polyps budding off of this. So this would be what we refer to as polyp leaves. The one we saw earlier, it was a bit unclear if the polyps were arising directly from the main stem or if they were on very small leaves that we couldn't see. This is certainly in the genus Penatula. And we see swimming in front, that's a mysid shrimp, what's called a possum shrimp. Possum shrimp, so named because it carries its embryos in a little chamber underneath the thorax there. And I thought I saw maybe something, yeah, you see there's a worm crawling on one of the polyp leaves on the left side. He's scurrying along there, kind of tucking himself away from the light, I think. Thank you, pilot. And if we go out on the left side, there is a very small, please, yeah, I just want to, I don't know if it's the same because it was so much smaller. There we go.
Yep.
Yeah, it kind of looks the same one we just saw, right, Chris and Astrid? I think so. So maybe a little bit smaller.
Yeah, I think it looks the same, to me at least.
Yeah, I agree. It's that Basitera's after color. I think it's the same one.
Thank you. So we can move on when we're ready. You can see a bunch of depressions here, and there's something in the middle of the one that's in the foreground. I don't know if this is rocks that have tumbled down or if something has excavated a burrow. It could be a feeding trace of a fish. It could be a crab that's dug a burrow. Could be, I suppose, winnowing around some structure.
It could be, yeah. We haven't seen too much of this at this scale.
Looks like a sponge to me. Can we get a close-up on that, pilot? Lower left. That's the right. Yep, that's the right. I wonder, could this be a lithistid sponge? Sponge folks on shore?
Well.
I'm looking at
No.
No? That's kind of burrow there, huh?
Oh, I don't know. Is that sponge or is that old carbonate?
I think that's all some kind of structure is built. Oh, I don't know. Huh.
So whatever it is incorporated a lot of these, I guess, foraminifer shells. So maybe that's a Xenophys or a Okeanos, which would be a single-celled animal. That, yep, there's a small crab tucked in underneath. Huh. I think I am going...
I don't think it's a sponge.
Okay, so I'm going to go with Xenophys or Okeanos. So those are basically amoeba-like animals or foraminiferal-like animals. And I'm calling them animals, I shouldn't. They're protists. They're single-celled, but they have the capability of gluing together objects from their environment. That name Xenophys means strange or foreign particle. So they embed those in the outer cell membrane. So maybe the presence of that is causing the currents to create that swirling action, a little depression.
That could well be an excavate a little moat there.
Indeed.
Right.
Yep. Tina Molodtsova thinks it's too shallow for both a Kamoki or a Xenophys, but Tina, I'm not sure. It sure looked like a protist type structure to me. And Les was noting that we saw an Iridogorgia earlier, and it seemed too shallow for that as well. And yet there they are. This to me Chris Kelly, if you're on, looks to like a Henricia type sea star.
Okay, I wasn't on mute. I thought I was on mute again. This is Zoroaster. It's I think it's in that family, Zoroaster, but I'm not positive. I'm trying to find Zoroasterity, at least used to be. So I think this is given the name because of these really long, sort of sword-like arms it's got. That would be my guess, but unfortunately Chris Ma is not participating today. And but it's very, very distinctive just based on those extremely long arms, and it's got also some spines on the top. I was just looking at a bunch of these with Chris at the Smithsonian about a month ago, and I was visiting him as well. I'm pretty confident of the identification. So this is a great find. Again, these are sea stars that you don't commonly come across that, on these dives. It's really nice to see one.
Chris, do you know if they're feeding on the sediments? Are they scavengers, or are they detritivores, or are these some kind of predator?
Well, they don't raise their arms, and every time you see them, I think they're on sediment, so I suspect they're detritivores.
Okay, great, thank you.
And yeah, their arms are all spread out just like that, kind of at least the ones that I've seen.
Doesn't look like he's in a hurry to go anywhere, so presumably he may be feeding on some of these particulate materials you said is settled on the sediment still. Just went up in the water column trying to find a fish that we saw in the Sirius camera. Oh, there we go. Nice job, Bobby.
Nope.
Oh. Had him for a second.
When you throw sand in his face, he's liable to take off.
Yeah.
All right, Ashton, I'll let you tackle that one.
Thanks, Chris. I don't think any of us got a real good look at that one. We'll leave it at fish.
Yeah, that's as far as I got too, fish.
Well, we've got a good video clipping because I'm seeing the replay, so perhaps on the still image we'll see enough. Looks like a couple of sponges in the sediment there off on the left side. It's a lot flatter here now. We've lost our rocks.
Yeah, we lost our rocks. I don't really see a lot of ripples either. A few you can kind of see in the Sirius view, but not as pronounced, which may tell us the current flows a little less right here for whatever reason. Some more of those sort of little compressions.
Yeah, Tina Molodtsova thought it was more likely that these depressions were burrows due to decapods digging. We did see that squat lobster underneath that structure we were looking at, but that structure itself, I'm quite certain that it's something that was built biologically.
So, Scott, are we still on sort of the flats approaching the cone? I don't have camera 3 feet on right now. Or are we actually seeing a sedimented flank on the cone?
Yeah, it's a bit hard to tell. Let me see if I can get Nav to zoom out on the high pack. Nav's watch lead. Would you mind if we zoomed out just for a couple of minutes or a few seconds on the high pack so we can get a sense of where we are relative to the cone?
Yeah. So yeah, Chris, to answer your question, folks are looking on camera 3. They can see we're on the kind of the lower slope part yet. We're not to the steepest part of the cone quite yet. It looks like so we should see a little bit more rock exposure coming up. Would be my guess.
But it is interesting because we're not on that flat plateau part, at least what I'm seeing in that.
No, we're kind of coming up on it. Where we started, it was a little flatter than it is now.
And we've gone up in depth. Let's see, we're 5 meters above the bottom, so we're at about 1,043, I guess. So we've come up at least 50 meters since we got to the bottom.
Yeah, 50
so
meters, maybe a little more.
Yeah. So there you go, Chris. This I would say this is the lower slope of the cone.
Yeah, it's kind of interesting that you see the exposed rocks further down on the flank, and then as you're starting to very slowly get up on the cone, it's all sediment here.
Definitely backwards from what we think. The pilots right now are just tending to a tether issue, and when that's finished, we'll get back to you. I'm just wondering if that's a burrow or a feeding trace.
Yeah, you really see that thing sticking out there.
Yeah. I don't think that's where the ROV sat down.
No.
I don't think we were up there.
Yeah, there's another.
Yes, Nav, thank you for that zoom out.
Yeah, like I said, like Scott said, the RV pilots are just dealing with a tether issue here, so to twist it around a little bit, and so they want to straighten it out here before we keep moving.
There's a pretty cool sponge. If they could get a look at one of these things that are down below, I think I know what it is, but it'd be really good to get a look at them. Those stock sponges that are just underneath us.
Okay, yeah, time we're untangled here. We'll see. Yeah, so if you're watching on camera 2 here, you can see they're sort of backing D2 up a little bit to kind of take the twist out of the cable that connects it to Sirius. All right, Chris, here's your shot.
It looks like there was a fish just below that.
Yeah, so this is a sponge we don't see very often. They kind of live in sort of shallower to, I guess around 350, 400 meters down to about 800 or 900 meters typically. This is in the Amphipod, and it's in the genus, or it's in the Pheronematidae, certain type of glass sponge that has a special type of spicule, and this genus is called Xenophys. It's a catcher's mitt sponge, is the way we used call them. And even though we see only 2 of them here, often, at least in Hawaii, you'll see these immense fields of these things. They're sediment specialists. They anchor into the sediment with all of those threads, and then they may connect to each other under the sediment, kind of like roots or something like this. But you can find yourself in beds of hundreds and hundreds of these things in certain places in the Hawaiian Islands. So that was great. That was also something that we don't see very often. So thanks for that.
Great. Thanks, Chris.
Pilot, do you think that freshly excavated burrow that we passed was a skid mark from the ROV, or was that something natural? Okay. So if we see another one, let's have a look. And copy. Understood. I know the frustration. Sure. Completely understood. Yeah, so what we're trying to see is if there's something that's excavating this from below that's creating a burrow, in which case we would see a little bit of an opening and something living in a tunnel, or is this a feeding trace of something, some large fish or a mammal that has, a marine mammal that has come down and, taken some chunk out of the bottom. And you can see there's some pile of detritus right around the bottom center. But I'm not seeing an opening. So it doesn't appear to be a burrow in the sense that there's a tunnel below it, or an opening or a burrow below it. That's just a pile of organics that are running down from above, and lots of sediments. I see a couple of pteropod shells. Okay, thank you.
Well, mystery continues.
Yep. The fact that there's only one like this and it looks like it's all been plowed out, it suggests to me that it's more of a one-time feeding event than it is a long-term burrow. So here's another one of those structures that we saw earlier. And Shirley was quite convinced this was not a sponge. I thought that was one of the possibilities, in which case it must surely be one of the foraminiferal-like xenophyophores or something like that. Tina Molotova thought that this was too shallow to see a xenophyophore, but I wonder if there's something related that could also construct this kind of
really
plate-like foliaceous
really
thing.
An interesting shape there.
Yep.
Yeah.
Yeah, so Scott, the only sponge that I know of that kind of has that frilly type of, architecture and is like a leucistic sponge, and I just don't think we'd ever find it in this kind of habitat. But the only way to know for sure is to collect it.
Right. I suppose surely if it was a sponge, it would surely be getting buried in sediment. It would be having a hard time, wouldn't it?
Yeah, I think so. I don't think it's a sponge, and I don't know if it's a xenophyophore. If you tried to collect it, would it all crumble?
Yep, exactly. They're really fragile, and you typically have to get those in box cores, which is a sampling device that would take a big chunk of sediments.
Well, we do have the scoop, though.
And front row, I'd also really like you to introduce yourselves. We haven't had an opportunity to do that, so please do so.
Thank you for joining us at Watch Leads. Your pilot for this shift is Bobby Moore.
Navigator is Fernando Aragon.
Sitting co-pilot Sean Kennison.
And And I see we have Emily Nero in the back on clipping. So we are all engineers and videographers from the Global Foundation for Ocean Exploration, or GFOE, and we're the group in charge of maintaining, operating, and bringing these beautiful images to you.
So, Bobby, we had a few questions from Facebook and Twitter about the ROV. I don't know if you've got a minute.
Sure, go ahead.
Yeah, one I thought was really interesting is D2 neutrally buoyant? Or are thrusters needed to dive and return to the surface?
Actually, D2 is slightly positively buoyant. The vehicle weighs about 9,000 pounds in air. About half of that actually is our foam pack, surprisingly enough. The foam is called syntactic foam. It's made of micro glass beads or balls or spheres, which provide enough density and volume to actually make it buoyant. We make the vehicle. The vehicle also contains about 200 to 300 pounds of lead right now, and we always balance the vehicle so it remains positively buoyant. We do this for two reasons. One, in the event of a catastrophic failure, we want the vehicle to eventually come back up to the surface, which it would do. It might take a long time, but it eventually would come back up to the surface. The other reason we keep it slightly positively buoyant, wow, look at this, slightly positively buoyant, is so that our thruster commands are constantly pushing us down rather than up. If we were constantly having to hold ourselves up, all that thruster wash comes down, and on a dive like today and a bottom like today, all that dust gets kicked up. So we like to try to hold ourselves down instead of hold ourselves up.
Awesome. Thanks, Bobby.
Absolutely.
We're looking at a stock crinoid here, and I can see some reflection of white dots in that crinoid. Those are almost surely eulimid snails, which we have seen very often on a couple of the dives, where all the crinoids seem to be victim to these little specialist predators of crinoids. So I'm looking at some imagery online, and I'm certain that those were xenophyophores that we saw that had that sort of foliaceous plate-like appearance. The question is, what are they doing so shallow?
Yeah, a couple of those seem like dead matches there. Yeah.
Yeah.
That's, huh.
So this is, the common name is sea lily. This is a type of echinoderm, so related to the sea urchins and the sea stars, the brittle stars, and the sea cucumbers. All the little feathery things you see are holding tube feet. We can't quite see the tube feet at this distance, but that's one of the clues that it is related to the sea stars and those other animals that I just told you about. It's a suspension feeder. And a couple of days ago on a dive, 2 or 3 days ago, Chuck Messing from Nova Southeastern, who's an expert in this group, was educating us the difference between when the head is kind of nodding over in the direction it is now versus straight up is an indication of the current flow. And when there's very little current, they're sort of pointing straight up so that they can capture things that are sinking from above. There's a little hatchet fish swimming through the screen going left to right. And we're just having a pilot change, so we can't do any zooms right now. So yeah, when the crinoid has its head straight up, it suggests there's not very much current, and when it's pointed in one direction, it suggests that there is a current flow and it's taking advantage of that horizontal flow. Copy that, pilot. No, I think we can move on and continue upslope. Thank you. We're at a depth of 1,039 meters, climbing this cone on Whaley seamount. We're here just, we're about 65, 70 miles north of the equator.
That's right.
Yeah.
Yep, looks like about 65.
We're still in the boundaries of the Pacific Remote Islands Marine National Monument, the Jarvis Unit. And this is our, what is this, our fourth dive?
Fourth dive in here,
the
yeah.
monument.
Yeah.
There's a rock that almost looks shaped like a sea star. And this rock is shaped like a goosefish. Yes, I think I'm hallucinating
Yeah,
now.
maybe so.
No, Scott, it got me too.
And there's a new fish. I think we have a little Oreo up here, Astrid. Get a zoom in.
Yeah, I think that's our friend the Oreo again. Oreo Samaja.
Feel free to tell us a little about this fish.
I don't know too much about the group the Oreos, but you can see it has a very large eye, so it definitely uses its eye for predation. It's usually, there's actually a seamount fishery for some of these, for species of Oreo, so they are frequently found on seamounts and island slopes, and they are, I believe, vertical migrators. Chris, correct me if I'm wrong on that one.
Pretty knobby face to these guys.
Yeah, at this view you can really see that.
Yeah. I don't want to say only a face a mother can love, but it's...
So the lasers are about 10 centimeters apart.
Ooh, you didn't like what I said there.
This guy's about a little more than a laser's width here. Ooh.
Good stuff. Thank you, Pilot. We're good on video. Is good. I'm having a look through our chat room on this Sunday, and it's really a pleasure to see so many scientists that are joining us. I see Alan Murphy, who is a geologist who helped us a lot on the Cook Island dives. Asako Matsumoto from Japan. And let's see, I'm going down the list of names that haven't been mentioned yet. Santiago Herrera, who's at Lehigh University up in Pennsylvania. Can we get a snap zoom on, let's see, it's just below the rock. It's almost center now, below the lasers, or something kind of pink. Maybe a sea cucumber, but I'd just like to verify that that's what it is. Yes, indeed. Let's get some good imaging. This is different from any of the ones we've seen before. This is, again, another kind of sea cucumber. And it's a deposit feeder, so it's feeding directly in the soft sediments. These are the sorts of animals that you would see in a soft sediment as opposed to on the hard rock, although there are a few that make their way around the hard rock picking at sediments. There's actually maybe another, what was it, the batfish.
Yep, right there.
Tucked over there. We'll look at that in just a second. But I don't recognize this morphology of sea cucumber.
A little knobbier than some of the others.
Right, so those are basically the modified tube feet, and you can see it's kind of flattened. The mouth is at one end, so it's a little bit different from your sea stars and your urchins in that it's been kind of elongated and tipped over on its side. And so it's a deposit feeder, kind of non-selectively ingests sediment, although there have been some studies that have shown that some of them are capable of choosing what sediment they want to ingest, and they will ingest the one that has more organic particles, that is more of the stuff that's settled on the surface than others. Thank you, Pilot. As you pull out on the left, up on the rock, there's a little sediment patch, and there's a black fish sitting in that sediment patch. There it is. You can see the tail on the left side. So I think we were looking at this earlier. So this may be our second batfish, which is interesting because I think Chris Kelly said they're fairly rare to see. Astrid or Chris, can you confirm that this is what we're looking at?
Yeah, I think that would be a good idea. I am not good at these guys. I had misidentified it earlier. But if it is a batfish, then it's in the Ogocephalidae, which is really hard to spell and pronounce. But these guys are quite rare and really neat looking. They're very, you can see, flattened ventrally. So very neat shot. We'll see if we can get anywhere closer than family with these guys later on when we send these images around.
Thank you, Astrid. And then one more thing, Pilot, as you're coming out further to the left, there's a colony. It's kind of whitish. It's kind of bottle brush-like. Yeah, there it is, the tall one. Oh, I'll see. I'm glad I'm getting a look at this. So I think this is Metallogorgia. At first I thought it was another Chrysogorgia, but it is not. And this is interesting because it appears to be in the sediments.
Yeah.
But I would submit that it's not.
I think you're probably right. Looks like that little ridge there probably dives down and got buried.
But presumably, when it was first growing, that would have had to have been exposed for the larva to be able to attach. So what I think is the sediments have accumulated since it began growing. And it's interesting. I wonder if that could give us some clue, you know, to what's the minimum age of this thing.
Yeah.
So this is in the family Chrysogorgiidae. Again, we've seen the Iridogorgia earlier in a Chrysogorgia colony, but this is the genus Metallogorgia. The colony doesn't look in that great a shape. Looks like it's missing polyps from the tips, but it's still alive. And if you've been on any of the previous dives, you've heard us say that there's always 100% of the time in these live colonies, there's going to be this brittle star living associated with it. And this brittle star is Ophiocrius oedipus. So we know what species it is, assuming we're getting the species of coral right, Metallogorgia melanotrichos. They have a very, very tight relationship. Again, it almost looks like there's no tissue on the skeleton, but there is. It's just extraordinarily thin. Thick enough, though, that the digestive cavities of all these different polyps are connected to one another. Thank you, Pilot. That's all we need. This just made me realize, Del, that we've moved out of what was all sediment, and we're starting to see some exposed rock again.
Yeah, we're getting back into it again. And again, this kind of black color we see the rock is just this iron ferromagnesium crust that we've seen quite a bit. Really can make it hard to see what the rocks are.
There's a second Metallogorgia colony on that one rock that's just to the right of the lasers, and so found itself a little island.
Lucky there. Huh?
Yep, little island in the sea of sediment. No, that's okay, thank you. Now I'm getting fooled, you know, by all these rocks that are sticking in the sediment thinking they're fish. Is this, what's wiggling, this white thing wiggling in the, I don't think so. I think it's a hatchet fish. Yeah, I think this is a hatchet fish. Sorry, you're really broken up there, Astrid. This is a really small...
Sorry, disagreeing with you. Yep.
Great.
Hatchetfish. Really nice imagery of this guy. This is great.
Yeah, it's hard to get these. If you saw before, they move really quickly. Astrid was Amanda was describing them last night, that they're usually really hard to see. They're really, really skinny, and they reflect the light so much that it's hard to get a good image. But this is awesome. You can see that its mouth is kind of pointed upward.
So it's interesting, actually, that we're seeing these guys so close to the bottom. I mean, these are vertical migrators again. We normally associate them as being water column fish, so we get a nice mix today. Lots of fishes, a lot of diversity. And you can see those really shiny reflective sides. That's actually a form of camouflage in the midwater. And then again, very compressed to minimize its shadow. Then if we could get a shot from the belly, the ventral side, you can see it also would have photophores down there to basically eliminate the rest of its very, very small shadow by mimicking the same amount of light as would be downwelling from above. So they can actually disguise their shadow by producing light that matches really well that downwelling light. Really great imagery, guys. Thank you.
Fantastic, Astrid. Thank you. I think I noticed on that close-up just below the fin, you saw a row of dots, and that's would be where the photophores are that she was referring to. That would create the illumination. Good stuff. It's weird that it looks like a hatchet, but you can actually see there's some transparent flesh just below it. It's kind of cool. Thank you, Pilot. So there's that other kind of more yellow sea fan. And again, you can see it doesn't have that one large astroskematid in it, which is a kind of brittle star. Instead, it has a bunch of the smaller ones. So that's a consistent difference that we're seeing in these two different kinds of sea fans. And I need to check with folks on shore to see if any of these are of interest for collection. And I was making some notes on who is on shore, so I'll continue to go through that. So I said Santiago Herrera, who is the biology science lead on the first leg coming out of American Samoa earlier this year. And I see Sonia Rowley has joined us from University of Hawaii or the Bishop Museum. I apologize, Sonia. You'll have to type in. I'm not sure what your affiliation is. My bad. She's done a lot of work in the mesophotic, so we're getting up into depths where she's going to be a lot of help identifying some of this stuff. Tim Shank from Woods Hole Oceanographic, and Tina Molitsova from the Shirshov Institute in Russia. So thank you all for joining and contributing with the observations and the identifications and the background information.
Yeah, so we're at a depth of 1033 now, and just to mention, salinity, temperature is 4.6 degrees, and dissolved oxygen is 2.6 milligrams per liter. On the way down, it looks like we hit the minimum zone at about 1.8 at about 500 meters. So for most of this dive today, looks like the oxygen levels on the way down were sort of in this 2.6 range. So.
Oh.
Time flies.
Yeah, this is another Oreo, like the one we were just imaging a little bit earlier. Yep, but it's good to know the abundance down here, so it's always good to get an image. So, Pilot, we are, there's some interest in making a collection if we see one in a good position again, and I'm not sure I do right now, but it would be one of these yellow sea fans, and the trick is that there's actually two down here. There's one that's a bit deeper orange color, and there's one that's a lighter yellow color, and it's the lighter yellow one that we want. Thank you. See crawling on the sediments below here an amatocarcinid shrimp. That's a thread-legged shrimp. It's got these very long legs. And I see an urchin up above on the sediments. Looks like a pancake-type urchin. Looks like maybe if we just get a zoom coming in from the left towards the lasers, a very tiny fish. I think it's another of the midwater fish, and we were just having this comment about it's interesting that we're seeing several of these midwater fish down near the bottom. I think this might be a
Yeah, so
bristlemouth.
this looks like a bristlemouth.
Yeah. Thanks, Astrid.
Maybe a cyclophony. Sorry, did you hear me before?
Yep,
Yeah, and
yep,
so this
great.
is a bristlemouth cyclophony, so very neat. Yeah, we were seeing a lot more of these kind of mid-water deal vertical migrators here.
Thanks, Astrid. Thanks, Pilot. It's good. Here we go. Earlier we were looking at one of these very large stalks of these stalk crinoid, and we saw there was nothing on the top, and at first we weren't sure if it was a sponge or a crinoid, and this is basically the crinoid that it was, and now it has the calyx, which is the main part of the body with all of its branching arms. Nope, we've got several of this, thank you. So again, you're seeing perched on the rocks, this difference between the corals that can need a hard substrate to grow on versus the sediments. And so many of these yellow sea fans, these Gorgonian sea fans, are perched on the sediment. There's either the same or a different Oreo fish. I've seen a few of them now. Another shrimp kind of settled onto the sand there. A little bit more rippling here in the sediments, which is an indication of currents that are sweeping over the bottom. And we mentioned this earlier, but you notice that all of these yellow sea fans are oriented in the same plane. They're all kind of perpendicular to us, and they have their wide face facing the dominant current, so that suggests that there's a current that's passing across our field of view. Our field of view, we're kind of looking towards the southwest now, so it suggests that there would be currents maybe coming from the northwest, moving to the southeast, and maybe back from the southeast to the northwest if it's a tidal current. I'm not sure what direction it's going, but the corals kind of give you that long-term average of the coral flow, of the current flow, as do the ripples in the sand. So the coral fans are actually oriented in the same direction as the ripples. I'm seeing a comment in the chat room. This is from Astrid, who you've heard from a few times here, who's at the University of Hawaii Exploration Command Center, and she says that 1,040 meters, that's our approximate depth, we're actually at 1,024 right now, seems kind of deep for a diel vertical migrating fish. And I find that interesting because we've also heard a couple of comments that, well, we're too shallow for xenophyophores, but we've seen xenophyophores, and we're too shallow for iridogorgia, but we've seen iridogorgia. So a little bit of a confusion about this particular feature and some of the organisms we're seeing. Just kind of nice. It's one of the reasons we're exploring, to get more patterns. We have a big fan coming up here. Let's get a look at this. It's probably the biflabellate primnoid that we've seen before, but let's check it out. Top right corner, there's a big fan. So indeed, this is a fan in the Primnoidae, the family Primnoidae. One thing that's different about this is it's got a big crab. Might be a homalid crab, and if it is, and sure enough, these homalid crabs, they like to hold on to anemones and corals and held them above their head. And this one looks like it's got a little piece of coral that it pulled off of some other colony. It's kind of dangling it above itself there, is presumably some form of protection. If the corals are able to sting predators, maybe this is something that keeps it from grabbing the crab at first. And it's funny, this might be a piece of the coral that we're looking for to collect. So let's just follow the crab. Huh, how about that? I'm not sure how effective that is as a defense. It's a pretty small piece, but sometimes we see them holding anemones. In the last leg, we saw them holding black corals, different pieces of octocoral, so nice view. And sure enough, it's on one of these primnoid octocorals that we imaged earlier. Thank you, pilot. I don't see any other associates on this particular coral, and I believe that was the case for the last couple that we imaged as well. There were no crabs, there were no brittle stars. There you can see the holdfast, which is the way it glues itself to the bottom. Basically, the little bumps you see may be other little polyps that are growing there down on the holdfast, and I'm not sure what the pink is. Huh. I wonder if that was a nudibranch or something. see there's plenty of shrimp, plenty of crinoids, repeated views of urchins. This is a large crab on the coral that just came into the top left. Looks like it might be one of these Yeti crabs with the long, long arms. Oh, no, I see. Maybe it's a coral in front of the crab. Sorry. So quick zoom where the laser should be. Right in the center. Right, okay. So that's a Narella primnoa colony in the foreground with one brittle star in its arms, and then one of the Paramycia-like sea fans, the yellow one in the background, also with a brittle star hanging on there. Thank you. Sonia Rowley is suggesting that maybe this other coral form might be in the genus Bebryce. Sonia, if you're referring to the non-Paramyciid one, we did have a close-up earlier. It wasn't a great close-up, but we are actually actively looking for one now. And the next one we see, we'll get a really good close-up on, and if it's in an appropriate location, try to get a collection. Thus, Watling believes that the crab that we were just looking at, the homolid crab, is a different species from the other crabs, other homolid crabs that we've been seeing, which is really interesting, and it's nice to be able to increase the diversity of species that we get to see on these dives. Stocked crinoid on the right. I think it was 3 dives ago, dive 4, these stocked crinoids were probably the dominant organism that we saw. They were very, very abundant. Can we get a quick look at the coral that's coming on the rock just into view on the right, about halfway down the screen? Thank you. This may be another of the scleractinians, the stony corals. The shape of it suggests that it is. It's kind of like a radar dish. No, that's an Oreo. We're okay with that. And yeah, in a minute, I see another fish we'll get a look at in just a second that's on the bottom. That'll be different and new for the dive. But let's just get a quick look. I'm pretty sure this is that same stony coral that we imaged earlier. And sure enough, and you can see all the polyps are pointing in one direction. And what we saw is the surface is pretty rough. So there was a suggestion this is in the genus Selenosmilla. Thank you. That's enough for our information. So below on the sand, looks like there's a frogfish. It's reddish in color, just to the left of the right skid.
I think we've got another batfish, maybe?
Could be a batfish. Yeah, we're getting in position for a look at it, Astrid.
Hey, Scott, this is the Dry Lab. I have a question for you from Twitter.
Go ahead, Dry Lab.
So Bone Doc wants to know what you want to see today because his 6-year-old is personally loving the fish.
So everything we're seeing are things that I want to see today. There's basically, as a biologist, what I want to see is biodiversity. That means I want to see a lot of different kinds of species. And that could include the fish, like the one we're about to look at now, which Astrid Leitner just suggested is a batfish. And I think, Astrid, this may be different from the one we were imaging earlier, which was a lot darker, or have I already forgotten? But yeah, to continue that answer, we just want to see as many different kinds of animals as we can. That's really the objective of the dive in terms of the biology. And then later on, from a scientific perspective, we want to know, why did we see the animals that we saw in the places that we were looking? Yep, so Astrid believes that's the same fish that we imaged earlier, and we got really good video of it, so we're good to move on. Thanks, Astrid.
Yeah, so I think now we have 2 of these batfishes that are different species. So we got that red morphotype that we just saw another one of, and then that darker one that we imaged before, I am venturing is actually different. It had a couple of different characteristics. So not only are we seeing rare batfishes, but we might even be seeing multiple species of batfishes. This is pretty exciting.
Yeah, I agree, and that's what I couldn't remember if we'd seen the red one before. I knew we'd seen a couple of the black ones. So that may be 4 batfish now today. Yeah, that was the hatchet fish again that went by, and yeah, we have. Thank you, video. That hatchet fish is sticking with us, enjoying its popularity. I don't see the anemone, but I do see in the very upper right, there's kind of a bluish gray color on the top of our, oh, I see, yeah, okay, we can have a look at that anemone. Thank you. There's also homolid crab on this. So here we have 2 primnoids next to one another. We don't know the identification of this particular species, and we collected one on the last leg. And I see both of these each have a crab, and I believe they're both homolid crabs. And we just had a close-up before of the crab. I know you saw that it was holding a bit of coral. So here again, it's holding a bit of coral. Here all the polyps are out, so you can see the coral is alive. And is able to survive, and is probably still able to feed. And, you know, our suspicion is that this is a defensive maneuver, and I'm kind of talking a bit haltingly because I'm studying the coral itself, and I'm wondering if that's an Acanthogorgia coral, and if it is, we haven't actually seen the colonies. So I got to figure out where this is grabbing these bits of coral. As we've been going over, I have been noting some slightly lighter yellow colored colonies, and I've been ignoring them thinking they're Paramaricia, but now I know I need to check them, so I gotta owe that crab a thanks. So here we're looking at this very large anemone that is settled on the top of this coral. Looks kind of precarious, but very interesting. Normally it would have kind of a flat lower part to the polyp, but here it's kind of wrapped around. It almost looks like fingers. This is something that we would refer to probably as a Venus flytrap anemone. Oh, here we go. Looks like one of the golden crabs. This looks like one that has not molted in a very long period of time. Oh, thank you, crab. You're showing me where that other plexaurid is. That's the coral we're trying to find. This is a female. You can see her abdomen is curled up underneath her, but it's kind of swollen and pushed down. I believe she's got a bunch of eggs in there, is my suspicion. A lot of dark staining on the carapace, and you can also see these white structures on the carapace. These are barnacles, gooseneck barnacles or pedunculate barnacles. You can see a whole bunch. No, thank you. So that tells me that this crab has not molted in a very long period of time. It's also missing some legs. It should have 5 pair of legs, and you can see there's only 3 pair on the right side of the crab. So it's missing its last 2 legs. And there are 4 on its left side, so it's also missing a leg over there. And there's something else. Is that a solitary hydroid? There's something hitchhiking on its left chela. Can't tell exactly what that is. Huh. Maybe I'm just seeing the soft membrane between the joints. Thank you. That's good on the crab. We have seen that crab on other dives. Now we can put an identification to that. And I'm okay to move on. And so off on the top right, there was something bluish on the top of a rock. And as I was noting, as the crab was going by, it bumped into a very small colony there, and that may have been a small version of one of these bits that the crab was hanging on to. So I wonder, this may be an encrusting sponge. Yeah, so this is a sponge, and I think, unfortunately, our demo sponge expert, Shirley Pomponi at Harbor Branch, had to step away. And so we'll just get an image of this.
I'm back.
Oh, you're back. So you're seeing the encrusting sponge?
I heard sponge, and I quickly looked in. I think it is a demo sponge, most likely in the order Peaceful Sclerata. Beautiful, thank you.
Thank you for that, pilot. Just passed over a Chrysogorgia colony as well. They are the ones that are sort of pinkish hue and look kind of like a bottle brush. So the colony that the laser just passed over, it's now at the bottom center of the screen. Let's check out to see if this is that other yellow one that we're trying to get a look at. Yep, right at the lasers. So maybe, I'm not seeing from this angle any brittle star. Yeah, I think it is. This might be the Acanthogorgia. This might be what that crab was hanging on to. Maybe that might explain why we weren't seeing those big astroscumatids earlier. Yeah, they're really crowded close together. And one of the things that separates, for example, the Acanthogorgia from the Paramercia is the relative thickness of the tissue that's covering the skeleton. And so in Acanthogorgia, it's relatively thin. It looks like those polyps really stand out from the skeleton, whereas it's relatively thick in the plexaurids. What's all that brown stuff that's, yeah, bottom right, it's in the sand. It looks like it's dripping and, huh. No idea.
Huh.
Is that just stained rock, Del?
I don't know.
Yeah.
It's...
Yeah, I've been looking in the chat room, and I'm not seeing much in the way of response, so I would say no. Thank you. And we can move on.
So we didn't take a sample?
We have not taken a sample. That's the first one that we really saw that this is definitely different from these other Paramerciids. We think that's an Acanthogorgia, but I don't think it's what we were looking at earlier, that brighter yellow one. So I will leave it in your hands, and if you see a lot of screaming in the chat room, that'll be a sign that maybe you should collect it.
Okay, scream it way, people. Yeah, let's just do a quick zoom here. not entirely sure we've seen this this morning. Someone on shore want to ID it?
I just walked out and got as far as the dry lab and saw this. So we've been calling this Victorgorgia. This purple colony is probably a scleraxonian, which means it doesn't have an internal skeleton. It has cemented sclerites. And I'm looking at the base. I think there's two, maybe there's yeah, absolutely. Those are aplacophorine mollusks, and this is a really different view. The one that's on the right is actually curled away, and you can see there's a groove down the center of it. So these are worm-like mollusks, and then you can see what looks like a little fold at one end, and that is probably where the mouth is tucked in. So fantastic view. Two aplacophorine mollusks, specialized predators, and this would be a Victorgorgia.
All right. Thanks, Scott. So, pilot, we'd be inclined to get a rock if we happen to see one that looks collectible to you. I don't see much right here, but keep your eye open. Angular would be better, and bigger the better. Okay.
So, Del, you keep talking about getting a good rock sample, and it's the bigger the better, the angular the better. Why does that matter?
Oh, yeah, good question. So most of what we're seeing on these rock outcrops, this black coating, is really what we call an iron manganese rich crust, which sort of precipitates on the rock through time. We'll let people enough. Astrid, you still there? You went to lunch. You want to say anything about this fish?
Looks like it's...
Yeah, this is another Oreo. So we got another Oreo stomatidae, probably another Neositis.
Okay. And so what we'd like to do with these rocks is, well, here there's a little bit of a mystery still. This coating kind of keeps us from actually identifying it. It can be really tricky even to tell old carbonate from basalt. And so one thing we like to do at the most basic level is get a rock sample and just see if we can tell what it is when we break it open. And if it is an igneous rock, then we'll be very interested to try to look at its geochemistry, see how it relates to nearby seamounts, and also try to get an age date. And we can do that with a technique called argon dating, where we actually look at the decay of radiogenic isotopes through time. And so there aren't a whole lot of age dates in this part of the world still in some of these seamounts. And we're really trying to unravel if they all form at the same time, but there are different generations of seamount formation, and then what that really tells us about plumes and hot spots and plate tectonic processes, and kind of very big picture story of how the Pacific Basin formed.
Is that the only way you can tell the age of a seamount?
That is largely the only way we can tell the age of the seamount is to, the way we can do it most accurately would be get a geochemical date. We can do other things. We can look at how that seamount has bent the plate that it loads on. That tells us how old the seafloor was at the time it was erupted. So it gives us a different kind of constraint. But primarily, yeah, we need to actually get a rock and determine its age geochemically. Yeah, even determining the age of the seafloor gets a little tricky here. We're in pretty old seafloor, and this is a period during the Cretaceous period when there aren't magnetic anomalies. The Earth's magnetic field was stable for a long time, and so the way we usually determine how old the seafloor is by looking at these magnetic stripes, and that's if the poles don't reverse very often, it becomes very difficult to do. So. Sorting out the tectonics of the early Pacific here is really a challenging task.
So why would you go, though, for an angular rock? What does that tell you?
Well, hopefully the angular rock is something that's broken away more recently. You can think of this rock that's been free for a while having more surface area. So you can imagine seawater is interacting on all sides with the rock and potentially altering it. What we hope is that when we break this thing open, there's enough unaltered basaltic rock inside, if it is a volcanic rock, that we can actually do those geochemical analyses. When they get altered, that changes the mineralogy, that changes the chemistry, and it's not suitable for some of the things we'd like to do.
And what is smooth? Oh, you're getting called.
Yep. Go ahead, Pat. Okay. Yeah, I think we can just head on up when you're ready, and we're trying to see if we can find a rock bigger the better, more angular rocks sort of broken off more recently, and so that's potentially something that it won't be as altered. But you can see what we've been seeing is a lot of this sort of consistent look to the slope as we've gone up. I think we're past that anemone there, so we can keep moving. I think we.
So would you say it's fair to say that the rocks on the seafloor are similar to like rocks in a stream, that the longer they're there, the more weathered they get?
Yeah, that's exactly right. The longer they sit there, the more they interact with the seawater, and a lot of that has to do, in some places, of course, the water's hot and gets hydrothermally altered. In other places, this lava that's erupted on top of the seamount here is doing a lower temperature form alteration, but the longer it's exposed, the more that water can diffuse into the rock. Even if it's fairly solid rock, that water will, through time, diffuse in, and we'll see this sort of alteration migrating in towards the center.
So there's another squat lobster on a coral. This one looked like it might have had a sponge on top of it instead of a coral. Folks on shore, do we have any ideas on these corals that we keep passing?
All right, someone will have to help me out with this.
This looks like one of those siphonophores we saw the other day. But that's really an Amanda question.
Yeah, I think it's a siphonophore. But yeah, is Amanda around?
Amanda went to lunch.
We do have a fish there in the background, though, too. Yeah, it's going to the fish. I think we don't have our siphonophore experts with us. So
this is a different kind of eel. We haven't seen this guy yet today. I'm going to go through my eel guides before I throw it out there, but thanks for this imagery. It's really good. I'll get back to you.
All right, yeah. See if we can get a good shot of the head here, I guess, if possible. Yeah, not going to cooperate.
There's a bunch of those shrimp, though. There's at least 4 on that rock.
Yeah, let's take a quick zoom there.
So I'm thinking that eel was a snake eel, an Opixidae, but I will take some good notes and send them on to Bruce.
Okay. Let's let him deal with this. So we're just going to come up a little bit. We got another little loop there in the tether that the pilots are going to try to deal with. So we just take a second here.
So if you're just joining us today, this is the NOAA ship Okeanos Explorer. We are in the midst of conducting a transect on the seafloor of a seamount that's been dubbed Whaley Seamount in the Jarvis unit of the Pacific Remote Islands Marine National Monument. So far we've had quite an exciting dive, some interesting geology and biology, and in just a few minutes we'll be acquiring bottom again and see what lurks up just ahead.
What exciting things. Yeah, we're diving on sort of a flat-topped Whaley seamount is sort of a flat-top seamount. It's got a few little cones built on top of it, and we're sort of climbing up on one of those cones. We started our dive at about 1,100 meters this morning, and we're up to about 995 meters at the moment.
So Del, while they're working on the tether, we have a question from Facebook about hydrophones.
Oh, great.
So a gentleman named Donald has asked if we have hydrophones on the ship and if the seabed is noisy.
That is a great question. It's one of the things I work on a lot in different environments. As far as I know, we don't have a passive hydrophone to listen down there. That would be a great thing to do someday. Maybe we can work on that. In shallower waters, we might expect shallow water fish to have swim bladders, all right, and producing a lot of kind of percussive noises. We see that a lot. These depths, I'm not sure that we would expect to see that. And so it's really not entirely clear what it sounds like down here. There are very, very few observations. Presumably other animals besides fish, though, could make noise. Crustaceans and urchins, these things are probably scraping against each other. That makes some noise. We may also have a little bit of noise from sort of current interactions with the bottom. Yeah, that's sort of an unknown, and this is kind of an important field of study. We can hear all kinds of things with the hydrophone, but one of the things that scientists are starting to look at is sort of this emerging field of soundscape ecology, where we're trying to understand how organisms interact with their acoustic environment. And, this is kind of a natural thing to study because we're down at these depths. We're, as humans, we're used to seeing things, relying on our sort of optic abilities. Down in this environment, there's not a lot of light. Vision may be less important. So a lot of animals in the deep sea have probably, you would think, would have evolved to actually use sound as a sensory mechanism. And so that's a great question, and really unknown in sort of these deep habitats.
All right, so I have my own question about those hydrophones.
Oh,
so
sure.
when you're in the deep sea and you have a hydrophone, how can you tell what each of those sounds are? What do you use as your control to figure out, to piece it all together like a puzzle?
This is very tricky. So we, like I said, there isn't a lot of this deep sea work done. We've been doing a little bit of work actually, and with some colleagues in some cold seeps in the Gulf of Mexico and the Atlantic. And we did some kind of pilot data where we put just one hydrophone out, and we didn't have any video. And so there are tons of things that we hear, and actually we have no idea what they are. And so one of the things we have to try to do is also do observations like we're doing today. We see what species are there, what might be making noise, and then try to merge these two things together. It can be really challenging if we just have a sound recording. But there are other things. The marine mammal calls are very well studied. In shallow water, we have a kind of growing catalog of different fish calls and snapping shrimp and things like that. But it can be a bit of a challenge. And when we work with these data sets, we see things or we hear things all the time that we can't yet identify.
Do you hear things besides animals?
We do. I actually started in underwater acoustics because I wanted to study earthquakes. And so if we're in the mid to low latitudes, like we are here, as we go down in the water column, the speed of sound first decreases, and then when we get down below 1,000 meters or 1,100 meters, it'll start to increase again. And this has to do with the temperature and pressure effect on the speed of sound. And if you remember back from your physics class how you probably learned about refraction in terms of light, well, it turns out sound waves can also refract. And so this low sound speed, kind of, we think of it as a channel, actually guides acoustic waves. And so little small earthquakes beneath the seafloor can generate sounds, which gets trapped in there, and they can actually propagate hundreds or thousands of kilometers. And we're able to use those sounds to locate earthquakes out in the middle of the ocean that we couldn't see if we were just listening on land with seismometers, because those sounds propagating through the solid earth attenuate much more quickly. So of course, yeah, we hear earthquakes, we can hear landslides. We work in Antarctica, we can hear ice sheets breaking up. Of course, lots of marine mammals, ship noise, any number of things. That's one of the great things about these passive acoustic data sets, is we can really collect one type of data and then use it to study many different processes. So it's a really efficient way of doing science. And we can complement it with some other observations. We can really push the number of fields forward like that.
Do the different physical properties of different bodies of water change the way you hear things or how long it takes for you to hear things?
Yeah, so the overall travel time doesn't change very much, but it's really the structure as you go down in the ocean. So as I said here, there's sort of this wave guide what we call the sound fixing and ranging channel, or SOFAR channel, down about 1,000 meters depth. If we were to go to either pole, the water column overall would be getting colder, and what we'd see there is that sound no longer have that sound fixing and ranging channel. We have sort of we have a sound speed profile that just increases with depth, and so sound propagates differently there. It sort of reflects off the sea surface and then turns and refracts and bounces again, if you can imagine. Maybe it's a little hard to describe. Pretty much easier with a little diagram. We kind of call this a half channel, and so the kind of mechanism of how it's propagating laterally changes, but the speed is fairly similar. But certainly the more efficient propagation is when the sound is sort of refracting out here in the middle of the water column. If you don't bounce off the sea surface and you don't bounce off the seafloor, you don't lose a lot of energy, and that's one of the real benefits of this technology. We can hear things over a long distance. And this is...
That's really interesting. Thank you. So one of the things that you notice about the rocks here is they all look like they're the same color. And we talked a little bit more about what that color would be. But how do we get these ferromanganese deposits?
Yeah, so if these are deposits that we see really all over the world. And we need to get kind of free metals in the water, so often these are forming down where oxygen is fairly low, and then we'll get this mineral precipitates all over the rock. So they tend to be most prevalent between maybe 400 meters and 2,500 meters, but we can find them at shallower and deeper depths as well. And one of the things that we're doing here in Capstone is trying to, as we sample the rocks, we talked about analyzing the geochemistry earlier of the rocks to try to look at how the seamounts form, what their ages are. Well, we'll also have samples of these crusts, and we'll be looking at the composition of these crusts in different areas, trying to understand what the concentrations are of different elements. Not only do they have iron and manganese, they also have a lot of rare earth elements, things like cobalt and nickel, many things that are in short supply now in today's world, and these are things that are used in a lot of our everyday technologies. They're used for metal alloys, things that go into solar cells. If we talk about mining other kinds of deposits off the seafloor, some have large amounts of copper. And so there's a lot of interest in, understandably, there's a lot of interest in resources in the sea. And so that's one of the things that'll be able to be assessed from this work. Of course, this ship is not promoting deep sea mining. We're trying to take baseline information here. And it is really by doing this sort of scientific diligence that we can really decide if people do move forward, how can we do this in the way that has the least impact on the environment. So that's one of our many goals here on this series of expeditions that we have in this part of the Pacific. Yeah, so if you're just tuning in here, this is the Okeanos Explorer. We're diving today on Whaley seamount. You're still seeing on camera one a view of the seafloor here. We've pulled off bottom just a little bit while our pilots try to deal with a little kink or a little twist in the ROV cable. This is the cable that connects Sirius, the upper ROV, to D2. ROV you'll see in the camera view there on camera 2. And of course, this is sort of a
hold that.
So yeah, you can see the twist there in the camera, and of course they don't want to keep bending it or twisting it in that way. We could break the
video.
many wires in that cable, and so this is what we're trying to deal with, and hopefully we'll be back underway here in just a bit.
Dive soup this
So Del,
video.
this is Amanda Nuppern joining. Gonna
We've
sit here for a
rewound the
little bit.
camera.
One of the interesting things I've
We
been
rewound
noticing on
the,
today's
Sirius
dive,
camera to the
possibly
time when that spun
more than
happened,
some of
if
the
you
other
guys wanna
dives
see
we've
it
had, is
there
how
on the
many
monitor.
midwater animals seem to be intersecting with this feature. We've seen a lot of hatchetfish and
Include your small ones, we've got two
siphonophores, the
nibbles at the
surgested
top.
shrimp, all of these animals that are really canonical midwater animals. Something about this depth, there seems to be a lot
Yeah, we saw
floating by.
a few of those at Jarvis when we were a little shallower.
Yeah.
Right, but we're quite a bit deeper now.
Yeah.
We are on this kind of interesting little knob that's sort of sticking up in the water column. So I wonder how much is them coming down, or are they sort of getting evected over to the side? It's a little hard to tell.
Yeah, and I'm peeking at the EK60 data. It's the first time today I've really looked at it, and I mean, it does look like that broad, deep scattering layer comes pretty much down to the seafloor here. It seems to be a little bit deeper than it's been the last few days.
Oh.
So I wonder, sometimes around these seamount features, there can be some local upwelling or other aggregating effects because of the interaction with the pelagic environment on the seafloor. So I wonder if there's something going on here that's
that's
makes
certainly
for some good
interesting.
habitat.
Am I reading this right? It looks like as we came up on the seamount, that layer actually dipped down deeper. Is that right?
Yeah, that's definitely what it looks like there.
Yeah. So our EK60 is a high-frequency sonar, and basically it's looking for backscatter from things in the water column, which might be fish or gelatinous animals or all sorts of interesting things that are poorly understood, as I've come to understand in this cruise. Some fascinating shapes.
So Amanda.
Yes.
Shirley. Amanda, this is Shirley.
Hi, Shirley.
Because I woke up this morning about the sea scattering layer and the avoidance last night with those metophids. And so I was thinking about this a lot and wondering, well, first of all, and for anybody who was watching late last night, because that deep scattering layer kind of disappeared once the ROV was in it and the fish were all, I guess, swimming away.
Yeah.
So here are questions. One is, where did they all go? And is it possible, so they're not scared away by the tether, because obviously the tether is going through that scattering layer. Is it the mechanical thing, or is it the light that's scaring them away?
Yeah, it's a great question, it seems to be both the mechanics and the light, and I haven't had the opportunity to do this myself, but people have done things like turn off the lights and transit through, and we still see that avoidance. We obviously can't really turn off all the mechanical noise safely to test that side of it, but I think both can have those effects. But the other interesting thing is that sometimes light attracts things in the ocean. So I've actually had it where we've had, a ton of midwater fish following the ROV around for the course of the dive. So it seems like some animals avoid the lights and some like the lights. And it can even be somewhat inconsistent with the same species in the same locations. I've talked a lot with my Mbari colleagues who've done, a lot of work in the same part of Monterey Bay. And they say the same thing, that they'll see the same animals. Sometimes we'll completely avoid the ROV, and sometimes we'll follow them around. So there's a lot of mystery still as to why we see these behaviors.
So I know that, Amanda, you and Astrid were kind of having sidebar conversations about this last night, and I was wondering if you put red lights on the ROV, would that make a difference?
Yeah, so it does seem to help for certain animals to do the red light, and certainly for observing more natural behaviors, the red light does not interfere so much with the animals. I'm not sure how much with the fish that helps, which seem to avoid the ROV regardless of the light situation.
Yeah,
but for other organisms, I think that can help a lot with causing less interference.
Yeah, and what was kind of curious last night also, because I know I've been at sea many, for decades, and sometimes at night, when you're on the surface, on the ship, and you can see all these squid coming up to the surface, but we haven't seen that yet, and they're attracted to the light. We haven't seen that.
No, we haven't seen that, and I don't know if it's just because there just aren't as many here to attract, or if it's something different about the vehicle. My guess would be it's the former. We're still in fairly oligotrophic water. Not so much as we've been close to the equator, but I think we just don't have these huge, pools of squid and other animals to attract to us right now. Although we sure seem to be good at attracting Holothurians.
That in itself is an interesting observation, actually.
We almost caught one.
Yeah, so I mean, a lot of the work, and I'm still relatively new to using ROVs for midwater work, but a lot of the work that I've done has been off the coast of California, which is really highly productive water, and you regularly will see those, big schools of squid bombarding the ROV at the start and end of the dive. We'll see. I mean, we feel like we've been seeing a lot of midwater animals, and we've seen a great diversity of animals, but sometimes you'll get into layers of siphonophores that are, take up the whole screen with numerous siphonophores, and I think it's unlikely that we would see that in this part of the ocean. So.
It's been really interesting that you're doing the midwater dive because, we have so little data from this area, of the planet in terms of like what's living in the midwater. So these observations are all really brand new, so it's pretty cool.
Absolutely. It's very, very exciting for me to be able to come out here and observe this. It's, we're definitely some of the first eyes to ever see what's down here, which is pretty awesome.
So Amanda, how is most midwater work done then, if not with an ROV?
So traditionally, and still probably most of it, is done with trawl nets. So you take large nets and drag them behind a ship, basically. It's very, very simple. However, over the decades and more than century that we've been doing that kind of work, technology has improved what we can do with those nets. So there are nets that have open and closing systems so that you can either target a single depth, so that you'll send the net down closed. You can send a messenger down the net, open it up when it gets to the depth you want, tow for a while, and then close it up. And that way you know exactly what depth those animals came from. And there are also versions of those kinds of nets that have multiple open and closing nets so that you can get 4 or 5 different depths that are, discrete depth sampling. Oh. Some nets now are leveraged with different sensors, so you can put a lot of the same sort of CTD sensors and oxygen sensors that we have on the ROV or on a CTD rosette onto the net itself, so you can see what the environment was where you collected those animals. And more and more we're seeing people start to put acoustics and imaging systems onto the nets. I've even come across some work where they don't even collect the animals at the end of the net. They have a net to actually, drag the animals through, but then there's just a camera looking down at the animals that are coming through, so you get really nice images of what's there, but you never.
Them and,
and it's kind of a nice low interference way of seeing what's down there. So although nets are relatively simple, the technology has really developed even in that realm. Of course, like we're doing here, acoustics are another way to survey for what's there, and you get incredible spatial and temporal resolution. And a nice thing about the acoustics is that you can run it while you're doing other operations. So during all of our mapping transits and during the dives, we're running the acoustics and getting a really nice kind of map of where animals are in the water column. Of course, the problem with acoustic interpretation is you get backscatter. So you know where things are, but interpreting what they are is a whole other challenge unto itself. So we've been lucky that we have the ROV and are able to get down into those layers and see at least some of what's there.
Right. Well, you've been using this data from the sonars to actually help plan some of those depths, correct?
Absolutely, absolutely. So when we did our midwater dives yesterday, we, were looking at this acoustic backscatter throughout the whole dive and targeting different layers. The first midwater dive we did, we saw that deep scattering layer made a migration. So a lot of the animals that live there migrate up to the surface at night to eat. And because we were doing an extended dive that started while we were still down at the ROV, so all the depths that we had targeted at the start of the dive when everything was down deep, all those things moved. So fortunately, we have some ability to be adaptive, and we were able to intersect those layers a little bit shallower than we had originally planned. And that was a fun time to observe those animals because they were really active active and hungry on their way up to the surface. Sometimes you can see really large animals. You'll see, there's so much backscatter right now, I don't think we'd be able to make them out.
Right.
But when there isn't so much throughout the water column, you can sometimes see individual targets, like a large tuna,
Oh,
or
really?
I think we might have been seeing dolphin the other day when it must have been at Jarvis.
Right.
We were seeing some larger targets under the ship, and I think it's very likely they were the same dolphin we saw outside up at the surface, coming down and feeding on this layer under the ship. Dolphin are certainly one of the organisms that when you look in the guts, we see myctophids and other kinds of midwater animals.
Yeah, I didn't know that. Huh.
So part of the reason I started studying the midwater fish, we really don't have much in the way of commercial fisheries for these fish. There are some in parts of the world, but not in the U.S. But they're far more abundant than anchovy, sardine, squid, all the things that we kind of think of as the mid-trophic level prey. So, all those smaller fish are feeding on plankton and then are fed on by tuna, swordfish, sharks, marine mammals. And so there's always been a lot of focus on those epipelagic animals as forage fish. But it turns out that a lot of those animals also have a significant component of their diet that comes from the midwater. Some of those animals are actually diving down to feed on the deep scattering layer, but a lot of them just wait till nighttime when
wait it out.
the scattering layer comes to
arrives.
them. But that was my initial interest in this community was really because of that tie-in with a lot of the things of commercial and conservation value. Can't tell what we're seeing on that screen.
Yeah, so if you're just tuning in here at the Okeanos Explorer, we're diving today on Whaley seamount in the western Central Pacific. And we're just dealing with a little issue here with the tether that the pilots are trying to sort out. So we're come off bottom by about 30 meters now. Fortunately, we didn't come up enough to get that cool midwater layer yet. We're below that. We're seeing a few things here on the camera one screen.
Yeah, I'm definitely seeing some critters floating by. It's hard to get an idea on much of it without zooming in and taking a closer look. A lot of the midwater creatures that live at these depths are gelatinous. They have a really high water component. and that often comes along with being transparent.
Yes.
So from a distance floating by, it can be hard to differentiate between the different taxa that live at these depths.
Yeah, so if you're watching on camera too, then you're seeing the cable which connects the two ROVs and this little twist that they're trying to get rid of. There's also this kind of looks like a rope that goes there. That's actually a lead that they wrap around that cable, and they use that when they retrieve D2.
If you like, I can
And it
describe
looks like-
that a little bit for you, Science.
Yeah, that'd be
That'd be
great.
great.
Great, Roland. Thanks.
So there's actually two ropes. One of them is the lift line, and then there's another one that we call a daisy chain. It's a much smaller line. We use that to keep the rope next to the umbilical so that it doesn't actually get twisted around it. And what has happened at some point today, the umbilical, the yellow part, has gotten a twist in it, an unexpected twist. So the reason we're working so patiently to get this twist out, because if the ROV were to get too far ahead of Sirius and put too much tension on that, especially as the Sirius is heaving up and down, that could pull that very tight, and that could actually break the fiber optic wires and in there, and then we would lose all control of the vehicle. So that's why we're being so methodical about trying to get the twist undone, and what you're not seeing. In the control room, we have several of the pilots discussing it and determining which direction which vehicle needs to turn in order to get that out. You can also see that lift line and that daisy chain line is also a little bit tighter, so if we get the twist, don't get the twist just right, that line needs to slide back and forth in order to get that kink out. Even if we get the kink, the umbilical twist correct, if that lift line and daisy chain line doesn't slide, then that could keep that bend in the umbilical, and that puts the whole dive operation at risk.
Cool. Thank you so much, Roland.
Hey, science team, this is Web Coordinator. I have a question for you.
Go ahead.
I had a question on the internet about the precious corals. Can you talk a little bit about the precious corals that we've seen so far and how those fit into our mission?
Del and I are both looking over at Scott.
And I was looking back at Amanda and Del. So the precious corals is a collective name for a group of corals that come from mostly the family Coralliidae, and I think what people mostly think about are the pink precious corals, and there's been a thousands of year fishery in the Mediterranean for Corallium rubrum, and the skeletons are really cemented together quite well, and they're quite hard, and so they can be polished, and so they have a beautiful surface when they're polished, and they can you can take advantage of the shape of the branching and so on to form some nice coral jewelry. So there have been some fisheries in the past for some of the deeper water corallium. Certainly in the Pacific in the 70s there were fisheries, and for a variety of different species. Sometimes people also include some of the black coral in there because those skeletons can also be polished, and even sometimes some bamboo coral. So one of the things that we're interested in as the capstone mission is identifying where we may find beds of the precious coral, because they're actually managed fishery. These are corals that take a very long time to grow, and so we want to make sure that they're used responsibly so that the entire population isn't wiped out inadvertently. Obviously, anybody who's harvesting them for a business also doesn't want to wipe them out, because you would want to use them long term. But in the past, some 40 years ago or so, one of the ways that they went after the deep sea ones was basically drag chains. And anything that would get caught up in the chains would come to the surface. But as you could imagine, it's pretty destructive, and it leaves a lot of broken colonies on the bottom. So, of course, with the ROV, we're now able to image them in situ, get a better sense of where they're growing and how they're growing. And so better understand the populations and make some good decisions on targeting if there is an interest in using them. Now, one of the things you may hear us refer to is that here is a coralliid. That's a short name for the family Coralliidae, which would be a member of the precious corals, but isn't necessarily one that's of interest for using, for example, in jewelry. So there's only a limited number of species that are used. It has to do with their color, the thickness that the skeleton can grow, and probably their ease of capture.
So are there shallower water cousins to some of these then that have been used for that as well? Or?
Yeah, absolutely. So the ones that are in the Mediterranean come up to about 30 or 40 meters depth. So I believe you could even scuba dive for those ones. And the question, if I recall some of the papers, the conservation type papers, no surprise, since they've been harvesting them for almost 2,000 years, the biggest beds of the shallower waters are gone. So you have to go a little bit deeper now to get them. And I'm not sure of the species that are used around Hawaii and some of the seamounts and so on, how shallow or the populations are going for there, but I believe we're talking about in the 100 to 200 meter level.
Okay.
Thank you, science team. I had another question from the web that maybe you guys could answer.
Sure.
I had a question about the pressure down there. How much pressure is there?
Well, yeah, pressure increases linearly with depth, and so it really depends on what depth we're at. So
it's what, 1 atmosphere per 10 meters, I believe.
I believe yeah. I'm trying to put it in common units,
oh, good.
and
good for you.
not Pascal. So yeah, something like that. And so yeah, it really depends on the depth that we're working at. You can think of it as kind of a linear increase, but I think that
Sure.
yeah, the number
yeah,
you
1
gave
atmosphere.
is fair.
So what you're feeling at the at sea level, basically that doubles for every 10 meters of water depth. And one of the ways that I like to have people think about it is if you've ever gone into a swimming pool in the deep end of your public swimming pool, and that's probably going to be approximately 15 to 20 feet deep. That's a little bit less than 10 meters, and you can definitely feel that pressure pushing in on your sinuses.
Mm-hmm.
And so now imagine every 10 meters you go, that's doubling. And so the depths that we're working, so today about 1,000 meters, that means 100 times that pressure. And then go down to 2,000 meters and 3,000 meters, and you can see that it's really extensive. And another analogy that I've seen for, I think it's used for the 2,000 or 2,500 meter depth range, is that imagine an elephant sitting on every square inch of your body. So that number of elephants would be the weight of the water on your body. That's a lot of elephants.
Great. Yeah, those are good things to kind of put over perspective. As a geophysicist, I was going to start doing density times gravity times depth, and then that'll get us into pascals, and then I didn't know where to go from there. So, but it's a simple calculation, but yeah.
Yep. One of the few simple calculations. I know when I took oceanography classes, I remember one of the things that was always so confusing and hard to figure out was adiabatic heating and pressure and so on. And as the pressure of water changes, or the pressure in the water column is changing, it's affecting temperature and your ability to measure temperature, and so you have to make corrections for whatever tools you're using to measure temperature based on the pressure, and what happens if you bring that parcel of water up, and what's the temperature at sea level and all this. That was fun.
So we had some questions the other day about how much the pressure change affects these animals as we bring them up to the surface. So as you were saying, Scott, when we dive down, we can feel that pressure change in our ears, or if we go up in an airplane, but we don't really feel it in any other part of our body. And that's because there's air inside our ears, and that's a compressible. So anything that's filled with gas or air will change a lot in its volume with a change in pressure. But if it's filled with water or tissue, it really doesn't have much effect, that change in pressure. So for most of these animals, it's not the pressure change that's too much trouble for them as they come up. The bigger issue for most of them is the enormous change in temperature from down there to up here. So our bio boxes are designed to thermo-insulate and maintain the temperature and keep the same water in that those animals were collected in. And typically when that's all working really well, when we reach in to collect those animals, even sometimes several hours after we collected them on the seafloor, that water feels really nice and cold.
Yeah, sticking your hands in there can be fun, especially if it's from 4,000 meters.
It's not so bad when we're here in the tropics, but in the colder part of the ocean, it could be a bit unpleasant to have to stick your hands in there.
When I was a grad student, again, going back to grad student stories, there was a friend of mine who was doing physiological work in amphipods. I may have told some of this story earlier in the expedition. This is Ron Kaufman, who's now at the University of San Diego. And he was looking at various metabolic features, respiration features of deep-sea amphipods. And so we would be collecting them in the Southern California borderland basins, which go down to 1,500 meters or 2,000 meters if you're in San Clemente. And so we could collect them, and just as you say, as long as you kept them cold, you could bring them to the surface, and then he would bring them back to shore in these aquaria and keep them cold. It was in a cold room, and so he could do many months of studies on these deep-sea animals. And I distinctly recall helping him with some of this work, and it wasn't pleasant because you would have to go into the cold room with them. And so you're trying to do all these measurements and manipulations, and it's very cold after a couple of hours to work with these things. It's, really? This is the way you like to live, amphipods? But the aquaria were not pressurized.
Yeah. Yeah, so the organisms that do have trouble with that pressure change are fish. Some fish have gas-filled swim bladders to help them maintain buoyancy. Not all fish by any means do, but many do. So if you bring those up, the pressure change will certainly affect them. People may have seen that before if they've gone fishing. You don't even have to bring fish up from too deep to have that gas bladder expand. You can sometimes see it come out the mouth or give the eyeballs kind of a popping effect.
Are there midwater fish that have a swim bladder?
There are midwater fish with swim bladders. In fact, some of the migrators do, which is always kind of surprising because if you've looked at the EK60 data, that migration happens fast. Sometimes half an hour, 45 minutes for them to go several hundred meters. So they have ways of being able to change the volume of the gas in those swim bladders. But we do see some gas expansion as we bring them up, but not like you do with like rockfish, say, some of the fish that don't.
We had a question Casey asked earlier about what does it sound like down there, and I was suggesting that, well, one of the main sources of sound we'd see in shallow waters might be fish, and one of the main way a fish makes noise is by kind of percussing its swim bladder. And so it's a matter of if a fish living at 1,000 meters actually has a swim bladder or makes that noise. And I wasn't really sure. I'm not sure we have that much kind of recordings of these depths. So that'd be interesting.
That's a great question. I don't know of anybody ever documenting the midwater fish making noise themselves, like the way that drums and croakers do, intentionally making noise to communicate with each other. However, an interesting observation some colleagues have made recently, they had hydrophones in the water to listen for whales and dolphins off the coast of California. And as they were going through the recordings, they realized that every evening around sunset and every morning around sunrise, they would hear this in the water. And they hypothesized that sound was coming from
static.
the actual.
That was a man making the sound of vertically migrating
Yes.
fish.
So yeah, they hypothesized that that was the sound of just all those fish and zooplankton just moving through the water, creating, small-scale turbulence, but on a large scale altogether, when you add it up, created this kind of whoosh noise. So I know they've been doing some follow-up work to see if that is in fact what that sound is coming from. So sometimes we go out there looking for one thing and find something completely different.
Yeah, of course, that kind of diurnal crepuscular behavior is very common in chorusing fish in shallow water, but this is a different mechanism here. So,
It's a cool little jelly there. It's a lot harder when you're trying to get the jellies to swim to you. There's something out there.
yeah, it's just a perspective, or these jellies look like they can really propel themselves.
Like that?
Yeah,
like they can propel themselves?
Like at a fairly good clip, or is it just our perspective? Or
that's a good question. It varies quite a bit from species to species, but some of them do move pretty fast, and certainly the siphonophores. We saw some yesterday. You could see their swimming bells pumping, and they can certainly really cruise. Some of the siphonophores that's a nice copepod. Some of the siphonophores actually vertically migrate, just like the fish do. And sometimes, if you catch them in the right moment, off in the distance, you'll think there's a fish swimming away from you, but it's actually the siphonophore kind of making a S-shaped motion similar to an eel. A lot of the organisms that we think of as being plankton, and plankton means drifter, so plankton canonically are things that don't really have control over where they are. A lot of plankton actually do have the ability to change depth, so they can't necessarily fight a current or decide where to go in the horizontal axis, but they are perfectly capable of moving up and down in the water column to follow certain features or light levels, like with their nightly migration.
go ahead now. All right, thanks, Neff. Go ahead, pilot.
Great. Thanks for the update. Thanks for the update, pilot. And we were all watching with interest back here, and glad to see you got the problem sorted out. We're ready to resume the dive, and we're ready to head up slope. So for the folks on shore, we would have approximately 2.5 hours left in our dive. And we're at almost exactly 1,000 meters.
Yep.
So how high up did we get before we came off the bottom?
We were a little above this.
That's what I thought, okay.
I have to look back at the... It was a little hard to say when we came off. Yeah, so.
Let's see where it was. I went to lunch. Well, we may still have been below 1,000. When I went to lunch, it looks like we were about 1,015 meters.
Yeah, maybe so.
Okay. Oh, I see my problem. I'm looking at camera 2. I'm like, wow, we're so far off the bottom. Sure, we can have a quick zoom on that. Thank you. This looks like another halosaur, Astrid. Is this what we were looking at earlier?
I think this is the eel that we were looking at before, and I've been trying to figure out if it's a snake eel or a congrid or a clopsid. I think the dorsal fin is too far back for it to be a congrid. It's got these bright white fin edges. The tail doesn't look like a snake eel because the snake eels normally have those very pointed tail tips so that they can dig back into the sediment, and they're actually exposed sometimes in those snake eels. So I wish Bruce was here, but that's what I got for this guy.
Thank you, Astrid. Thank you, pilot. Ready to resume? Yeah, so it's like we're on the same Victorgorgia. If you mean the purple one, or we've actually seen all the things in the view here, pilot. Yeah, thank you. This is a stock crinoid, is the yellowish-orange thing on the right. We've seen several of those today, and we see a small purple octocoral in the center. We imaged that earlier.
Yeah.
And then I think this is a primnoid fan that has several crinoids on it, and 2 different color morphs.
Did we come back to the exact same outcrop? Or?
I wonder.
It looks similar pattern. Yeah.
Is this a rock jumble? I don't know. These kind of look like they're in place. They're just kind of covered with sediment.
They do, I think, yeah. We're still looking for a rock. The bigger and more angular, the better. So.
Why is angular good? It means it's fresher?
Angular might tell us it's broken off of something bigger more recently. And, it's really a kind of diffusion problem at low temperatures, the water diffusing into the rock and altering it. So the less surface area
we don't
over a longer period of time is better.
Gotcha.
But we'll make a record that we saw one. Thank you.
We saw one right after that outcrop last time, too.
There you go. Dell's suspicious. Well, if nothing else, all joking aside, it says that there is a certain kind of a habitat at a certain depth, and we're seeing similar sorts of fauna and associations in that depth range.
Yeah, for Oreo specifically, people have noted that they're often in these depressions or in flow refuges or hiding out near rocks and things. So it's interesting to note these kind of habitat associations as we observe fish.
Thanks, Astrid. No, sir. We've seen all of the fauna earlier. In fact, we're seeing repeats again, so the lasers are just passing over this biflabellate primnoid colony, and I can see another homolid crab on there, and we've seen that repeated several times. They're the ones that are carrying their little parasols of coral above their head.
Scott, earlier you were talking about two different yellow corals. Can you just sort of describe to us visually what the difference we would be looking for the one that you're trying to?
Sure, it's a little bit hard. Thank you, video. So we are looking for a specific yellow fan, and I'm seeing on the right side of the screen there are two yellow fans. The one that is lower is a deeper orange color, but the one in the upper center, so Pilot, if we can get a zoom, upper center light yellow coral fan. So this may be the other morph that we're looking for. In fact, we've now seen 3 today. So we also have genus Acanthogorgia that's going to cause a little bit of an extra problem trying to find it. But from the distance that we're looking, we're basically looking for a pretty full-looking fan. And by full, I mean there's lots of branches, so there's not a lot of space. This one that we're looking at now from a distance looked like it had kind of space between the branches. So I'm going to predict that this is the Acanthogorgia, which is not the one that we're looking for, although I should say that every coral we see is of interest to have some part of it collected just to ascertain that it is what we think it is. But because we don't have a huge sampling program, we typically don't sample more than 4 things per dive. We are judicious in what we target for collection and make sure that it's either a representative, it's very common, so we want to know what is very common on the slope, or that it's something very unusual and we're quite certain it's a new species. So this colony does have that large brittle star in the center as schemated, and so the ones that we were looking for, the yellow fans we were looking for, didn't have these very large brittle stars in the center. So I would suggest that this is just a slightly off-color morphology of the other more golden yellow. So that looks like a kind of a typical Paramuricid octocoral. And I'm using that long term, Paramuricid. It's there was a family called the Paramuricidae, got knocked down to a subfamily within the Plexauridae. And the Plexauridae are where you have most of, or many of your typical deep sea fans. Thank you, Pilot. We can move on. This looks like, I guess that may have been a sponge once upon a time. That's my best guess of what we're looking at.
Yeah, if we could just take a, okay, yeah, I wanted to take a look at that. I wasn't sure what that was, and I'm not sure what that is.
It's old and crusty. It's been overgrown with a number of things, and there's a lot of this material that's falling through the water column gets bound up in old sponge skeleton. Makes it hard to identify.
And when we zoomed in, we actually could see more of those smaller circular and thin patches of sponge.
See another one of those eels off to the left. So, Pilot, just to give you some context, so we've seen a lot of these corals down below and some of the other fauna, and so we would like to make a little bit more headway up the slope until we start to see some different sorts of things. And as you know, probably as soon as we pick up speed, we'll start to see the new stuff. Is it my imagination, Del, or is the surface texture a little rougher?
Yeah, it does really have that look here. In case one of our questions we came down was where we're going to find carbonate on top of this, or actually lava. It's all covered with manganese, which makes it surprisingly difficult to say for sure. I haven't seen anything definitively today that I would say for sure is a flow texture, a lava texture.
Fish tucked into the corner here below the lasers.
So this may be carbonate material. It's crusted over, but.
Well, I know this is not a rat tail.
No, this is a cusk eel. This is an ophidiid. And I believe we've seen this one before. Let me go through before I make a wrong ID. But he's definitely a cusk eel. And you can tell by those modified pectoral or pelvic fins that he's got that have been moved forward and almost look like just those two filamentous bits. And you can see the lateral line here is actually disjointed. There's a break in those yellow dots that run along his midsection. And then if you look at the tail as well, you'll see the classic ophidiid tail where you can still see a caudal fin in there, but it's kind of contiguous with the dorsal and anal fins. Nice.
Astrid, I've heard you fish folks refer a lot to the lateral line. What is that lateral line?
So the lateral line is a line of pores along the midsection of a fish that is basically a sensory organ that the fish can use to get information about the environment around it. They're very sensitive to differences in pressure, and so it's a sensory organ, really.
Great, thank you.
Astrid, do all cusk eels have the disjointed lateral line? Or is that just certain species of
No, so not all cusk eels will have that.
Uh-huh. Interesting.
It's actually a useful ID characteristic we can use.
Uh-huh. Cool.
As we move over the bottom here, I'm seeing lots of crinoids. Actually, to the left of the lasers at about 10 o'clock on the edge of the rock, there's something that's kind of white with black tips. Not sure if this is a new crinoid or maybe it's just a dark crinoid on a white skeleton. I see. Yeah, that's what it is. Oh, let's see. 1, 2, okay, more than 5 arms. So this is not one of the ones that we'd heard before the dive, before the expedition was a collection target. But that is a crinoid, a feather star, and it's perched on a scleractinian coral. That is a stony coral. And earlier we had the suggestion that that would be in the genus Selenosmilla. Thank you. Yeah, very nice perch. I don't see any rocks.
If you can get one of those in the basket, and
maybe a quick zoom on the white fan coral to the left of the lasers. Thank you. So this is a primnoid again, the family Primnoidae, but the structure seems somewhat different from the ones that we've been seeing earlier. So this may be a 3rd or 4th species for the day. I'm not entirely certain, but you can clearly see all the polyps are pointed downwards. There are a couple of different genera in which that's the case. So this may be another species of Narella, but I'm not 100% certain. So a little bit of imaging will always help for our later identifications. And if nothing else, if we can't identify it, we can at least say that this is a same or different morphology or something that we saw earlier. Nice crinoid perched on the edge. It's a bit yellow. Might just be a function of its age. We've seen lots of the yellow ones, so maybe as they age, they deepen in color. It's another one of those possible congrid eels that we just saw earlier. Wait, yeah, there's like a goosefish or one of those sorts of fish top right. Or the bat fish, it's, one of those bottom. I don't know if well, hold on. Let's make sure it doesn't have lure. Oh yeah, this one has a lure, Astrid.
Yeah, so this might actually be what I thought was a goosefish. It might actually be a goosefish this time. We did see a species of goosefish earlier. And I think what we're seeing now is its really big mouth. So yeah, I think we'd be safe to say it is a goosefish this time. Very cool.
So if I understand, this is a kind of anglerfish that would be trying to attract its prey to close to it so that it can gulp it up.
Yeah, so it has a lure, and it tries to sit very still and attract a small crustacean or prey item towards its rather large mouth, and then it'll ambush it, so it's known as an ambush predator. Oh, there was another fish up in the Sirios view. I just saw it flit by. I didn't get a good look at that.
Beautiful imaging. You can see the those little spines. Not sure if those are spines or modified fins that are used as the lure. Yeah, you can see how well those fins are adapted to kind of grasp onto the seafloor, waiting for its prey to come near. All right, good observation. I keep seeing this eel, and I don't know if it's just the same one that's kind of moving through the local space here.
I'm pretty sure he's different. Looks different length. I think we're seeing several of them.
Yep. The white fan just left of center at the top of the screen. Yeah. Is that the one we just imaged, that fish, or is that another one?
Oh, I think it's the same one.
Okay.
I don't think so.
Yeah, that might be something different. It's going to be a primnoid, but what I'm seeing here is it doesn't have, it's not biflabellate. That means it's not two fans oppressed to one another. So this may be the first of the day for this particular species.
Not right here. Let's. Okay. Looks like a lot of this is pretty well crusted in. So.
Oh, I'm wrong. There you can see that there's actually two fans very close together. So this is in fact that biflabellate primnoid that we still don't know the identification of, but we did collect a specimen on the last cruise, and again you can see a homolid crab. So this is fascinating. Many of these corals we zoom in on have these homolid crabs, but we haven't actually seen the homolid crabs walking on the rock, have we?
No, I don't think so. All right.
But we know they must, because they were all holding bits of coral that were not the coral that they're on.
Ah, that would be rude, right? Yeah.
That'd be really in your face. Those are Amphipods pilot, small kind of brooding crustacean, very common in the deep sea, and you often see them hanging out on the corals, but they're not necessarily associated with the coral. That is, the coral to them is just a substrate that they're sitting on. That's a nice view. I suspect that's a fairly old coral that we're looking at there. It's a really large size. You can see that main skeleton that was, or the main part of the axis that was attached to the rock. It's really quite thick.
Huh.
There have been a variety of studies trying to age different kinds of corals, and there have been some. Oh yeah, is that a fish? I'm going to pause on that, or is that a sea cucumber? So it's almost above the lasers now.
There was a fish off
Sea
on the
cucumber.
left.
Sea cucumber, yeah.
Yeah, we saw this one earlier today.
I don't know.
It's another one of those eels below the rock up there, so they're not uncommon in this stretch of the seamount that we're traversing. Pilot, why don't we get us up? I'm not sure what you're looking at. Well, I was going to look at that eel that was just under that ledge over there in the center. Now it's left of center. Oh, yeah. Okay, we're going for the shark. We see a large shark here from Sirius.
Shark always wins.
Shark always
Abandon,
wins.
abandon the eel. Abandon the eel.
Oh, there he is.
There we go. He's looming out there in the darkness, folks. Believe us.
Huh. Wow.
Oh, he's got
Very
a copepod
different than
on
the
his
last
tail.
one we saw.
That's exciting. Yeah.
Look at the copepod. Well, he does have a copepod parasite on his tail. I do not know what kind of shark
Wow,
it is.
this is really deep for sharks. We've gotten lucky. The last couple days we've actually seen sharks. Normally we don't see sharks. This is great.
So, not to put you on the spot, Astrid, but what kind of shark? Looks like it's got a lot of lateral line pores on the head. That's a really interesting pattern.
Well, I can't say I know my sharks. Down at the abyssal depths, we don't see any. They actually don't go down to abyssal depths at all. So I'll be looking up my shark ID guides.
Doesn't have very much of a dorsal fin, I notice.
And it's got 5 gill slits I counted. Was that, did you also count 5?
Yep, 5. We're getting confirmation from Caitlin, our videographer. That's what she counted. Yeah, good stuff. And again, I'm sorry, but I'm going to point out that there's 2 parasites on it. There's one on its side, and then there's a copepod hanging on the tail. You can see these 2 strings getting waved back and forth. Those are the egg sacs. Don's done a lot of midwater work. I think he saw the copepod first, so it tipped him off.
It's a little banged up, the shark.
Yeah, he looks like he's banged his snout into a few rocks.
It's back inside, too.
I didn't notice. There's a question of whether it had claspers, and if it didn't, would it be female?
That's a big copepod.
Isn't it? Thank you. I mean, I don't know how anyone else didn't see that right away. The first thing I saw.
It seems enormous.
There's 2 of them,
2 of
actually,
them, yeah.
side by side.
It's that drag slowing him down here.
Yeah, sure, could be.
Really big.
So those copepods have basically burrowed their head into the shark and are sucking on its blood for their nutrition. And I'm not sure what the parasite is that's on its side. That could be an isopod. It could be another kind of copepod. There are some 5 different families of parasitic copepod, and their morphologies are all quite different. Les Watling is suggesting perhaps a sleeper shark. Oh, maybe he's kidding because he says seems rather somnolent to him. That is correct. Co-pilot, or nav, yep.
Yeah, just brush that coral there. Huh?
That's good for me.
That was great.
Well, I certainly think the sharp eyes of the front row deserve introductions, so when you guys are in a position, please introduce yourselves. Understood. I suppose as the pilots get ready here, we might as well introduce the back row again. I'm Scott France. I'm a deep sea biologist at the University of Louisiana at Lafayette.
And I am Del Bonensteel. I'm a geologist at North Carolina State University.
I'm Amanda Nepburn. I work for the Cooperative Institute for Ocean Exploration Research and Technology in support of NOAA's Ocean Exploration Program.
And in just a moment, you'll hear from the front row. What is...
This is Dan Rogers, sitting pilot.
And in the front row, off in the corner, you see Roland Bryan, video engineer, also from Global Foundation for Ocean Exploration.
And the last person in the back row is Caitlin Bailey. I'm also a videographer with GFOE.
And we are all here on the NOAA ship Okeanos Explorer, exploring the Pacific Remote Islands Marine National Monument today on a seamount that is provisionally named Whaley Seamount. We haven't talked about the names yet. We should do that at some point. And we're at a depth of 972 meters, working up the slope of a small cone-like feature that is on the top of a flat-top seamount. And, pilot, sorry, are we in a position just off the lower corner of the screen, lower left of the screen, that bright yellow colony? That might be what we're looking for, next to the brown one for sampling. So what we're looking for is to see if it has a large brittle star in the center of its branches. The ones that we saw earlier did not. They had a series of much smaller ones. And the polyp structure and the what's called the senenchyme, which is the tissue between the polyps, was quite different from these other sea fans that we've been looking at, that we've been referring to as Paramaricia. So I do see a homalid crab on the backside of that colony, but I don't see any brittle stars from this distance. And I don't know if it's just the shadow, the position of the light right now, but the axis looks kind of dark.
Yeah. I agree with that.
Yeah. Check out what my folks, the scientists on shore, have to say. I'm wondering if this is an Acanthogorgia. And if it is, it should be fairly spiny. You should be able, from this view, to see a range of scleroids up near the tentacles that almost look like points in color. And I'm not really seeing that. You can see parts here in the center of the main stem where the tissue has been removed, so you can see that protein-based axis underneath it. And while I'm waiting, so if you heard me on shore, folks, I'm wondering if this is what we were interested in collecting. I don't think it is. I don't think this is what we saw earlier and suggested a collection. This massive stuff may be both polychaete tubes and hydrozoans. So yeah, I don't think this is the same colony, so thank you. Thanks for that, pilot. Les Watling was doing a bit of searching online in the meantime, and he identified that shark as a giant Pacific sleeper shark. And he further goes on to say that the genus name for that shark is Somniosis, so a play on sleep, and maybe referring to how slowly it moves through its environment.
Yeah, if we can see if one of these rocks there is kind of to the right. Is there one that looks loose? Yeah, maybe right up in front here, one of these, huh?
That may be good.
Yeah, that kind of more round but angular one there, just to the right of the lasers, if that's a possibility. Or just up from the crinoid one, it's a little blockier. Yeah, the one the lasers are on now. We could.
Has a brittle star on it.
Has a brittle star. I was looking out.
Fine choice.
I'm looking out for you, Scott.
Choice. Yes, indeed.
Well, I collected you an urchin the other day, but it didn't come back.
Yeah, that's too bad. Yeah, so we spent some time looking, hopefully, in that rock box.
No sign of an
slipped
urchin
through
in
the
there.
opening. Nope.
Okay.
Fooled again.
Fooled again. Yeah, this is a rough environment here. Rocks. All right. We'll move on and look for another bump test candidate.
So basically, Del, my rudimentary understanding of the geology, this is effectively all draped and cemented together by precipitates right out of the water column?
That's right. Yeah, so these iron manganese crusts that form. Well, we've seen this pretty much everywhere except for the carbonate reefs we've been diving on, all the younger carbonate reefs. And this may be carbonate, some of this may be carbonate under this crust. It's just really hard to tell, actually. So.
For some of our younger viewers, can you sort of describe what precipitate is?
Yeah, thanks, video. So we go down in the water column. There may be certain elements, in our case, some metals that are dissolved in the water, and they can actually form a crust or come out of the water column and adhere to that rock surface. And through time, they build up. And in this part of the world,
yes,
a lot of the seafloor is quite old, so we can actually get very thick rinds of this stuff that are covering the rocks. And as you can see here, even things that kind of look loose are often more or less glued or adhered together.
That was a polychaete worm that swam by, and then we have one of those pelagic holothurians. We're going to get a closer look at this yellow fan. This might be the coral that we've been looking for since very early in the dive. And at first we thought it was one of these plexaurid fans, the Paramericiid fans. But when we got close to it, we saw that it was quite different and didn't have a brittle star in it. And we realized we don't know what it is, and we've never actually collected one. And so there were a variety of comments that this would be a sample worth getting. And you can see in the far left are the other Paramericids, and you can already kind of see there's some difference. There's also another Chrysogorgia there at the side. Huh. So what do we think? Is this the same thing we were just looking at? Yeah, this again I think is another Acanthogorgia, which is not the colony that we saw earlier. So, yep, it's got plenty of associates stuck in the branches there.
And a lot of amphipods swimming around.
A lot of amphipods, and so there were the last time the pilot had asked, what are all these little things swimming around? So that's kind of interesting. And
like
I don't want to give anyone the impression that we're not, hatchet fish.
They're hatchet fish.
We got a really good video of hatchet fish earlier.
Saw that.
I don't want to give anyone the impression that we're not interested in Acanthogorgia. And in fact, my student James isn't here, but that's what his PhD is on, so he might be really upset that I keep moving on. But
keep dissing.
The one target we had is something different. Yep, we've seen the Oreo several times. Yeah, so we have this trade-off on every dive where we've got a certain number of things that we're hoping to see, both in terms of biology and geology, but there's a certain distance we would like to cover. So today, we're actually diving on a very small cone, and it's completely feasible for us to go all the way from the bottom of that cone to the top. And so that's one of the reasons we're not stopping every time to see things that we have already seen before, because we really would like to reach the top of this summit. I see Nav is pulling back on the HiPAC screen. No, keep going on. Thank you.
Yeah, so we're at about 964 meters now. Looks like the summit is up around 880. It's at least that upper contour there.
We can have a quick zoom on the fish. This may be the same congrid eel. No, sorry, I was replying to a scientist in the chat room. Yeah, this looks to me a lot like what we had imaged earlier. Astrid, can you confirm?
Yep, I think this is the same guy we imaged earlier, so we can move on.
Thank you. That's good, pilot. Sorry, I was just making some conversation, some notes with the scientists on shore.
So Emil Petruccio is from Naval Academy here. He's just commenting that a video from 2015 by National Geographic claimed that was only the third time that shark had been videoed. So
wow.
maybe actually be
that's very cool. Fourth time now.
Fourth time.
Yeah.
Fifth at worst, maybe. But, so quite a rare sight.
It was a giant Pacific sleeper shark. Somniosis is the genus. So it is a sleepy shark. None of these fans, correct. I'm making an assumption that the yellow one on the right is that Acanthogorgia we've stopped for twice. Yeah, so there's a jellyfish that's just floating by. You can see it's a dark red color, and at these depths that would basically effectively appear invisible. Amanda's been telling us all about that during the midwater dives.
Have a rat tail up front.
Can we get a look at the fish in the center just going off the bottom? Is that what you were asking about, Astrid?
Yep. I just want to take a quick look. I think he's a rat tail, and I just wanted to see if he's the same as the one we've been imaging.
Yep.
So this is actually different. He's got those really long, extended rays on his pelvic fin, and that really long dorsal. So I think he's actually a Coryphaenoides, although Chris ID'd a previous one as maybe being in that genus. But very cool.
I seem to remember Bruce Mundy talking about something like a marlin spike rat tail. Would that be something along those lines? Osteid shrimp swimming by.
I can't say I know the common name marlin spike rat tail. Sorry.
Okay. Bottom right. Oh, that's a really small one. I think that's one of the midwater bristlemouth fishes.
Yeah.
What do you think, Amanda?
Yeah. Hard to tell from here,
yeah,
but
really
we've certainly
small.
been seeing them. Yeah, it's likely what it is. It seems like the ones we're seeing here are really dark, so they're definitely a different species than what we were seeing up shallower, and I'll have to see if I can figure out which are the common species in this part of the world to try and sort out which is what.
Sorry, it's a sponge. We had a look at that before. It's an encrusting sponge. Thanks. And the sponge may have had a small sea spider on it, though, if you want to zoom back in. Sorry about that, Dan. So on the left side, just left of the laser, I think that might be a little sea spider. No.
The tail end of that. That looks pretty cool.
No, they're not sea spiders. I was wrong. They're probably like dead baby crinoids.
So this one seems like a demo sponge. It's really very interesting. What's that now?
It's hard to hear you, Shirley, but I was wondering if it's actually a tunicate.
Yeah, Shirley, you're barely audible.
I'm wondering that. Oh, I'm sorry. Not sure why I'm barely audible. How about now?
No change. It
no
actually
change.
sounds like you're talking on a different mic than the main mic open in the room.
We can get a quick look. We've seen a lot of these in the last couple of days, but it's good to note that it's there. This is a Plesiodiadema urchin, long-spined urchin. Tube feet are just kind of hanging down there. Maybe it's also sleeping. Maybe it's the time of day for the sharks and the urchins to be sleeping. I think this squiggly coral that's in the center is probably a primnoid that's had a bunch of its branches broken off. Lots of fins up here.
Yeah.
So we had, and Malcolm Clark had said this earlier today, that as we were moving up this slope, one of the things he was hoping to see, that would be a lot of, there's something really good in the upper right. We got something new. Hope Les Watling's here. He's going to like this. So we expected to see a lot more of the corals as we went up. Yes, top right corner is a new coral for 2017. I don't believe we saw this at all on the earlier leg. The cousin it coral, right smack in the center. Or it's also been referred to as the Chewbacca coral. Yep. This is a good one.
That's
Thank
pretty
you.
descriptive.
So
yeah.
this is actually something that has not been formally described yet, but has been seen up around the Hawaiian Islands. And this is a relative of the Iridogorgia that we've been seeing. And so when we get in close, you'll see that the main stem is spiraling, but you see it's got a lot more branches. Good stuff. So I see Les is in the event log, the chat room, sorry. It used to be called the event log.
That's really
So
cool
this
looking.
is, yeah, it's really cool looking. It's so it's a Chrysogorgia. It's one of the gold coral family, and it. So the question is it in the genus Iridogorgia, or Les Watling had described a second genus, Rodan Iridogorgia, and so I believe he thinks this is in the Rodan Iridogorgia. And one of the things you can see as you look at the top, as much as you can see of the skeleton here, instead of forming these big graceful spirals, it's more like tightly coiled, twisted back and forth. So it's almost like the main stem has been twisted rather than growing up in a coil. And that's one of the major differences between Rodan Iridogorgia and Iridogorgia. Eric, there, Les has just chimed in. He's seen it now. He says it's the Wookiee. So, as I said, the Chewbacca sponge. This has a couple of crabs in it. I don't believe I've seen that before. Yeah. These are chirostylid squat lobsters. See a couple of them. One of them may have eggs. I'm looking at the lower one that's a bit more pink. They also carry their eggs under the abdominal flap. No, that's the upper side. So, oh no, that is the underside. That's the underside, but I don't see any eggs there. Oh yeah, thank you. Let me ask that.
Yeah.
Sure. Yeah, so Les Watling, who's described the only other known species similar to this, says this one has very long branches, much longer than what we have seen before. And so I suspect this would be nice to get a little clipping from the top. Yeah, I mean, this is a big, tall coral. Yeah. And these are fairly rare. I have not seen many of these, maybe 2 or 3 in all my diving.
I don't know. It reminds me of a sheepdog or something.
Yep. So pilot, just so you can set up and think about this as we go, the ideal situation would be to take a clipping that has at least one full twist. And you'll see that, yeah, it's going to be hard to see, but you'll see it when you're looking at the tip of this colony. And I noted it before as we were going by, and the twists are not very long. No, I want this thing to stay there. It's really pretty. So I have consensus on shore that this would be a good collection target. So unfortunately, Malcolm Clark had to leave, and I would have liked to have known if these are also seen in New Zealand waters. Yeah, you might be able to see, actually you can kind of see there in the center that, that tight twisting, that tight coiling of the main axis. Yeah, you can see it's almost like it's, if you took a straight axis and then you twisted it from the bottom to the top, that's kind of like what we're getting, instead of one axis that grows in a circle. Yeah, they're almost like waves instead of coils. And did we have the lasers on this at the beginning? Did we have the lasers on this in the beginning, just to get a sense of scale? Okay, thank you.
Yeah, we had them on a couple different times.
Thank you, Roland. Just so long as we had some scale, yeah, I see one of them there. So we're trying to, we got some questions about its height, and I'm not sure if we're settled down, but I would estimate this to be a minimum of 2.5 meters tall. Oh, that's plenty. Yeah, yeah, that's maybe too much for the, well, it's not too much for us. If it's too much for the box, it's never too much for us. Yep, lovely. And this should fold up really easily once it's in the box. Thank you. Yeah, it should cut. It should cut.
Yeah, seems pretty cooperative.
Yeah. I'm looking into my database at the last time we saw one of these and sampled one of these was in 2006, and it was at 663 meters, so it's really interesting that it seems like this is a shallower water thing.
Oh, yeah. Well, maybe we'll see more as we go up here.
That'll be fun. Yeah, it was like twirling spaghetti there.
All right.
Nice touch, Dan. That's coral in the business we call a floater. So you never know. You try to get in a bile box and just a bit of current or thrust pushes it off. Absolutely. Yep. This is not our first rodeo. Thank you. Great. So we have a successful collection there. And then, Pilot, once you're all stowed, before we head off, the large yellow fan on the left, been asked to check to see if that's the other one we're looking for. Copy that. We're just getting a switch in the front row and seats, and I just want to remind everyone we have about 1 hour and 40 minutes of bottom time remaining. Our depth is 959 meters, so we still have probably at least, close to 90 meters of elevation
I think
to climb.
880 was the highest,
okay.
highest contour we're seeing here. So.
Pilot, we are looking on the left side of the screen, lower left. You see there's a large yellow fan just sticking into the screen. Thank you. Some really large, impressive primnoid corals in the far background there. I'm seeing one that's white and very wide. It seems to be wider than it is tall. And I wonder if that's a Calyptophora or a Paracalyptophora. Yeah, this is that same Acanthogorgia-like one. So this is not the one that we were looking at for earlier. So I think we can move on. And I'm just going to consult with some of the shoreside science. And I have a feeling that we may have moved out of the depth zone for the yellow one that we were seeing, because I haven't seen it in quite some time. Actually, oh, look at that. Is a new crab for the day. A new crab for the cruise. So this is a true brachyurine crab. It's a true crab, meaning that it has a very highly reduced abdomen that's folded up under the thorax and tightly oppressed to the thorax. So unlike a shrimp or a lobster, the good meaty part that you're eating, this one is really highly reduced. And you see it's got a very robust claw there. I think I see another one tucked under the rock behind it. I believe it is the same type under the rock, yep. Looking for some help from shore for some identifications, if anybody has a sense of what this is. I also see on the rock above that crab there's a yellow ribbon. That's a stoloniferous octocoral. You can see it grows in ribbon fashion. There's several polyps that are popping out from it. So I'm satisfied here. What I was going to note when you were zooming in, look, I think these are all dead skeletons that are lying in amongst the rocks there. That's what it looks like.
Yeah, it's definitely something different.
Wonder what happened right there. So a couple of exciting things there that we hadn't seen before. A sleeper shark, and then that very large Rodan iridogorgia. I guess these large, long, platy rocks that are just kind of upper crust, not the thing you're looking for.
Yeah, we'd like to get some a little thicker.
Yep.
But it's getting a little late in the dive, so we may have to take what we can grab here.
It's a dead sponge that just toppled over. Looked like maybe an old vase sponge or something. So this is kind of interesting because we just saw a field there where we saw a whole lot of these very large fans, and now suddenly there's nothing.
Yeah.
Well, I wouldn't say nothing, but we don't see a lot of those large fans. Yeah. So I wonder if that has to do something with the step of, I don't know, something about those are at the base of a step and the way the currents are swirling at the base of the slope here.
Definitely seem to be a cluster of them. Yeah.
And they were different species, and so it's not something you could say was just a local recruitment because it was a good spot. There were many different species, or at least 4. I do see a Chrysogorgia colony down there. Got its pink dot in it for evidence that it's got a squat lobster in it. A couple more of the yellow fans now. Actually, again, they're sort of perpendicular to us, so it's a little hard to see because their faces are
into the current.
Correct, into the current, so you may not realize they're there until you're right on them. Yeah, I've seen several. 1, 2, 3, 4, 5, 6, 7, 8. So there's at least 8 in view. But it's still kind of thin. And I think we didn't see the Oreos when we first sat down, and then we started seeing them later, so I wonder if they're also characteristic of somewhat shallower depths. I don't see the anemone. I see a crinoid. Is the brown thing in the center? Oh, you're right. My bad. Yeah, definitely interested in this. I believe this may be a relicanthid, which would be also new for the cruise. Good eye, Don.
Yeah, we
of course.
We actually saw one earlier, but we did not image it.
Thank you,
An hour
Roland.
and a half ago.
Okay. Yeah, so this would be good. So this, by all appearances, is a sea anemone, but the molecular genetic data tell us that it is actually something at the same level as order as the anemones, but it's separate, meaning that it would be at the same level as you would say stony corals are to sea anemones, are to zoanthids, are to black corals, as they are to this thing. And it's been given the name relicanthus, and relic implying that it's a very old and ancient thing, and maybe saying something about the possible evolutionary history and its relationship to other anemones. So I'm being told, yeah, we saw these when I went to lunch. One of the things you can see about it is it's got these extremely long tentacles and that get really thin at the end. And we saw some, a year ago, 2 years ago, we were following the tentacles for several meters, I believe.
Huh.
They can get extremely big. This is not a huge one. And I think, let's see, Deb Glickson isn't there today. I think on Cruise She Was On, we were seeing a lot of them on sponges, and we were in an area that didn't have a lot of rock. And it seemed like they were using the sponge stock as the only thing they could.
Take what you can get.
Yep.
Yes.
Yeah.
This is Shirley. We saw a lot of them on the Mariana Trench National Monument cruise.
Thanks, Shirley. I couldn't remember if it was your leg or if it was Deb's leg.
It was actually on both of them.
Got it. So far as I know, there's only one species that has been described. I think it's Relicanthus galathiana, something like that. And I believe it was described from the Pacific Ocean. Oh, I think it was Relicanthus daphniae, named for Daphne Fountain, who's a very well-known, esteemed anemone biologist. There's so many tentacles, hard to get a look in there at the mouth, at the center of those tentacles, but it is there, trust me. I see another one of those hairy-armed, long-armed galatheid crabs in the base in the background. So that's good when video's happy. So at least the molecular data that were available suggested that this animal is more closely related to the black corals and the zoanthids than it is to the sea anemones.
Huh.
And of course, there are other characteristics of the histology of its internal digestive cavity that also point to some of those clues, but when you're just looking at it, and if you haven't collected it, that's why you could get fooled. Thank you, pilot. Let's get a zoom on the fish. Oh, I see something the fish is going to take us to that is also brand new for the entire expedition.
Thanks, fish.
Thank you, fish. It's passing over a soft coral. This is fairly unusual for these deep sea depths. Well, you're asking a coral biologist.
So this is going to be another ophidiid. This is going to be another cuskeel, although they're going to be really hard to ID from a dorsal shot. But I can go to cuskeel because I saw those modified pelvic fins.
Should we follow it, Astrid?
If you think you can get a lateral view, that would be helpful. It was also missing its tail tip, if you noticed that.
Okay, we'll see if we can get the lateral view here.
More tricky.
The fishes must know this, and that's why they put their back to us.
Shy.
Well, absolutely, absolutely. I would do the same. All right, I'm going to make an executive decision on that because we have seen a number of these cuskeels. I'd like to go back to that coral because we have definitely not seen that coral on the expedition, or even, I suspect, up here at all on any of these 2017 expeditions. So it's going to be a soft coral in the grouping Nephthia. And yeah, you have to go one more. Yep, one more outcropping. What's the word I'm looking for here?
Yeah.
Yeah, it's going to be still a little bit back. Okay. Well, you got good eyes. Yeah, I see it now. I didn't see it when you said that. So this is an Octa coral, and if you see those two yellow fans in the background, we've imaged many. We just collected a Rhodantha retigorgia. One of the things in common with all these Octa corals is they have this internal axial skeleton. And when we've seen the tissue peeled back, you can see, and I said it's described a protein. When we saw the bubblegum corals and the precious corals, I said they had sclerites that were all cemented together. So instead of an axial skeleton, it's kind of like the whole thing is cemented, and the live tissue actually runs through it.
Okay.
There's a swimming crinoid.
Wow.
So just sort of proof that they are free living and can move around, unlike the stocked ones. And there's a nice display of all the cirri that it uses to grasp onto the branches of corals. And I think he's putting himself into the bio box. This one, what we're going to see, does not have any internal skeleton. It will have sclerites, and the sclerites are in the tissue, and they're in the polyps, but they're not consolidated into a skeleton, and there's no axial skeleton. Instead, it's all fluid inside. And these guys can actually eject that fluid so they can kind of squish themselves up, kind of like a bubblegum mass, retract everything, and then they can inflate themselves again to the size that you're seeing here. And as near as I know, there are not too many deep sea soft corals known or described. I only know a couple of genera, and I only know those from the North Atlantic. I'm really not familiar with what's out here in the Pacific, so I know the genus Gersimia and the genus Drypha, but I really don't know what this would be. I know some from down around New Zealand and New Caledonia that look quite different from this, Scleronephthia, that have a lot more, they seem to be more rigid because there's so many sclerites, but these are not that. Yeah, and video's pointing out that there's a bivalve. I don't know if that's a mollusk or if it's a brachiopod, but down just above the right laser on the front face of that rock. We've talked about the Anthomastus mushroom corals, and we've described those as soft corals. They're not soft the way these are soft, because they are more like the cemented kind of bubblegum coral. They do have some rigidity to them.
Yeah, this thing's flapping around like jelly, almost.
Yeah, exactly. These are the true soft corals. Copy that, pilot. No, let's keep going forward. Yeah, we got a look that at least we know that it's these soft corals down here, and we'll keep our eyes peeled for more. Thank you. Tina Molosova suggested that bivalve that we saw was indeed a brachiopod, and that's a phylum of its own. Looks like another relicanthid here. What was it, anemone-like? Not Medusa, polyps. With the very, very long tentacles. So the brachiopods, they kind of look like clams, but they're not clams. Clams are in the phylum Mollusca. The brachiopods actually have a short little muscular peduncle that they looks like a little stalk. They hold onto the rock. And then they have a feeding structure called a lophophore, which is a couple of arms that have small kind of tentacle-like structures on them that are ciliated, and they generate a current to suspension feed. And they have a very rich fossil record. Oh, what's underneath that rock? Oh, it's another Relicanthus is underneath the rock. It's another one of these acanthogorgids with the homolid crab on it. That's the bright yellow-orange colony. Kind of look sort of loose. There's a bottle brush Chrysogorgia. Go ahead, Dill.
No, I was just saying, yeah, I think they just have a move in, but when they get in, maybe we can try again for another rock here.
So you have a Chrysogorgid colony on the right. That's a Chrysogorgia, a bottle brush type. Very dense branching, so this may be the first of this kind we've seen today. I see a shrimp in the upper branches. On the left side, the white one is another primnoid octocoral, family Primnoidae. And then you see a whole bunch of these white quarter nickel-sized things on the rock. Those are all sponges. And then I see a little polychaete worm just went off the bottom in the center. Actually, this very light brown colony, that's definitely an Acanthogorgia, so it may just be a small version of some of the big ones we've been looking at. Oh, and some of the ribbon-like purple Stoloniferous octocoral that we saw earlier. Another one of the, I think, congrid eels in the background on the rock. So I imagine, you know, you could have some rock. I'm sorry, I'm not sure if somebody's talking to me or. Yeah, sorry, I see.
Yep.
Yeah, we can have a quick look at it. Looks, might be dead, but sorry, I've turned you back on video. I had you off for a second there. There were too many voices. So it is a cup coral for sure, and it appears dead. Yes, so that cup coral means it would be a solitary stony coral. It wouldn't grow into a colony. That one's dead. It could still be identified because most of the taxonomic characters actually are the skeleton rather than the live tissue. Thank you, pilot. But it does tell us that at least at one time, these cup corals lived here. We would need to see some live ones to know if it's still an active population. So I was going to say, Del, that if we had, let's say, a mini landslide or rock slide or something, I suppose if that happened long ago, those could then become cemented in place, right?
Yeah, I think so. A lot of these rocks look like they were displaced at one point, but when a few times we tried to nudge them today, they seem to be firmly attached.
We're creeping up to that 900-meter mark. Oh, there's another Rhodaniridogorgia. This one has, if we can get a look, we don't have to get too much of a zoom, but this looks like a smaller version of the one that we sampled. and the branches aren't as dense, so you can more easily see that wave in the center of the main axis. I think I may see a little crab or something in the branches on the far side. So a side view like this would be good just to see the waviness of that central axis. Thank you. We don't have to stop because we got a really good image earlier. We actually collected a piece of this. Thank you. I just want to try to get a sense of how that axis is spiraling. Yeah, so you can notice at the bottom it's hardly spiraling at all, and then you got some twists and so on.
Oh yeah.
And one of the things you can kind of focus on is how these side branches are coming out, because they're telling you that this axis is actually kind of rotating about itself. The way Les Watling describes it is, imagine you had an axis and all the branches were coming out one side, just like a comb, and then you grabbed it from the top and you twisted. That's what would give you this sort of spiral appearance. That's good. Sorry, do we have the lasers on?
Yeah.
Okay. Oh, great. Yeah, okay. The lasers were on. Thanks. Emil is asking about what evolutionary advantage the spiral would provide, and I wonder if Les might provide an answer in the chat room. But one thing is, if the morphology is such that those branchlets are all coming off on one side, by spiraling it means you cover more of the water column by having those branches go out and basically sample different parts of the water column. I know that Les has talked before about some of the coiling bamboo coral whips, and that would be one way. If something is growing upwards, once it got to a certain elevation, it sort of curls over, so then it's starting to grow horizontally, and it grows in a coil to kind of funnel water through, I guess, so that you can capture particles more efficiently. So I know you're looking at this sort of slope, and what you want to see is something broken off right in front of it, right?
I mean, that would be ideal.
Yeah.
And, you know, that may be the only rock that's loose enough to pick up here, even in this environment.
Yeah, what about just to the left?
Let's give that a shot if you would.
Lasers,
yeah, we'll see if it's loose here.
Looks kind of big, but
I think the record's about 32 kilograms. So if you want to go for that. I don't know. I could barely carry that one up the stairs. I'm not sure I wanted a bigger one if you get in the basket. So. Yeah.
Yeah, that's going to be pretty big.
It's a good size.
I see some Anthomastus recruits there. Can we just get a quick zoom just above the lasers? There's some red dots there. I don't mean the lasers, other red dots just above the lasers. Just to the right of the lasers. Yep, two dark red dots. Yes, so we're seeing 1, 2, 3, 4 different Anthomastus colonies. I think it's actually kind of an interesting ribbon.
I was about to say, yeah.
Wow. For two of them. Wow. Well, that's interesting. You know, I've actually seen that in the North Atlantic seamounts, where it was almost like the new coral recruits were dripping down from the ones higher on the rock. Sea star, yep, white sea star on the rock on the left side. Another Oriole. I was thinking about that question that Emil asked about why, what would be the advantage of having a spiral morphology, and the way I answered it, I said, you know, if it grows and kind of has its branches coming out in one direction, one solution would be to spiral. And it leads to the point that you can't just evolve whatever you want. You know, you're at some starting point, right? You've been given some building blocks, and then you can modify those building blocks given the conditions, but you can't create something entirely new. And so different corals, starting from different starting blocks, may have different solutions to the same problem.
It's this
That is
swim in here.
another Oreo that is on its side, I believe. Yeah.
Yeah.
Two Oreos, one lower and one above it. Yeah, it looks like maybe, yeah, way in the far distance. Yeah. Looks like maybe a rat tail. Unfortunately, we're just going after a fish, and we saw, I think, a new kind of coral for the day. But if I'm guessing, if that was a bubblegum coral, there may be a lot more as we get onto the top of this feature.
Yep, so that's a rat tail, and I'm going to wager that it's in the genus Coryphaenoides.
Coryphaenoides. When I was a graduate student, there was a lab, George Somro's lab, that studied physiology of Coryphaenoides and its relatives, and one of the students had a dog named Cory for Coryphaenoides.
Oh, this is the one that Chris called Coryphaenoides, I think, and I was arguing that it wasn't because it actually has a very rounded snout. This will be some good imagery for us to debate about later.
Thank you. Yep. I'd really like to try to get to the top of the summit, so I'm trying to hold off on the number of things I asked for zooms on, unless it's something that's brand new for the dive, or it's a rock that you want to collect, of course. What about this? Copy that, Nev. Thank you. Do you think those are loose?
Yeah, if those
Is
are
that
loose.
good?
Yeah, that'd be great if it's
Yeah.
loose.
Can we check out the rocks where the lasers are, just to the left of the lasers? Because that looks like a jumbled pile.
It's up a little bit. It might be kind of hard, though.
Yeah. But you wanted us to consider how you could sit, too? No, I just meant you want us to find you a rock in a perfect location. That's not going to happen.
Yeah, we can barely find a rock anywhere. So.
Yeah. Not quite so loose looking once you get down here.
We found this a few times, yeah.
Yeah.
Oh, it's pretty
I don't see
solid.
anything moving.
All right.
All right. Oh, well.
Thanks. That was pretty solid, wasn't it?
Looks can be deceiving.
Yeah.
Looks like there's a pink orangish ribbon around that big rock where the shrimp is.
Pink orange ribbon.
You can sort of see it in the replay.
Yeah. So we have about an hour of bottom time left, and we are trying to get our way up to the top of this cone feature to see if the number of coral fans and sponges might increase as we get to the top. We certainly have been going through different patches here where we see lots of fans or fewer fans. Sometimes we see really big fans, and we've seen lots of small ones. And so here I see, Roland, what you were referring to before. We're just passing a rock on the left side there that has ribbons growing along the edge. Those may have been ribbons of barnacles, actually. Oh, I see. That's both a ledge and an eel. There's a couple more Oreos, or the same Oreos following us. I see in the center there's one of the purple Victorgorgia colonies. They really blend into the rock. These, yeah, if you're going to park it, if you could point left, those two white fans are interesting. They're off screen left now. No, they're off screen left. There they are. Yep. Thank you. Exactly. This is what we saw, or what I saw earlier, and I wondered if it was Paragorgiid. Oh, there's a rock. You're moving a rock right now with the skid. Yeah.
Look at that. Looking at biology pays off.
Sometimes. Good choice to settle down, Don.
Yeah, great, Don.
Look at that. And you parked it right in front.
See, you found a parking spot and a rock. So. Sample 2?
Yep. Masako wants to collect that rock.
Yeah, we can get some on it. We're also doing a little pilot change here, so just take another moment, and we'll see if we can grab the rock. All right. Thanks, John.
Yeah, if we're settled out, we were hoping to see something else there. There were 2 white fans right next to where we were collecting the rock. There's another one, well, you picked up move, but there's another one in the foreground just left of lasers. Maybe that's the same thing. So this is really interesting. This is not what I thought it was. This is not a bubblegum coral because a bubblegum coral does not have an internal axial skeleton, and this one does. So this is something else entirely. It's probably a plexaurid of some kind. It's a plexaurid sea fan, and I have no idea what genus or species. Thank you. It looks like a cusk eel tucked down there in the rock. And so, Pilot Nav, we were hoping to get to the summit, maybe at least to the crest, just to see if we would get a lot more of these large fans, get a sense of what's on the top. Understand that we are likely not going to get to all the way on the top, and that's just fine. Understood. Copy that. Okay, Pilot, thank you. So far, most of what I'm seeing in the image is stuff that we've seen earlier and got some good views of. I guess we might as well get a look at that eel. The sponge above the eel will be new, though. But let's just get a quick look. And yep, I think this is that same one. The congred eel, I think, is what Astrid said last time. Just above it on the ledge, there's a sponge that looked different. Yep, right there.
Yeah, we'll see if that stays a congred. I've been calling him white fin edges.
Congred with white fin edges, thank you.
That's a really interesting sponge. It's probably a tetractinellid sponge. So it's a demo sponge. I don't believe it's a hexactinellid. And all that, all those things sticking out from the surface are, I believe, yes, they are spicules, long spicules upon the sediment and stuff has been, kind of trapped. So really very interesting. But it's demo sponge. And you're right, Scott, this is first one we've seen.
Great, thank you, Shirley. And I'm also seeing on this rock face some new species of the mat-like Stoloniferous octocoral, very transparent. You can see it there on the left side of that sponge, and then a whole bunch below it. And then just to the right, I noted the yellow one that may be like what we saw on the slope of Jarvis Island. Great, thank you. Good stuff. Yeah, it's so interesting that these octocorals can grow in so many different ways. You know, making these fans, making that tall coiled spiral, the bamboo coral with the really bony skeleton, and then just these ribbons. And then we also saw a couple of the really soft ones today. Look more like anemones than corals. Lots of variations. Still several Oreos along for the ride. Pretty platy. What do you think? Does this have like the sense that it's a coral reef that's been encrusted? I noticed that was really pale in color, and I don't know if that's...
Oh, I kind of missed that. Yeah, I have not seen anything since we've been up here that strikes me as a primary volcanic feature. So my guess is that, this mound may have a volcanic origin, but I think what's below this crust, and this crust makes it really hard to see, it can be very deceiving, is probably carbonate.
Okay. Oh, so here's a homolid crab that isn't holding anything. So he must be on the search for coral or something. So you can see his little back legs up above his carapace, up but is not carrying anything. We've seen several today that have been on corals that have been holding other bits of coral. Yesterday or the day before, we saw one that was holding an anemone. So a rock that we passed over there, Del, was kind of scoured at the base. There's some erosion, and it was a light brown in color. And I don't know if that's just some weathering or if that was some indication that maybe there was something like carbonate underneath it. What's this red thing in the center? Just, yep, above your lasers. Looks like 3 Anthomastus in a row. Yeah, so 3 Anthomastus. These are what I was referring to as the mushroom corals, and I had said that they were kind of like soft corals, but now that we've seen soft corals, I want to distinguish them from each other.
Mm-hmm.
Great, thank you, pilot. You can see in the horizon there's some bigger fans coming up. That is, yeah, so there's a good, nice close-up of what they call the thread-legged shrimp, and you can see why it's called thread-legged.
Yeah.
Really long. It's got tiny little pincers at the end of those different legs, and it's got extremely long antennae that better allow it to sense its environment. And this one has its swimming legs tucked up underneath. They're kind of a dark red. They're under the abdomen, which would be, the good part of the shrimp to eat. It's full of muscle. And I can't tell if it's got eggs in there. It might be holding eggs. Looks to me like there's little berry-like structures. Yeah, I think that's a female. It's just packed with eggs. Lots of these different decapods in the group Pleosomata hold onto their eggs using the abdominal limbs. Thank you, pilot. Orange things just above lasers to the right, maybe a cup coral. No, that is an anemone of some kind. Is that a seriantid? Huh. I think this is... wow, I'm going to hesitate again. So what I'm trying to see is right in the center of those long tentacles, you can see what look like a bunch of grapes, orange grapes there. And I'm trying to see, are those retracted tentacles, or is that just no, that's just the raised mouth. That's kind of like the lips, so to speak. And I think it's pressed so much together that they're taking on that sort of cluster of grapes-like structure. Some kind of interesting anemone. I wondered if it might be a tube anemone, but I'm not sure. Good image, thank you. I will definitely send that to Tina. I see she's still in the chat room. I wonder if she's still awake. She's in Russia, so she's on a completely different time zone. We have people on the East Coast, in Russia, in Japan, and in Hawaii. All kinds of scientists who are contributing their valuable insights. Looks like a siphonophore drifting through the screen here. Nice, yeah. You might not know it, but that's a colony of hydrozoans. And I wonder if that might just be the end of the colony. There's actually a floating part and a swimming part, and I'm not seeing it. And they do release the long parts so that they can float and take up their own existence. So I'm not exactly sure if this is part of it or the whole thing. Kind of looks like I see the floats in there, kind of tucked in the coil. You can see all the tentacles. These are all the tentacles of the zooids that are sticking out. Cool. Thank you.
So Tina, amazingly enough, is still awake at 4:47 a.m.
4:47. Spent all night with us. So Tina believes that is an actiniarian, so a true sea anemone and not a tube anemone. Thank you, Tina. There's another one of those white fans. So that'll be a species of plexaurid that I'm not familiar with. Is no surprise. There's lots of plexaurids that I'm not familiar with. So let's see. This is another nematocarcinus. This one, the legs look a lot bigger to me. I'm not 100% certain. It might be the same thing. Yeah, another thread-legged shrimp. Also with eggs, I think. You can see it's really thick where she's got her abdominal legs pulled up to the body. So this might be to do with spring, and if we have a spring bloom of phytoplankton overhead in the water, although I'm not sure that there's really a seasonality down here on the equator.
At one north, yeah, maybe
yeah,
not.
Exactly.
Yeah.
One north, there's light all year round. Further north, when you get up into the temperate zone, you have to wait for the end of winter. So here's another homolid crab, and this time it is holding some coral up above it. And this is not the same coral that the ones that we saw further down the slope were holding.
No.
Which tells you it can use a lot of different kinds of things as protection. In fact, that is holding a hydrozoan. It's not an octocoral, that's a hydrozoan that it's holding. Which is interesting in and of itself because I haven't seen that hydrozoan yet. That's the second time that the crab is telling us that there's something here that we haven't seen. And the colony that it's on, this is a primnoid, likely the genus Narella. Not entirely certain about that. There are other possibilities. I feel a little bit bad for Tina that we've only seen one black coral and she's been with us all night.
Up all night, yeah.
There's a fish lower. Looks like another cusk eel.
Yep, so this is another one of the cusk eel. It's a bifid, so it's probably Dicoplastopoma. Oh, that one's so hard to say. I'll write it into the observation log.
Yeah, under the ledge. Stay tuned, Astrid. There's another one coming up. Not sure if it's the same. We can't linger on this one, so quick zoom. Yeah, it looks like another cusk eel with the broken lateral line.
Yep, that's the same one. Thanks for that.
Thanks for teaching me about these things.
Yeah, lateral line's pretty diagnostic there compared to some of these other features.
Yep. There's one of the relicanthid sea anemones growing under the ledge. We have a very bright white urchin in the foreground. Another one of the primnoid octocorals with the homolid crab on it on the right side there. So far, Del, is it your impression that those crabs have only been on those sort of brownish primnoids?
I don't think we've seen that crab anywhere except for there, yeah.
Yeah, I agree. So that's an interesting observation. And I don't know if that's just something about, those are pretty stiff corals. Those branches have a lot of calcium carbonate in them, and so they're really robust versus some of the yellow ones. Actually, I don't know if that was one that just landed on the ground. If you could, can you turn, no, I see. Never mind, pilot, it's okay. It's okay. And let's see. I think maybe a cusk eel from the top, but I'm going to let Astrid confirm.
Yep, I think that's what we're looking at. Another Dicoplastopoma. There we go. Now I said it. So those are brachylas, the bifidae in the cusk eels.
Brachyla, great, thank you. And I noticed another one of those interesting anemones tucked into the rock down there. What do we got going on here? So the white stuff fringing the rock, the ledge. So this is what we've been calling the zigzag coral. This is a stony coral. And I think it was the genus Madrepora, perhaps. And we have not seen this today. So we may be getting up into a depth zone that allows this to survive. So this is a stony coral, close relative of the corals that build shallow water tropical reefs. And then again, you can see on the rock to the right, another one of these stoloniferous ribbon-like octocorals growing all over the surface. There are various squat lobsters inside the stony coral. Sponge just below that we had been imaging earlier. So several clumps of these corals, and you can see parts of it are dead. The lower parts are dead. They're the older parts. But the tips, they're a little bit more beige in color. That has the live tissue on it. And every once in a while, you can see the tentacles of the polyps extending. Cool, great, nice view. So this is new for the day, and there's a cup coral. There's a live cup coral just up above there. One thing I'll note, I don't know, Del, if you've mentioned this today, but oh, are you kidding me? This is the yellow coral that we've been looking for earlier. Well, okay, so now at least we know it goes shallower. Just for whatever reason, it wasn't deeper.
Yeah, there was.
Or it wasn't when we were looking for it.
Quite a gap there.
Yeah. All right, well, so that's good to know. So what I was going to say is that at the very least, Chris Kelly at the University of Hawaii and his team in the Hurl office at Hawaii Undersea Research Lab, look, they're all over the place just putting it in my face. We've been looking for these corals for hours. Wow. Oh, this is great. This is why we wanted to get up near the ledge. Starting to get a bit more abundant and diverse and colorful. Yeah. So I was going to say that, we got really good images of that hatchet fish earlier.
Is this the same crab or a different kind of?
The crab, if we can see the crab, so this is not the homalid crab. That's very bright white with red legs just below lasers. Thanks. Yeah, so this is a chirostylid squat lobster. That's not a homalid, so that's a good observation. Right. That's what we saw before.
The way he was sitting there, I couldn't quite make it out.
And the reason we wanted to see it was to know, did the homalid get off of a coral? But it's not a homalid. And now we can see the 3 rostral spines. Those are those prominent spines that are sticking out between the eyes. And my colleague Tim Shank has been using those spines in part to help identify it. You can also see very prominent spines on the lower part of the chelipeds. Those are the arms that end in the pinchers. Thank you, pilot. That's great.
Another cup coral there.
Yep, another cup coral. That one's dead. So I was going to say that Chris Kelly and the team at the Hurl office will be annotating these videos, meaning they'll be replaying these videos and trying to identify and count as much of what they see as possible. And you can see we're getting into areas that are going to be making them work long into the night. And when we call out some of these observations we're making, even if we don't get a close-up, they have told me that that helps them focus their eye and so better get those counts. So it's good to bark out cup coral.
Yes, it does.
Go ahead, Tim.
No, it's Chris.
Hi, Chris.
Finally, I finished all my chores. Yes, it helps immensely, Scott, if you and others were to throw out some IDs there. So please do.
Thanks for that feedback, Chris. This is our first view of this bracingid today. So this is a kind of sea star. You see it's got its arms curled up in the water, so its mouth is underneath against the rock, and it fishes its food from out of the water column. And Chris, I think that might be different and new, maybe for certainly the last several dives. I don't remember seeing that morph.
Right, so I don't often see them with their arms curled up like this.
Look at all the Oreos. Sorry, Chris.
It is. I think it's Novodynia. And the one that Chris Ma usually defaults to out in this area of the Pacific is Novodynia pacifica. But that's my best guess. But you're right, it's not a bracingid. It's got a lot more going on its arms than your typical bracingid.
Great, thank you, Chris. Hopefully you're seeing the view now. We're seeing a dozen or more Oreos all near the top of this feature. We're getting really quite close to the rim at the top, and this is why we're trying to make this dash to see what's going on at the upper surface. Look at them all. This is great. This is probably like the first what I would call school.
Great image.
Yeah, this is fantastic.
Wow.
Yep.
Yeah, definitely the most fish you've had in any view, I think.
Absolutely. I think Astrid has fainted.
I've been fainting the whole dive. There's so many fish and such diversity of families.
This is terrific. Plenty of them. Cool. Les Watling was just passing on some information here about the homalid crabs that we've been seeing on the primnoid corals that are holding those little bits of coral up above them. Says there's a nice paper by Mary Wixton, and we often hear Mary calling in helping us identify the various crustaceans we've been seeing. Hold on. I think in the center here we have a very tall black coral. It looks like a feather. It's just to the right of the lasers. Yes, indeed. So what is this black coral? It's packed with eggs, I can tell you that. All the polyps, they've got that very peachy material in there. I think this one is ripe for reproduction. And I think this might be a Parantopathes genus. Tina, it's getting on to like 5 in the morning. We'll see if she's still hanging in there.
This is what you've been waiting for.
This is what you've been waiting for. Sweet black coral dreams just before you finally get to bed. This is a really tall one. There's one tiny little squat lobster I saw there.
Now, after you finish zooming in close on this black coral, can you zoom in closely on the rocks? Because I'd like to just take a quick look at those little spherical white things that may be sponges. Thank you.
You're welcome. Will do. Pilot, in case you didn't copy that, we have a request to zoom in on the rock. Go ahead, Chris.
Are those eggs? I mean, I've never seen a black coral that quite looks like that. I guess that was in a reproductive state, but is that what you think they are?
Absolutely, that's what I think they are. Yep, I definitely do.
Well, that's great. I've never seen that before. Again, maybe you saw some other ones today when I was out watching, but this was fantastic.
No, Chris, that's the first. It's only the second black coral we've seen today, and it's the first one of that species and the first one with eggs. Right in the center of the screen, just above the lasers, is this time there is a little tiny sea spider. It's, let's see, I can't see the lasers. It's almost at the bottom of the screen. It's slightly to the left. It's brownish in color. Looks like there's 2 of them next to each other, but it's only the lower one. It's okay. It's so small, we don't have to try and go and get it. But Shirley was asking about all these little nubbins on the surface, and some of them are sponges, but I'm not sure what that thing is. That looks like it's a 5-petaled flower. I wonder if that's a really tiny ophiuroid or asteroid. There's a nice looking reddish.
And the other thing too is that those little white spheres. Oh yeah, I see what you're looking at right now. Those little white spheres are ones we saw a couple days ago, and we weren't sure if they were snails or not.
Right.
But this close-up is giving me an idea of what we've been seeing with those circular patches, and that Chris and I were not sure if they were hexaxonellas or demosponges, all the way down when we were a little bit deeper as well. And I think they're demosponges. So this is very cool. Thank you.
Yeah, they have that feel of a demosponge, but they sure are tiny. You can see on the rock, there's all kinds of stuff growing down here on the rock. We could spend hours and hours just looking at rock surface. Cool. Oh, yeah, nice big anemone on the rock. We're looking at the back side of it, so you can see the yellow column, the body column, and then the tentacles blossoming from the top of the column. And I remember seeing something like this back in 2015, and it had several scale worms crawling on its oral disc. So let's see if we see those there.
Yeah, so these big, huge anemones, these brown ones, the best that anyone's able to ID these 2 is the family, and it's Actinostolidae. So we just call this one Actinostolid brown, and there's an Actinostolid orange. It's a pretty pathetic ID, but that's where we are on these giant anemones.
We need to find a giant anemone on a rock that Del likes. Then we'll be in business.
Yeah.
So while we're getting a nice image of this, I was telling you before that Les Watling was giving some information via a paper that Mary Wixton wrote about those homolid crabs, and they've been seen to carry all kinds of different things from bits of Gorgonian coral, black coral, sponges, or whatever might help them be distasteful. Huh. So that was published in the Journal of Crustacean Biology. Thanks, Les. Well, I don't see any polychaetes on the oral disc. Beautiful view of all those yellow plexaurid sea fans in the background, and there's one of our brown primnoid colonies on the left side, and it's got the homolid crab and its branches. That's what we were just describing, that carries all these different things to make it distasteful. Beautiful view. Beautiful view. Yep.
Yeah,
Copy
as
that.
you know, Scott, the big problem with anemones is most of the time you got to collect them to ID them because they're differentiated by their internal structures, their mesenteries, and that's where you can go further than family on them in many cases. So until one of those giant ones gets collected and somebody comes across the perfect one on the nice size rock, as you said,
Another batfish.
that Dell might want, then it's going to remain a mystery.
Yeah, you're absolutely right, Chris, and I'll say a little bit more about that in a second. It looks like now, oh, this is another one of the anglerfish, so a goosefish. I think we saw this morph before, Astrid, maybe the third time now.
Yeah, this is actually the same one we saw before. I'm thinking it might be in the genus Slodinia, but I'm not sure. It's definitely a goosefish, not like the batfish, although they look kind of similar in terms of weird compressed body shapes. But you can see the lure on this one and the big mouth.
Right, absolutely.
Great imagery, guys.
Yeah, I remember that batfish that we were looking at had a small mouth pointed down, and this you can see a big, huge, gaping mouth pointed up, and then it's got that lure just above it. Fantastic. Nice shrimp next to it.
So, Aspen, I think you nailed the genus on this. I do agree. It's Slodinia. It looks a little different than the Slodinia remiger that we've seen elsewhere, but I certainly agree with your identification.
Chris, stamp of approval. Nice.
Well, I'll tell you, if you worked at Hurl for 20 years and it's the pilot's favorite fish, and Hurl has gazillions of these images in its archive, you get to know Slodinia.
I was just going to follow up what Chris was saying about the anemones. We think it's hard to identify the corals, and there's another golden crab. We saw one of these earlier that had a lot of staining on the carapace, and we figured it hadn't molted in a very long time, and it was a female that was carrying eggs. So there's at least the second one today, possibly the genus Chasion, very common at these depths around the world. I've copied that. So, folks, what's happened is we've come up to this local high on the rim of our cone, and rather than go further to the southwest,
Right.
which would take us down into a local low point, instead we're going to stay in this high point and travel kind of along the contour. Thanks, pilot. So, yeah, the anemones, you would actually have to look in their gut. They have folds in the gut that basically subdivide the gut into little chambers, and it's the histology, the fine details of the structure of those folds that you use to identify them. It's really tough. I think this is an echinotheriid urchin. We were seeing a different species on previous dives. I don't believe we were seeing this white one with the pink on it.
No, I don't think so.
Yeah, and I don't know if the color has anything to do with their particular species. Maybe it does, maybe it doesn't. It's certainly a beautiful one. You can see the inflated tips of the spines. So there's tissue on the outside of some of those spines, and sometimes they are filled with toxins to ward off predators. We have a beautiful tableau. I'm not seeing where you're looking. Oh yeah, sure, yep. That is another sea star, and that was the one that we were seeing yesterday. I think this is a tremaster, if I recall. Let me, might even still have that. Yes, I do. I have it open still. Yes, Tremaster mirabilis. And one of the things I was reading on Chris Ma's blog is that there's very, very little known about this particular sea star, even though it's seen not infrequently in the deep sea. But there's little known about its biology. And you can see it. You can't really differentiate the 5 arms because they're kind of like filled in between, and it's unclear why that's the case. If we could get a look at that coral just to the left of the shrimp, it's kind of white in color. It's a stick, yeah.
Also, I wonder if you could look at the sponge after that that's directly below the ROV frame right now as well.
Okay, will do. So this might be that same Gorgonian that we were seeing earlier. Yeah, this whitish gray color. It's not something I recognize at all. Okay, thank you. And there's a nice Anelopsamia, I believe. That's the Stoney Coral that just went off screen to the top right. And the sponge, Chris, did you hear what direction he said?
Yeah, it said just down below.
Oh, yeah,
see
okay.
yeah.
That ball-like sponge on the right side.
Below right there, yeah.
Yeah, Chris, I think we were getting a close-up of this before. Shirley was saying that she thought it was a glass sponge.
No,
Glass?
No, it's a demo sponge, and it's a tetractinellid. And Chris, I think it might be a packagstrellid, or it could even be a geodea.
Yeah, that's what I thought from a distance. That's why I wanted to get another look at it. So
I'm
yeah,
sorry,
okay.
I was getting mixed messages there, Chris. Is it a glass sponge, geodea, or is it a demo sponge?
It's a demo sponge.
Demo sponge, thank you.
Yeah, it's one of Shirley's kids.
Okay.
My kids are the glass ones.
Understood.
These are deadly. Don't touch one of these.
Is that right? So they, because of the spines, because of the spicules, or because they have a toxin associated with them?
Not because of the spicules. Those things will go right through like double layers of latex gloves, and they'll be in the skin for a long time.
Wow, okay. Good to know, thank you.
All right, let's leave this alone then.
Don't worry, it won't get you from here. All right. Chris, is this goniaster of some kind? I am trying to remember.
Yeah, this is a goniasterid. This could be something like, maybe mediaster, but I'm not really sure.
Okay.
But they're definitely in the family Goniasteridae.
Saw a nice
Chris
cup.
Moss checked in at all today, or not yet?
No, he has not been with us today. I've been checking out his Twitter feed every once in a while, and it looks like other people are more active than Chris last time I looked anyway. And there was a cup coral just next to that. You could see this solitary little pedestal. That is one of the stony corals that doesn't grow into a colony. So clearly a lot of abundance of sessile suspension feeders up here. So sessile means that these are organisms that are attached to the substrate. They glue themselves down as a larva, and then they grow fixed into one place, and they're dependent on water currents to bring them food. We're looking at an environment here that is 870 meters depth. We are on Whaley Seamount in the Pacific Remote Islands Marine National Monument, and we're seeing this great abundance of sessile suspension feeders on the top of this feature, this cone that we're on. And then you can see every time we zoom in, there are all kinds of other animals that are associated with the colonies climbing up on them. Brittle stars, and we just saw a sea star, and now we're looking at this might be a dromeid crab, but it's a kind of crab. 1, 2, 3, 4. I'm seeing 4 pairs of legs, and I'm not sure. It probably has a reduced fifth set tucked in, maybe in the abdomen. So part of the point I was trying to make is that with all of these suspension feeders, they're providing additional structure. Is there a fish? No, that's a shrimp. It's okay. Keep going. And here's this big white fan that's coming up just in the center. This is another primnoid. I'm not sure, maybe Calyptophora, Paracalyptophora. And we have Chris Kelly with us now. He may recognize this. Definitely in a family Primnoidae. Lots of amphipods in it again.
Boy, it's sort of an odd angle there.
Yep.
I agree it's a primnoid. I'm not really sure which one, though.
Polyps are pointing up, Chris, upwards.
Yeah, is that plumate or not? I'm trying to determine.
I
whether
don't,
the
no, it's not really plumate. Not the way Caligorgia is anyway.
Yeah, really small polyps.
Yep.
So
it's an old colony. It's got a very, very thick stem to it. It's a big sucker.
yeah, I think Paracalyptophora, what you already tossed out, is a pretty good guess, Scott.
Okay. So we haven't seen this fish in a
Oh,
while.
look at that. You've got a
Oh, this is the Oreo.
Daphne
Sorry.
the Relicanthus.
Yeah, we've seen several of those now, Chris. We had some good image of it earlier.
Oh, okay.
Yep. See a couple more of these Novodynia Bracingid type asteroids. These are the ones that curl their arms up, the sea stars that curl their arms up, their orange color. Go ahead now. I can't see high pack. And can you look at the sponge pilot? Yeah, go ahead, high pack.
Yeah, that's a better look at that sea star. That's definitely Novodynia. They tend to have larger discs than Bracingid, and they have more robust arms with more spines on them, just to name a couple of differences.
Wow, that's a beauty.
Does that seem reasonable, Del?
Yeah.
Yeah.
That's one of yours, Shirley.
Yeah, I know. It's a demo sponge also, and pretty sure it's still in that same order, Tetractynelida, but oh gosh, I don't know. I can't get it any lower than that. This beautiful, beautiful imagery.
Are you sure it's a demo sponge? I think it might be a snowdrift up against this coral.
Yeah, really.
It's really interesting. I'm trying to figure out what's come first. Was the coral there and is the sponge kind of growing around it, or vice versa, or a bit of both? And this is that zigzag coral that we were talking about.
Chris, what do you think? Do you have
Oh,
any
yeah, I
ideas
do.
about that?
No, I'm just, within the last few years, I'm just starting to become aware of just how many demo sponges there are in deep water.
Do you want
I
us?
never really recognized them that much.
Do you want us to try to grab a piece?
The vast majority were glass sponges, Tetractynelids. So, I'm with every dive, we're seeing more and more of these. So, yeah, I don't know.
Chris, do you want to try to grab a piece, or Shirley, do you want to try to grab a piece? I mean, if this is something that you don't recognize at all,
Yes,
we
please.
still have like 15 minutes.
I
This
don't
would be the last thing.
I don't recognize it. I think it might be easy to grab a chunk. That'd be great. Thank you.
Chris?
Yeah, it works for me.
Pilot, if we can get a piece of this as our final activity, that would be wonderful. Thank you.
And I'm sorry, I won't be able to tell if it's brittle, crumbly, firm. I just don't know.
So, Scott, have you guys seen any of the blue plexaurids or any other colors besides the yellow ones up here?
No, we've seen a white one. We've seen the sort of typical paramuriceid sort of yellow-orange. We've seen some acanthogorgids, and then we've seen this, you may have been on when we saw these white plexaurids. We did see a Victorgorgia earlier, but I haven't seen any other color morphs beyond that, and I certainly haven't seen any other plexaurids that have the color morph blue or purple or anything like that.
Well, the reason I was asking is this is somewhat reminiscent of the swordfish seamount dive. I don't know if you remember that. It was in leg 3 of 2015.
I do,
Right,
and I know that I wasn't on that one. I wasn't able to follow that dive, Chris.
But it's lots of plexaurids. Yeah, lots of plexaurids, Acanthogorgia, Madrepora, and, most of them are right up near the summit, kind of like these guys are. So the depths are not that dissimilar either.
Perfect. Perfect. Beautiful. Spec 3, right?
Spec 3, yep.
Oh, awesome. That's really, really good. And then after they place it in the bin, if we can just get a close-up of what the interior of that sponge looks like, it would be great.
Copy that, Shirley.
Yeah, I'm curious.
Pilot, I know you're busy. Just in case you didn't hear, they would like to look at the inside of the sponge before you leave. I'm sorry, did that go in port inboard? Thank you.
While he's looking at that, Del, do you have any idea what type of rocks these are? Did we ever get, or did you ever get into volcanics? Do you think this is some type of manganese-crusted sediment or carbonate, or what do you think?
You know, I was keeping an eye out. You know, these when you get these crusts, it's really hard to tell sometimes. I never saw anything that was convincingly volcanic to me, and whereas on some of the other seamounts, we certainly have seen pillows and collapsed lava tubes. I never saw any of that, and that sort of blocky appearance that we're seeing here looked to me more like it's going to be carbonate. We did get a piece, so that'll be the something we can look at for sure. But you know, this whole big mound that we're on might be volcanic underneath, but it looks like this was shallow enough. We may have built some carbonate up on top of it.
Very nice.
I see.
Thanks very much.
So, Chris, do you recognize this zigzag coral that we're seeing up here? Casey was wondering if this might be the same thing that we saw on the slope of Jarvis.
Yeah, it looks like madrepora to me, Madrepora oculata. Every time I've collected a zigzag coral and sent it to Steve Cairns at the Smithsonian, he's come back not only with madrepora, but Madrepora oculata.
Great.
So I don't know. I was sure I had something different, at least on one occasion, and he said, nope, the same one.
Okay, great. That's exactly the information we're looking for. Thank you. That's kind of an interesting sort of, erosion below and different color crust on top.
Yeah, it's a kind of classic dark crust on top. You think it's some biological coating on the upper surface?
Oh,
or
the brown stuff?
Brown,
I don't know.
Because the lower stuff is more that classic
Sure.
manganese crust that we've been seeing. Yeah.
Well, I mean, I suppose there could be, microbial reworking. There's something is taking advantage of some of the minerals.
Sure.
Who knows?
Yeah. Now you're going to have your scraping crust off for OGL, aren't you?
Hi, Casey.
Were you able to get a rock here, Del, or did you just not see anything that you thought was worth collecting?
Oh. Oh. Okay. Sorry, say that again, Chris.
I was saying, did you manage to get a rock here, or did you not find anything that you thought was worth collecting?
We did get one down a little bit lower. So it was hard picking. Most everything was crusted in, but we did get lucky and find a pretty good chunk, I think. So.
Oh, good. Well, shall I give you my best Chris Ma imitation?
Go for it, Chris.
Well, I think I can get the family. It looks like a pteroasterid to me, a cushion star. Although it didn't look very cushiony, it surely looks pretty spiky, so that'd be a pretty uncomfortable cushion.
Yeah.
But I'm pretty sure that's what the family is. Going farther than that, maybe it's a pteraster. But I really don't know.
It's a pretty big one. Let's see.
Yeah, would you, Scott, in your opinion, would you agree with that it might be a pteroasterid?
Yep, I think that's a pretty good guess. Kind of looks like it's standing up on a couple of legs there. I wonder, I'm very curious to know what's underneath it. And there is, huh? Ah, yeah. Wow, look at that. Very interesting. It's just kind of a funny view because it looks like it's something that's standing on two legs. It's got a couple of arms wrapped around.
Trying to decide if it's going up or coming down.
Right.
Yeah.
So I was trying to get a sense with the lasers on it if that might be, let's see, 30 centimeters from tip of arm to tip of arm. Maybe only 20, 25 centimeters.
I'd say more like 20,
yeah.
A little more.
Okay, still, that's a good-sized star.
All right.
Shoreside scientist, he may be as thrilled as I am to hear that we have 34 minutes remaining in the dive. At one point there was thought we would come up early to do some testing, but we're going to come up at our normal dive time.
That's like an overtime.
Yeah,
All right.
It's like overtime. Exactly.
Excellent.
So, yeah, so should we keep going then? I'm not sure. Is this feature going to run out?
Hard to say from our maps. I know we've had pretty good luck kind of going along here.
Yeah.
Unless you want to try to go further towards the center.
I'm having a good time.
Yeah, I think this is working for me.
Yeah, I think let's follow the feature.
These little white ones, Scott. Do you think these could be Kushinoye? And the reason I'm asking is that swordfish seamount community also had Kushinoye on the summit as well. It's just, it's just very reminiscent, very similar type of community. And these look like they might have the polyps out. Maybe get a better idea of whether it's a Paragorgia or a corallid.
Sure, so we'll have a zoom on it in a second, Chris. We're talking, Pilot, about the white fan that is just above the zoom that we have now. Chris, the reason I have not been calling it the Paracorallium is because we saw one earlier with tissue scraped off and it had a plexaurid skeleton underneath it. Now this one is a little bit more flexible, so maybe it's different. But the one we saw earlier, either it was an overgrowth in the sense of Anthothela overgrowing something, or it was some kind of plexaurid that I'm not familiar with.
Hello, this is Chris Ma.
Chris Ma, welcome. It's been quite a day. Sorry, Chris Ma, can you just stand by just for a second?
Sort of off and on following.
Yeah.
I'm a bit late to the game, I'm afraid. Are you still looking at that white sea star?
No, and if you could just hang on just for a brief second. So, Chris Kelly, as we're looking at this close-up of the coral, what's your impression? The way it's shaking to me says Paracorallium and not plexaurid, but what do you think?
You mean plexaurid, not Paracorallium, right? The Paracorallium, that wouldn't shake with it, right?
Right, that would be a sorry. Yes.
Yeah.
Yes.
Right, left.
Yeah, I agree. This isn't Paracorallium Kushinoye at all. Now
Okay.
let's get a good look at it.
Well, so the question is, do you recognize it?
You think it's... no, I don't really recognize it. Yeah, no, I really don't have any idea, Scott.
Yeah, that's unfortunately the way I felt too. Is it definitely had a Gorgonian type skeleton, so that's how I knew it wasn't Paracorallium, or Pluricorallium. But other than that, I don't know. So Chris Ma, sorry, yeah, we are not on that sea star anymore. But if you want to give us just a little information, just keep in mind we're not seeing it anymore, and we've moved on.
Yeah, no, I figured that. My home internet is in a permanent lag sometimes. The main reason, mostly Chris has been doing a really good job filling in on a lot of the species. That particular one, however, so that's not a pterastrid, not a slime star, but is in fact a goniasterid, a hyposteria, one of those sea stars that feeds on coral. And if you'll go back and look at it at a convenient moment, you'll notice that one of the arms is actually kind of half sort of perched around one of those little yellow corals that was on that little ledge. It's interesting because it displays far more spination and is a slightly different color than I'm used to. So that makes me wonder if it might even have been something a little new. And, the fact that it is so spinos may really have contributed to this, perhaps why it was not recognized immediately, because, Hyposteria is a fairly distinctive animal, but the incredible the physical number of spines and the evenness might have just been a little disorienting. So, yes, your Brusingid that you were seeing is Novidinia, probably Pacifica, as Chris mentioned. And I think you guys also saw a really nice goniasterid feeding on a coral, like within an hour of the dive. I didn't actually see that myself. And that one, I think, was Cerciaster, which is the same genus of goniasterid that you observed in the Atlantic, feeding on those fallen Paragorgia. There are two of them in this area, Sandrae, which is named after Sandra Brooke, and Cerciaster pullus, named after Craig Young. Pullus is Latin for young, but I didn't get a chance to see the arm plating, so I couldn't really make a good species determination on it. But lots of interesting things today. I've been sort of peeking when I get a chance between sort of cleaning the bathroom and that sort of thing. But really an interesting one. And if we see another one of those kind of white Hyposteria things, that would be my vote, is to pick that one up, because I think that one is different from the others that we've so far. So thank you.
Chris, before you go, did you see that goniasterid before the Hyposteria that was just sitting on the rock?
Yeah.
Looked like it. Do you have any idea? Was that Mediaster or something else?
It looks like Mediaster. It can be really hard to tell from when they're really nondescript like that. The plates looked right, but it's one of those things where every little bit of resolution helps, and I think I'll need to wait and actually look at a high-res image, or it's just possible that some of them you just can't tell the surface texture from the look that we made of it. So I'll have to look at it again. I did see it, however, and Mediaster sounded right, but it could have been a couple of other things depending on variation in the surface texture that I need to see more detail for. So.
Thanks, Chris.
Oh, it's my pleasure. I'll talk to you guys later, or you're almost done, I guess. So have a good night.
Thank you, Chris and Chris. Very interesting. I'm actually glad, Chris Moss, that you said that was a Hyposteria, because that's what I thought it was at first, and I kind of defaulted to Chris Kelly, and I jumped on this bandwagon, and I should have stuck to my guns, because it did look to me like it was feeding on a coral there.
It's always fun to mislead, isn't it?
Yep.
I'm glad Chris called in and corrected that.
We're looking at a surface of another large sea anemone. You can kind of see all those lines are extensions of where the different tentacles are. And to some degree, that tells you something about how subdivided up the internal part of the stomach is. We were talking about the mesenteries, and so if you sort of continue down from where those tentacles are and imagine there are sheets of tissue, kind of like curtains hanging in a room, there's going to be very many of them, and they kind of divide the space up so that you can get maybe more efficient digestion in a smaller space with the enzymes. So Tina Molitsova had said about 5 minutes ago, it's unfortunate we missed it, but she thought she saw an octopus. Yeah, but that was 5 minutes ago, unfortunately. I didn't notice it in the chat room. But I got everyone's attention now. Yeah, really interesting. The surface seems to be holding this rock together.
It really does,
whatever it is.
yeah.
So what can you tell me about these little columns that are underneath here? Does that tell you anything about the geology, or is
It's
nothing?
really hard to see, yeah. They do look like columns, but yeah. Yeah, the rocks are certainly less blocky here. We're getting more pieces that are falling out, but you know, when we see columns or cooling features sometimes in rocks, but not usually in the submarine right at the surface like this. So my guess is still that we're on top of some carbonate material here, but.
Tina
So, Del, those little holes in it, those wouldn't be vesicles or anything, would they?
No.
Just that one seemed to have a bunch of pock marks.
Yeah, I wouldn't think so. I think they're more likely to be actually the carbonate. At these depths, we wouldn't expect big vesicles. We might have some lava tubes or something like that. But we saw a lot of overhangs like this when we were diving at Jarvis. And so it may be a similar kind of thing here where we get blocky pieces of carbonate. We also get more clastic sections that kind of weather out differently. So that may be the gross morphology.
This is interesting. Here we have a whole series of anemones with these fairly rugged body walls. They seem kind of green-brown in color, and then you can see the oral disc and the tentacles are more of an orange coloration. So these may be fairly new for the dive today. Quite numerous in this little pocket. You can see all the broken up coral skeleton rubble that eventually will turn into sediment, I guess, if it keeps getting jostled and broken. Thank you, pilot. Tina Molitsova made an interesting observation that I'll have to remember the next time we're doing these zooms on the that white octocoral, and Chris and I were talking about, could this be a pleurocorallium, or could this, oh, there's a new fish in the background. I think that's a new large fish, and I was going to say a grouper-like thing, but don't want to put myself too far out there. Oh, maybe it's just an, I don't mean to say just another batfish, but just another goosefish. Yeah, it's another one of the anglerfish. I can see its little lure at the front. Looks like the lure is bent forward this time instead of standing up above the eye. Yeah, you can see it hanging out there. I see this might be a different species. I see a little white tuft on the end of that lure.
Yeah, it looks a little different, doesn't it?
Yeah.
Yeah, I think this is different. I'm going to try to see if we have any more images of these guys in the goosefishes in the ID guide, but it definitely looks different to me.
Did the other one?
I don't think this is Slodinia. I think it's a Lophioides of some kind. Just looking at all of, but I could be mistaken there. I don't know. This is different, I think.
Yeah, I don't remember this pattern. I guess is that the lateral line there, or
yeah.
As it looks like? It has a very distinct pattern that I don't think we saw on the other one.
And what I'm looking at is the end of that lure, there's a little white tuft, and I don't remember seeing that on the previous ones that we looked at. Imagine it could wiggle that around.
I'm not sure we've ever seen this species. I don't think we've ever seen this species of goosefish. This looks completely different than any of the ones that I've seen.
So long as you're not proposing a collection, we'll continue to image this. That was a joke.
Might be able to get it.
Yeah, he's keeping an eye on it.
I'll dare you to try.
Yeah.
It won't fall for it, Astrid.
Yeah. We're not going to do it. We're not doing it.
It's not Wild Kingdom here.
Cool, great. Some more of that really brilliant purple, what I called the royal purple octocoral earlier. perhaps the genus Clavularia. That is a group of octocorals that really needs to be studied well. And they're really hard to collect in the deep water, because you usually have to get a rock or something that it's growing on, and you can't just scrape them up. Another Relicanthus, the anemone relative, but it's not a sea anemone.
And there's another tetractylid sponge, probably either a geodeid or a packet strelid, so it's a demo sponge. This one is another gigantic one. Same thing.
So it's interesting how we've come into this area and suddenly these Madrepora zigzag corals, all these little white clumps, have become really, really abundant. I see a couple of the pancake urchins on this wall behind the Oreo. Yeah, look at that. So, you know, it's kind of interesting to wonder, well, why are they all breaking up? Why aren't they just remaining in place? And I'm not sure what the answer to that is, but what I can tell you is in shallow water coral reefs, there are a lot of bio-eroding organisms. There are many worms and molluscan snails and things related to snails and various other organisms that will all drill into the coral skeleton. Some of them can secrete an acid to kind of wear away at the skeleton. So you've got the combined process of the corals depositing the calcium to build themselves up, but also the eroders digging in to make themselves little homes. A lot of sponges, in fact, will do that, will erode their way into the skeleton of these corals. So that could weaken it, and then you get a good strong current, they break off and end up in these piles. And it is certainly possible that sorry, I should unlatch too. It is certainly possible that some of the coral skeleton, if it's live and it breaks off, if it wedges in the right location, it can continue to grow so long as it's stable enough. And then once it starts depositing calcium carbonate against one of these substrates, a new colony can form and be more stable in place. This kind of looks like the goniastrid that we were imaging earlier. I think that Chris Kelly was asking Chris Ma about.
Yeah, it could be. I'm going to let Chris handle this. So we've got the Chris Ma bat light out, right?
Yeah, I'm not sure. He may have had to go back to cleaning the bathroom. There's a bunch of cup corals above this little goniastrid star, and these are a different color at the very least. And you can very clearly see the cup. It's made of calcium carbonate, and then you can see the soft tissue, which is settling on top of it. And just to make it perfectly obvious, in case it hasn't been, the soft tissue of the animal here is what is secreting that cup. The cells actually lay down the calcium carbonate. And, you know, all these different species there's many, many small details about the skeleton that allow us to distinguish them. So you could just use the skeleton. There's a nice golden crab playing King of the Hill. Copy that. So the pilot's telling me there's a lot of current up here, more than we had before, which is exactly what we expected when we came up here, and which is why you're seeing all of these various corals thriving up in this area. Let's see what's going on. Yep, you can see this crab is carrying purple eggs tucked under the abdomen there, and the things that kind of look like fingers coming from either side, those are the pleopods, the abdominal legs. And when we see the shrimp that are swimming and they're beating those abdominal legs, these are the same thing. It's just that the abdomen is highly reduced and curled under the thorax. Very nice. This is the second one of these that we've seen with all these eggs.
Yeah, that's right.
The other one was on one of the other dives.
Yeah.
Couple of barnacles on the shell there. So it hasn't molted in a little while, but it's not too covered. It's not as much covered as some of the others I've seen. I think I'm noticing on the rock to the right now where the lasers are passing over, you can see some brown lines over that rock, and I think that's probably a line of barnacles. Dead barnacles by the looks of things.
Ah.
If that's what it was. Yeah, I'm not sure anymore. Could have been maybe one of those large sponges that's broken away, and that's just the outer margins. I'm not sure. Thank you. Yeah, so I just said that last one had molted relatively recently, but this one looks like it's been a long time between molts. It's only got 2 legs left on the right side. You can see a lot of black staining. There aren't that many barnacles that have settled on it, but
No.
this is probably an old one. I wonder if anyone unsure can tell me how long these gold crabs and the red crabs, basically the Chaseon crabs, are known to live. I'm pretty sure that it's more than 10 years. So yeah, I think you're right, Pilot. She's carrying eggs. One of the things that they will do is they'll get in these high spots and they'll kind of open the flap up to make sure there's water and oxygen getting to those eggs. And no doubt trying to protect them. Definitely her.
She definitely needs a little lesson in personal hygiene.
Yeah, I definitely
taking
agree.
it long the most.
Maybe that's why those homolid crabs look so clean. They're using those corals they're holding to scrub their carapace. So we got lots of Oreos. We're zooming in on, not sure if this is a Xenophys.
Yeah, so this is a Xenophys. Different than the congers we've been seeing with the white fin edges. It's a different family.
So what are we looking at, Astrid, to say that? I guess maybe the dorsal fin. I've just realized, oh, no, there it is. Okay. I was like, I don't see a dorsal fin. It's got maybe a pointier snout, maybe?
It's got a nice notch on the end of its tail. That's characteristic of the Xenophys. That can help. It also just kind of has a different, I don't know, it just looks like a cutthroat.
Yep.
Looks like a Xenophys.
I'll throw out a couple things. The lateral line pores were white, which probably means that it was an Iliophene in that subgroup there. And also the Xenophys, the mouth actually extends back behind the eye. So the mouth is quite large in comparison to the congres. So one of the things I look at when I see a Xenophys eel or suspected one is how far the mouth goes back.
Great, thanks for that, Chris. That'll be a helpful identification tip. Now the trick will be just can I remember it in the heat of the moment. Had a really nice view there of Hydromedusa. That is a small jellyfish that's in the class Hydrozoa. And you can see it was pigmented a deep red color. And because red wavelengths don't penetrate into the water, you would expect, don't penetrate very deep into the water. They certainly don't get to these depths. You would expect that any visual system down here wouldn't actually be set up to interpret the red color, and so the red would appear as black and essentially be invisible. And what do we have here? We have a ctenophore. So this is a gelatinous animal, sort of distantly related to the cnidarians, which have all the other jellyfish in them. And it's got these two very long tentacles. We were seeing these a couple of times during the midwater dives the other day. And you kind of see those rainbow colors. That's the main part of the body. The mouth is currently pointed upwards. And what you're seeing are the reflections from bands of cilia. They're called comb rows. They'll have 8 of these rows of cilia that are organized in bands, and they beat those in order to swim. This is the largest animal that is known to use cilia as a method for locomotion, instead of using fins, or as jellyfish do, pulsations of their bells. And then they have those very long tentacles that basically are fishing through the water column for food. And let's see, another goosefish, I imagine. Yeah, I see a very wide mouth. So I think this is another of the anglerfish, but I don't think this is the same as the one we were just imaging. This is more like the one we'd seen earlier in the day.
Yep, I agree with that.
So, Astrid, do you think this was another Slodinia? I don't think that other one was necessarily Slodinia, the last one we saw. The one before that, I think we thought it was.
Yeah, if we could get a good look at the lure, that'd be helpful too. But yeah, I think this one is more likely to be Slodinia. It looks like it to me.
Yeah, I think so. That would be my guess on it, now that we got a better look.
We have about 8 minutes left of bottom time, folks. So we'll get our final impressive views.
Just enough time to find that octopus.
Yep. Or another shark.
I just want to see if I could conjure up an animal. So.
On the left side, on the flat slope, there's a whole lot of fans, and I'd like to get a sense of what those fans are. So this is kind of interesting, a little perhaps edge effect. Yeah, Chris Kelly, so here are a whole bunch of these white colonies that I'm pretty sure are plexaurids of some kind.
Yeah, what do you think about Unicella? Do you think that's a possibility, Scott? Or
I'm sorry, I didn't catch the genus, Chris.
I think it's Unicella, but let me double check.
Oh, Unicella, I
one
see.
that was described. Yeah, because they kind of look a bit like that.
Yeah, Unicella, that's not a gorgonid, right? That's, let's see, we got a Sacco here. She might...
Well, maybe I have the name wrong, I think. Yeah, no, Unicella is a plexaurid.
Oh, it is, okay. Well, that's certainly a possibility. There's several of them up here, and whenever I see this sort of thing, I'm always led to ask the question, did we have one colony, one larva settle here, really good spot, and then as it grew, it's reproducing, other larvae are settling nearby, and that's why you get this patch of lots of the same species in a close spot.
Do we know anything about the longevity of the larvae?
Fantastic question, and so I don't. Certainly not for these deep sea corals. Some of the studies that have been done in shallow water, it's kind of all over the place, so some of them can survive for weeks in the plankton. Others are crawl-away larvae, so they're probably not going very far. Some are brooded. So there's an area ripe for research, kids. You got to figure out some creative ways to study how far a larva can travel and how it can survive. And you have to keep in mind that it's the temperatures of the deep sea. What's the temperature down here right now is about 5.3 degrees plus the pressure. And so you can't simply bringing it into a tank in the lab, although people do those sorts of studies as well. You just have to make allowances for the fact that the physical conditions are quite different from what we're seeing in the deep sea. Tina suggests another possibility is from regenerating fragments. So I was noting earlier we saw all those piles of scleractinian rubble, and I said if it wedges itself in, it can continue to grow. So there's no reason that that could not also be the case for these ones. The one thing, though, Tina, about that observation is I'm seeing a lot of very nice holdfasts at the base. That is those discs that it's using to glue to the surface. That doesn't look to me like it's a coral fragment has reattached. I would think something would be a lot more sloppy on the bottom. But it's certainly a possibility, and we've seen it in other corals. And there's a good view there. You see the white, and then it turns into yellow-brown. So that's a good indication that this is definitely a Gorgonian sea fan as opposed to the precious coral that Chris was wondering at first. And some time ago, Tina Molitsova also said another thing you could look for are the siphonozoids. So I've pointed that out on some kinds of corals where you see these little sort of bumps, and those are highly reduced polyps, if you want, or zooids that don't have tentacles and are for kind of funneling water through the colony. They may brood eggs in there. The coralliids have those. The precious corals have those. These gorgonians don't have those. So that's another clue you can use to distinguish them. Gorgeous. Just a beautiful scene here.
So I'm not in the event log. Has Asako weighed in on what this could be? I'd just be curious to get her opinion.
All she said is she loves these coral gardens.
Come on, Asako. We need your opinion.
Let's see. I don't know that this is the Chaseon. But whatever it is, this one is looking after its personal grooming. It's a very clean shell. It's probably a relatively recent molt. I think I can also see the abdomen tipped down, and I think it's also carrying eggs. Asako would like to see us make a collection of one of those white things because she otherwise says we can't say anything. And I would agree with her. We only have like 2 minutes left in the dive, so I don't think that's going to happen. Those white fans would be fantastic. All right, we're going to try to get... Maybe not. Stand by.
We don't want to get greedy, Roland.
Oh, that's great.
Wait. I was typing. Did I miss the collection?
No, he's got it.
Yeah.
Wow.
He's got it in the basket.
Jeez.
It's got to go in the box yet, man.
Wow. Okay. I think our port side box is full.
Great suggestion, Asako, because there are a lot of these down here, so that's a good one.
And at the buzzer.
Wow. Wow, we are really spoiled. Josh, that was unbelievable. Thank you.
Starboard energy in it.
So we got a couple of amazing.
Did you cement that?
Yes, I did.
Oh, you did. Okay.
Yep.
Oh, you're just
as fast as them.
Yeah. Thought you were gearing up for another one already. So
just autopilot, just make sure I do that. Yeah, so we got really lucky. We got an additional 30 minutes of bottom time and then an additional collection there at the end. And this has been a terrific dive today.
Yeah, great. We declare victory.
Yep. I am not going to complain that we'll be writing dive reports for hours.
Well, we will complain, but it'll be worth it.
Not on the air. Not on the air.
Not on the air, though.
And you made it up to 860 meters?
We did.
Yeah.
Yeah, I mean, I think, so, we may not be on the perfect apex of this cone, but we certainly got up to the edge of the cone and saw exactly what we were hoping to see.
Yeah.
Clearly, the pilot said that the currents had increased. The Cecil suspension feeders were showing us that really good currents here. We saw all of those Oreos aggregating at the top of this feature. Just a real pleasure. And so I guess we will be having a post-dive call, is that correct?
That's right. So if you can just give us 5 minutes, we'll talk about tomorrow's dive. And we just get our friendly mapping lead set up here.
Thank you all for joining us today, and we'll be diving tomorrow to more than 4,000 meters deep,
That's
4
right.
kilometers deep, and tomorrow the geologist is going to be really excited, right?
I'm always excited.
Always excited, but tomorrow we're diving on a fracture zone, so something really different. Tune in then.
So you just have to do the post dive in 5 minutes.
Chris, just so you know, your mic is still open.
Oh, sorry. Okay.